Good morning, everybody. This is Schmidt Ocean Institute, research vessel Falkor, and ROV Sebastian. We are currently just getting on bottom on dive 427. Thank you for joining us this morning. I know it's early in a lot of parts of the world. It is for us too. It's just after midnight here in American Samoa time. We are just nearly at the dateline, but just before it. We are still working in the US EEZ, and we mapped a seamount over the course of the last 8 hours or so. It's actually kind of a double seamount area, and we'll put the map up here as we get farther into the dive. But we are going to be coming up along a ridge today, so lots of rocks and pretty steep slopes. The plan is to first start collecting some rocks and then see what we see along the track. So my name's Deborah Smith. I'm a marine technician here. We have Chris and Ben flying, and Steve is our scientist, for those of you who have followed along this expedition. And Steve will start the narration here in a little bit. But thank you for joining, and we hope you enjoy the dive. We should be in the water probably for the next 10 to 11 hours or so. So stop on by, ask your questions, say hi, and let us know where you're watching from. And we just want to say Happy Father's Day to all of those dads watching in the United States. Hope you have a good day of relaxing. And remember to call your dads, people.
Anything there you want to try to get?
No, I'm not liking how that looks.
Alright.
We'll keep going. But yeah, good try. I mean, these look like pretty good candidates, but sometimes it's just not gonna happen.
Are you still looking for the rocks?
Yeah, we're gonna keep looking for a rock. Alright, well, good morning everyone from Falkor. We are passing midnight here in the Central Pacific. And yeah, we are diving. We're diving 24 hours a day if we can, or at least have operations going 24 hours a day. My name is Steve Oskovich. I'm one of the scientists and watch leads on shore. Or, not on shore. My mind is on shore, it seems like. I usually say I'm on shore. I spent the last 2 Falkor cruises on the science party ashore, but fortunately I get to be out here this time. And I'm joined by...
My name is Anamere Dinava. I'm from Kiribati and I'm joining the scientist team at this time, which is a very lovely experience for me to be here today.
In the middle of the night. So we are going to be with you for the next 4 hours or so as we're diving the newly mapped, never before explored Seamount west of Howland and Baker Island.
Sea star.
Can we go to the left? There's like a pink blob right here. Just want to take a tighter zoom on that.
Wait, is this a cup coral?
This is I think this is a mushroom coral, but it
Mushroom?
looks very odd. So I think this is related to Anthomastus, or it is Anthomastus. Alright, I'm gonna go in. We can get lasers on the way out. I'm gonna zoom in first, but I'll do lasers before we leave. This is an interesting looking Anthomastus actually. It kind of looks like the stalked Anthomastus tahunotus, but it's a little bit different. The polyps are massive compared to the other one, and it's definitely very pinky.
There's something on it. Like,
Sometimes they have polychaete worms associated with the tentacles. Although it is very interesting. The polyps are much larger relative to the fleshy base part than we're typically used to seeing.
Umbellula.
Yeah, I would say Umbellula if it was in the sediment or something like that, but no, this is definitely Anthomastus. If you need me to jump out, just shout. Alright. I expect we'll see several of these. Good lasers, perfect. Alright. Looks good to me. Now we can come out and keep going. Tilt up a little bit. Yeah, anything, even a small rock, I guess, is better to have something in hand.
I can set up on the top of this larger mountains if any of these smaller single rocks are loose.
Yeah, I think that's okay. You know, we can always stop and pick up more rocks.
Okay.
You know,
So you wanna try any of these?
yeah, let's try them. You know, any of these look okay. It's deceptive. I'd like a rock something more like the size of these ones on the right, but you know, I know that
Alright.
at least 10 centimeters.
Set up on your map. See what we can do. No, because I don't want to put too much pressure on the wood for the set on it.
Yeah, so thanks for the commentary about the Anthomastus name. There's yeah, it's complicated. So you know, taxonomy is it's a bit of a moving target when it comes to some of these things because we are using new techniques and new you know, applying new thought to older grouping assignments, and Anthomastus was revised recently to a name change. I think most Anthomastus now belong to a genus called Heteropolypus, but it's just kind of one of these things that sticks in a lot of us who have been calling it Anthomastus forever. So we of course will use whatever the current name is, but it's just a matter of habit. But there's also a very, within the mushroom corals, there's also a number of things that look exactly like Anthomastus that on closer inspection, they might be a whole different genus entirely. Namely, there's a coral called Pseudoanthomastus, which looks very similar in seafloor video to Anthomastus or Heteropolypus, but it's different smaller differences within within the colony when looking at it under a microscope, for example. But we can use DNA barcoding and genetic sequences to help differentiate between species as well, and use that as one of the characters we do to delineate species.
What's that? Yeah.
So our dive plan for today, we are starting at around 20-50 meters, and we're working our way up a steep section at first that runs about probably 2/3 of our entire dive track, maybe, or definitely over a kilometer, at which point we'll kind of reach the top of a ridge, and we'll run up the ridge, which is a little bit less steep, but also might have different types of coral on it, coral and sponges and other types of fauna. One of the reasons we're trying to nudge these rocks a little bit is that we're trying to find stuff that's loose, or maybe stuff that can be easily broken, so we can get a sample. And the goal is to get something that's been in place for a while, that perhaps is more representative of the area, the depth that we're sampling at. Rather than something that perhaps might have fallen down slope and just kind of is deposited there from a totally different place where we might not know where it originated. That's alright, well let's go up a little bit and maybe things will change. There's nothing magical about this depth, but somewhere around 2,000 meters we'd like to get a rock before then. But a lot of these rocks are pretty heavily cemented, used in the hillside here, mostly due to this black iron manganese crust that precipitates out onto the rock. You saw earlier we cracked some off, and it's pretty light stuff compared to the rock in terms of mass. So you definitely know if you're holding a rock, or two rocks rather. One was pure basalt, which is the rock that composes the seamount, and one is crust. The crust will be substantially substantially lighter.
Wait, do you think like these rocks are pretty much the same with the ones in the river, like the ones in the river like super black?
You know, the a lot of the rock here is covered with that manganese crust, iron manganese crust.
Mm-hmm.
It doesn't really occur on land
Okay.
as much. It's something that precipitates from the seawater over long periods of time. I guess the other option is if you don't see anything loose, if there's anything that looks like you can get a good purchase on it and like crack it off, yeah.
You rigging it up then, or? No? You don't have the power? You don't have
Need the jackhammer of science out here.
So we're going up, yeah. Going up.
Yeah, we're going up. We want to try and grab a rock somewhere around 2,000 meters or below. Yeah, even like. Yeah. If you can get a good grip on that and just break off a chunk, we'll try. But yeah, go. I'm gonna take some images, but not sample images. I'm just gonna take some images so we know what it looks like, in case it does give way, that I can go back and reference them. Okay, go for it.
Okay, so they're trying to collect some samples from there.
What's that?
I haven't gone back to this since we did all of this, and all of this is being sent, these values being sent over to the So I actually think it flashes, which is annoying, so I don't have a way other than not.
Just grab it.
It's because it's refreshing
See if
the
you can just pick
website.
it. Grab it.
So that's constantly.
Every time you get an event, it refreshes the data, and it does this because the pictures aren't there, and then they come in and then the pictures are there.
I see what you're saying.
So it's what I would, problem is this machine doesn't talk to the rest of the network because it's a preference to use that. So if this was on here, I could make this be better, but it's still, it's like a early morning epiphany. But yes, I agree, we should
We're
work
gonna
on the
get a bigger,
overlay on the HD.
light on it.
Yeah,
It'll do it. Can you pull some more?
This is the time when me and Jack had to use everything.
Really?
Yeah, this is Tim's for the lab.
Oh, okay.
I'll make sure he gets it there.
Yeah, right.
Thanks.
Look around. Look around.
Let's give it maybe one more try, and then we'll go for a bit.
Alright.
I don't want to spend too much time on one area. Yeah. Yeah, we've tried over the years working out here, we've tried a number of strategies from crowbar, getting in there and cracking something off. We've also tried just ramming things with mixed results, usually more damage to the vehicle than to the rocks. Tried a lot of stuff. It's really hard, rocky substrate. But this is the exact type of rock we're interested in. It's just a matter of trying to get something that's manageable to get onto the vehicle is always a bit of a challenge. Got some sea urchins up there. These are deep water long spine urchins. You talking about this one here? What about this thing here? Is that big? It's kind of under the sediment, just the silhouette showing. No. All right. Well, let's pack it in and go up. And we'll just keep an eye out. Can't get the rocks. Sometimes it doesn't happen. There's lots of other cool stuff for us to look at. We're mostly interested in deep water corals and sponges. They like to colonize this type of environment. I actually might want to look at that unbranched coral in the distance on the upper left of the SciCam.
There's not many like corals appear in this area. Probably on top of it.
Yeah, there's. It's definitely
Just like very rocky and sandy.
there are some. It's kind of a perfect balance that a coral needs of good current flow and stable substrate. And the stable substrate is pretty obvious here. We can't move this stuff for beans. Question is the flow good? And it seems to be okay. There's a fair number of corals, more than I was expecting. Can I go ahead? All right. There it is. So we're looking at, looks like Candidella here, Crinoid coral.
Crinoid coral.
Yep. That's all right. I've got enough of it to make an ID. Yeah, we don't need to stop. Perfect. And then I'm just going to tilt up a little bit to get the quivers out of the frame. For reference, the lasers, the two green dots in the center of the screen are about 10 centimeters apart. So that coral was somewhere in the range of 60 centimeters or so.
It's dark.
Is that a shark? Or just a fish?
It's a fish.
Okay.
Yeah.
I thought it was a
Yeah, we
baby
can-
cat shark.
Do a quick, gonna do a quick look, and then we'll do a. I'll take a screen grab on Sea Log with the for our fish colleagues.
Oh.
Oops.
You can like be stuck in me, so you can't go anywhere.
Oh, he's right there. Oh, it's gone.
You're just scared of the.
Tried to escape, but he couldn't get past me, so he gave up.
There's one lone rock over there.
You're
there's
looking,
sea rocks right there, actually.
Yeah, I wonder.
There's two on top.
Yeah.
Those three.
So what we'll do, one more look at this guy, and then I'll set out and see what we can sit down and see if we can get another rocks.
All right. Yeah, I mean, don't worry about the fish too much. If he's gonna be darting all over, I want to put the rock in the box and move on. Let's hope. Feeling pretty good about this rock, but I said that about the last one, and the one before that, and the one before that.
So like,
Oh.
There's one below you, like,
Don't worry about it. I don't want to chase any more rocks right now. I think we need to start moving a little bit. It's a cucumber.
Sea cucumber.
Yeah. Holothurian. And then there's one more thing before you lift off. I want to grab an image. I saw a small coral right here. Right here. This can't be more than about 10 centimeters. So it looks to be a bamboo coral. So it's a really interesting bamboo coral from the perspective of it's so small, but it actually does have a branch on it, which is not really all that common of bamboo corals at this depth. They're often unbranched. How big is that? Tiny. Yeah. Nice. Okay. So we can get a good imagery of the polyps.
bring us in just a little bit closer to where we'll torch.
That's all right. All right, let's start moving up. I want to make some distance before we try for another rock. let's try and make it on our way towards waypoint 1. Just kind of at the most comfortable pace for
Wait,
you all.
is that a cucumber as well? Sea cucumber on the other side?
Yes, also, yeah.
It looks like it's blue or purple.
Both probably. I mean, very very close colors.
It's a big one.
That might be benthidites or something very similar.
Yeah.
That's one of the swimming sea cucumbers.
That's a cover raboya.
No rubble. No,
Okay.
there's some small bits of sediment and stuff.
Okay.
Flat. Similar.
Yeah. Tilt up a little bit. Lots of xenophyophores in the sediment here. Kind of characteristic of this depth. They're some of the most complex biological structures that are produced at this depth, in addition to corals and sometimes sponges. No evidence of sponges yet, but are probably going to be moving into the sponge territory soon, I think. But nevertheless, corals seem to be dominant in this entire area, both here and in the Phoenix Islands protected area, which we had the opportunity to go on a cruise and expedition to in 2017 on the ship Falkor. Bunch of fresh sediment there, washed down. Pretty steep. Any idea what the current's doing down here?
It's not too bad.
Any directionality?
It's coming from west east.
Cool, thanks.
Pretty much nothing in the area so far
West to south.
that can be found. It's just all rocks and sand. Oh, there's a fish right there. Is it a fish or?
Yeah, what is that?
Yeah.
Swimming there. Is that a shrimp? Let's go in. Ah, it's a lobster.
A lobster.
There it is.
Do you know what's the name of the lobster in this thing?
I don't know. We saw something like this yesterday, much shallower, but this is a deep sea. Yeah, we don't see those very often. They're kind of neat, but we do see them. Let's keep going.
We're making our way up up on top of the mountain.
We're seeing some pockets of bioturbation.
Oh, there's sea cucumber right there.
Mhm.
Another sea cucumber. We barely seen like animals in this area.
Yeah, it's a tough spot. The current is pretty slight to none here, and corals typically need, and sponges typically need a little bit of a faster current, since they're totally reliant on particles that drift down from the surface in the currents. And the stronger the current in a particular area, the more food you get per minute or second, and makes it a little bit easier for them to live at these depths if they have that energy source satisfied.
Is that a sponge?
I'm gonna just zoom in. You can keep flying just to get a closer look. Yep.
Oh, okay.
Sea cucumber.
Sea cucumber.
Yeah. Bit deceptive. Another sea cucumber. Yeah.
Whoa.
There's different types of sea cucumber.
Yeah. There's
A blue,
many.
pink.
Yeah. A lot of them
white.
are transparent. Yeah. You can see the intestines full of sediment.
Oh, there's a garter right there.
That one looks like another Candidella or maybe an unbranched bamboo coral.
Mm.
You might have some choices here, lots of good choices.
Yeah, I really want to rock, but wait, yeah, let's do a quick poke. I don't want to spend too much time since we've already spent about half hour, 40 minutes.
Yeah.
I'll take anything at this point, even if it's suboptimal. I mean, this is such a steep slope, like, I wouldn't expect anything to actually collect here. I would just expect it to keep on being shunted down slope. So anything that's left here is really well attached.
There's a fish right there.
Where at? Oh, see, that will explore real fast.
There's a dead coral.
Yeah, some sort of dead stalk. We don't need to zoom on the dead stalk.
Alright.
I'm gonna keep moving. We have a pretty ambitious, a little bit over 2 kilometer dive track today, and want to make sure that the other watches are in a good position to take their time. Let me bump one of these. Oh, there's it. All right. Can we image?
Alright, hold on.
Sure.
Finally.
Not the best, but good enough.
Much better than the other rocks we've collected so far.
Yeah. It's a good size too. It's just.
You may get two.
Yeah, we might actually do two. That's a good point. So let me image well first with lasers and can you hit the sample in situ button
Sample
or the sample? Yeah, for the photo.
here.
Yep. Grab that and then one more time. All right, and now you can set up to do that collection.
Okay.
So our first sample number is FK21-304.
304 rocks.
Yeah, it's going to go into the rock box.
Rock box. Any additional text?
Dark rock, approximately 15 centimeters long. We'll have some more information when we see the underside to see if there's any sort of alteration or something.
What is that supposed to mean? I'm not.
Yeah, do a quick shot. Alright. Good. Alright, let's stow it. Can you write worm tubes on it? Just write worm tubes, and sometimes they stay attached, sometimes they don't. It's a good rock though.
1, 2.
Torch out for you.
Might be alright.
Alright, you're on 4K bar.
This one. That's fine.
I know you're on 4K bar. You're already 50. Alright, yep. What's it? Alright, I'll stop. Try to shoulder up. That's not much. It's just a bad spot. Alright, there you go. Try to rotate. Alright, no, alright. I'll go back up. All the way out. Where the, all the ways. There you go. Should be good now. Oh. Before you do that. Happy Father's Day.
Very nice.
You guys.
So nice. Happy Father's Day, everyone. To the fathers.
Father's Day, Ben, and Father's Day, Steve.
Thank you. I thought we saw Constance out there earlier doing some
Yeah, you're fine, absolutely.
impromptu artwork.
Come on down.
Very nicely done.
Let's get that, but there's nothing goodbye.
I don't want to bend too far.
Just drop it. It's a rock.
Yeah, you could drop it.
You want
Good.
to try to get that other one?
Yeah, can you see if it's loose? Just curious. can hit submit on that one.
Is this okay? 1 tubes?
Yep, that's right.
Okay.
Just. Alright, can I, yeah,
It's
that,
crumbly.
yeah, that one's crumbly. I'm gonna leave that one. We got a good one though. Thanks. Just the one rock. Alright. One in the box, and let's make some way.
Going up, okay, we can see the sponge right there. Am I right?
Oh yeah, yeah, that's right.
Sponge.
Glass.
First sponge ever.
Glass sponge. Just gonna do a quick in.
A glass sponge.
Yeah, looks like Euplectella or some sort of euplectellid glass sponge. No associates that I can see. So sometimes these are shrimp or polychaete worms inside, which is a bit unusual. There's usually something living in there. Alright, I'm gonna pull out and keep going. So a lot of this this white discolored sediment looks to be result from bioturbation, which is just critters that are burrowing into the sediment, dislodging these streams of sediment from the holes that they create, and then that sediment kind of cascading down the side of the slope. A lot of the times these bioturbators are polychaete worms or other types of worms. But we are on a really steep cliff face, somewhere in the 40 to 60 degree range at any part of this dive. Seen a few corals and sponges so far, but I suspect it's current related why we're not seeing too much.
Stream.
This is a bit more intense terrain here. More vertical relief.
We're going higher and higher.
Yeah. No, that's how I think most of this is going to go. It's pretty much like climbing a vertical wall.
So, Steve, the current related is in too much or not enough? That's a question.
That's always it's always the question when it comes to, are the conditions right? Oftentimes, for animals that are feeding in the suspension, feeding in suspension in the fluids, they're capturing particles, marine snow particles that float by. And can we pop over back to the right on this cup coral here?
Yeah.
Oh, got it right here. I think I know what that is, and I can confirm with a zoom. If it is a cup coral, it might be a sponge. Let's see. Nope, it's a cup coral.
We're right above that as well.
Yeah. So the can I go in?
Yeah.
All right. Yeah, this is neat, and there's not many cup corals that look like this at this depth. I know what this is only because we collected one in 2017 in Pippa, but this is Vaunella multipalifera, and it's very wide, whereas a lot of cup corals are more thin stalked and tall at this depth. This is really good imagery. I can match this up with our collection from previous expedition. But I had this expertly identified by a colleague at the Smithsonian Museum of Natural History. And yeah, feeling pretty good about this specimen. All right, well, thanks for that. That was a great imagery.
See it in my sit cams.
Isn't too
On
much.
top of the rock here?
Or like, it is directly up.
Oh, yeah, good. Yeah, great.
A little mushroom top.
Right there. There it is.
Yep, I see it.
That one.
Yeah, alright, I'll fly it there.
Yeah, I don't have any more vertical on it.
Yeah.
Yeah, both of them are of interest to just take a look. See, just a matter of having the right flow conditions. I think oftentimes when you're at the bottom of a wall, like we were, when we stepped down on this dive, we ended up
Then Albert Dante's done. Got it.
Is that
that's an interesting critter up there. We've got it's a stalk tunicate, and the stalk is covered with all sorts of things like hydroids and zoanthids. I'm gonna take a look at this coral first. I don't think these are gonna be collection targets, so don't worry about that. But I just do want to image them. Is it a current thing or is it a
No,
toe
it's a-
thing? Getting a toe hold on it.
what color is that? It's alright.
Yeah, so this is They're always difficult, but I'm pretty sure this is a bubblegum coral, Chrysogorgia. Or am I a manual or autofocus?
I think you were auto. If anyone can switch that off of.
Thanks. Alright, I'm even gonna image the sea star, or brittle star first, since this is of interest to some folks on our team to get good imagery of these, even if collection's not really needed. But we sampled a couple of these a few days ago at Baker Island, so I'm not gonna sample this one again. Just get some good images of the ophiuroid. And then take a look at this black coral here, unbranched. What we're calling Stichopathes, but might actually represent many different types of black coral. Not all unbranched black corals are alike. Alright. That's good?
Okay.
Alright.
And then which is oh yeah,
Yeah.
something interesting for us to look at, or
It's just it's a tunicate. I'm not interested right now.
okay.
Yeah, let's move up. The zoanthids are cool, but we'll pass on them. I mean, personally, like from a personal perspective, but not not necessarily part of the science objectives.
Anyone prefer rock? Alright, go for it then. It's up to Steve.
We don't need any more rocks right now. Well, if we come across something that looks really obvious, we will. We spend a lot of time looking for rocks. I'm happy just to get the one for now.
Alright.
I feel like I need to put a bit of a certain distance between the dive start and before the next watch comes. Otherwise, I'll never gonna live it down.
5 minutes.
I know.
Quality.
Hey, I saved them the trouble of having to find a rock. They should thank me.
You know, the walls facing the line, so you want to start traversing instead of going up.
Yeah, I kind of just want to.
If you
I want
keep
to.
the distance.
Yeah, I know it. The line kind of goes parallel. You know,
Yeah.
kind of. Yeah.
So instead, just definitely start traversing. What do you want to do? Or we can keep climbing up. and see
Yeah.
what's there.
Let's keep climbing up. Yeah. So the vehicle, the head of the vehicle is oriented basically northwards. Is it northwest?
Yeah.
Okay.
Yeah.
So we're kind of crabbing along this face that's kind of north-south face.
Yep.
Okay. Yeah, because the ridge face is more like a southwest northeast. It's kind of got me a weird orientation here.
Smell it.
going back on there.
I'm gonna use it later, so I'm not gonna be in trouble.
I'm not sure if that would expect us. Hi. Oh, nice to have you.
What's up, Dad? Happy Father's Day.
Happy Father's Day.
Hey Dad, happy Father's Day. I just saw that you were watching. I did send him a text message, but voice is good. Happy Father's Day to all those fathers out there, my brother-in-laws and friends. So thanks and happy you're watching.
Oh, sea cucumber, another sea cucumber right
Rest of your
there.
parents were on over there.
I'm,
It makes sense watching. Feel that. Feel that there.
Bright color and weatherized, very see that?
Yep. Yeah, this is these are the depths, like 2,000 meters plus. These are the depths that the sea cucumbers rule. There's a certain depth where just like the amount of sea cucumber diversity skyrockets. I mean, I think if I wasn't studying corals, I'd probably be studying sea cucumbers, but they're definitely not as charismatic as corals. Although they can be.
Corals can't swim. Or escape.
No, that's good. That corals can't escape. That means you, well, okay. Escape if the current is not favorable. Those are heartbreaking, but yeah, we were talking about a really interesting occurrence we saw the other day, where a sea spider, a pycnogonid, was starting to eat a sea cucumber. Basically stuck its proboscis in, started slurping it up, kind of like a milkshake. I've never seen something like that before. I thought
Wait,
the
what
sea,
was eating? The cucumber was eating the what?
the sea spider was eating like a sea cucumber. Yeah, like
Cool.
one of the clear ones, a very
I thought
small
they were
one.
like yeah, okay. I thought it was a
No, it was definitely, yeah.
sea spider eating a sea cucumber. There's a lot of cut corals here.
Yeah, there are. Yeah, this is right. This is the depth for them too. This is great. Yeah, we do find cup corals this deep. Obviously not colonial ones. We're not seeing colonial corals down here. The environment's pretty stressful for them. It's more acidic than shallower waters, and it's basically harder for them to make calcium carbonate skeleton out of seawater. It's very energetically demanding at this depth, so we don't see too many organisms that are calcifying in that respect. We see a lot of other organisms that maybe don't have as much of a difficulty creating a skeleton, like black corals make a skeleton out of a protein, so it's not subject to the same exact stressors. And then the mineralogy of the calcium carbonate of octocorals is also a little bit different, and it's easier for them to sustain their skeleton at this depth.
Don't ask.
Long spine sea urchin.
What?
Don't even ask.
Ask what? No, I was looking at the time code to make sure that we keep forgetting to look before the dive, make sure it's 6. Because I was noticing the overlay, Nick's overlay is off by a second, which means I need to sync the photomosaic machine. Where that time's coming from. But yes, I know. That printer sounds like an alarming UPS. So it keeps triggering me to like panic towards something not working. Because it has that like same beep tone.
Tilt up a little bit. Cool.
That's like a coral right there.
That looks like an anemone. Can I do a quick,
Anemone?
just a quick in? Yeah, this looks like an anemone to me, possibly a corallivory, which is similar to anemones, but they're in a little bit of a different group. A lot of I feel like anemones get a little bit not enough love when it comes to deep sea exploration, deep sea science. They're kind of overlooked, but you know, they are just as important as the corals and sponges in this habitat. They represent a pretty significant amount of biomass. They're quite heavy, gelatinous. But yeah, if you ask a sea anemone biologist to identify something from video, oftentimes they will, oh, polychaete worm. Oftentimes they will not be able to give you a good answer, since a lot of the structures that are diagnostic for anemones are internal. So it requires doing a dissection or sectioning or something like that to look at internal anatomy. But great, thanks. Let's go on.
Yeah, at least it's mentioning that we did an entire expedition down in like Rubber Canyon, so only for uncut corals, so something we've been looking at.
Yeah.
Over the west.
The other thing,
Malcolm McCullough,
yeah.
group works mostly on.
cup corals.
Dead corals.
Huh?
Dead.
Well, they were looking for dead cup corals because they're looking at trying to date, you know, see climate change and things like that. So,
Yeah.
yeah, live and dead.
Yeah.
A lot of dead ones.
Collecting dead corals.
Yeah. You guys get really good at it though.
Very true.
There's a
Were
Falkor
they-
technique now.
Were they manganese coated or were they?
No,
Okay.
no.
There's a really cool, some really cool research that was being done several years ago. Probably still is. I see papers now and again.
What's that? A little orange thing?
It's Anthomastus, the mushroom coral.
Okay.
Yeah. Similar to the ones we seen earlier. Yeah.
Steve, like,
Yeah.
I always tell people I get like the, how you name things like slightly mixed up, but we say cup coral, that's like a common name, right? But
Yeah.
Sclerotinian is what this?
A sea star?
It's an order.
Order.
Yeah. It's on the same level as zoanthid or you know, black coral, Antipitherian. Yeah. Oh, look, that
That's the first
slime
sea
star. It has its the top part open. Name's escaping me right now. So it takes in water with that hole on top.
He closes his mouth.
Yep.
Yeah, not
Pretty much.
More for us.
Nope.
Cool.
Slime star. Yep. We were trying to get them to do that the other day. I forget which dive it was, but they were not cooperative.
To actually slime.
No, to open that.
Oh,
I don't know the exact anatomical structure. It's been a while since I've had to
orifice.
talk about sea stars. Yeah. So the interesting thing about cup corals is that where I did a lot of my master's work in the Southern Ocean, there are some pretty intense beds of fossil and pre-fossil cup corals, and they, in some cases, are so old that they're coated in this iron manganese crust that we see on the rock, and it basically encapsulates the calcium carbonate and protects it against alteration from the seawater. And what some folks are doing over in the UK, they are essentially using these as climate archives, like
Yeah.
you would use ice cores or you know, basically any other calcium carbonate structures produced. And it's really interesting because it basically seals and captures the state of the ocean at that depth when the coral was alive, and then after the manganese coat forms, it seals it like a nice little package, so that you know, from you date the cup coral using carbon dating, but you also can do radiochemical analyses.
I know they're looking to something similar at that McConnell lab, but but anyway, I just don't remember whether they were coated in manganese or not. But
Yeah,
it was Rummer Canyon and Lewin and all that, but I don't.
It's usually it's usually like the very high latitudes where that happens, like Southern Ocean. It happens more quickly. So I mean, it doesn't need to be that way. You can still get good information from pre-fossils.
She sent me an email at Christmas and said they had dated a number of things already from that cruise, and it was quite old, pretty low, and they upgraded.
Yeah. Bunch of nice rocks. I'm not going to zoom on any of these things. Seeing mostly mushroom corals now, but we've seen a few things so far. Oh, yep, another cusk.
Fish. It's a faceless, or assless, or whatever it is. Faceless?
Yeah.
Who was it? They were like a,
This
was the other one?
this
Was an ass?
Ash fish, yeah. It was
Now I'm just back and mashing them all
Yeah.
together. Everything was about this.
It would get automatically deleted.
Oh yeah, I'm sure. I'm sure.
Faceless cusk fish was what we had seen at like 4,000 meters, I think, for a while, wasn't it?
Gonna catch those nets.
I mean, was there a lot of rocky stuff at 4,000 meters over there?
Not a lot of rocky stuff. I mean,
Pretty
there's
soft.
some.
Yeah.
We were in a canyon, so there's definitely.
Yeah, you get that energy transfer.
Yeah.
Food moves down easier in canyons.
And they run down and roll down.
Can we zoom in right on the lasers? I saw like a branchy thing. I couldn't see it until it was center screen. Mm-hmm.
Wow, good catch. Like, runs in with the weight.
So we saw something similar to this. If this is a primnoid, then we sample something similar to this at a similar depth on a different dive. I think I can manage with that. Yeah. I just wanted to confirm something. Yeah, I think it's the same, and we have a piece of it. I just wanted to also check it for associates, and I don't see any. A lot of mucus on it. A good image, and yeah, it pretty much looks exactly like the one we sampled a couple of dives ago. But good. All right, ready. No instances of predation yet. That's something we're keeping our eye out for, is instances of coral predation or sponge predation. Up to the right, there's this weird blobby thing on the mud here. Doesn't look like a sea cucumber. If it is, we don't need to stop for it, but I want to double check to make sure it's not a sea pen or something like that.
I'm working on the colours, so it should be right with this.
I think it might be a glass sponge. Yeah, sponge. Okay, forget it. No, it's yeah, we got a good zoom on the last one. This one just looks like a smaller one. I'm surprised it's attached, honestly, to this kind of sedimented slope. There must be some pretty hard stuff under there. I'm gonna pan left to look off into the distance here, see if there's anything big. Sometimes you're diving up these walls like this, and it's really easy to be fixated on the substrate right in front of you, but if you look left or right, sometimes there can be just massive things in your peripherals that you totally miss. Kind of do another quick zoom on this stalk here. Looks like there's might be some polyp loss or tissue loss here that would be interesting to note.
It's a bamboo.
Exactly. Everyone's becoming so good with their coral IDs. I'm so proud. There. All right. Looks good. Thanks. Definitely starting to see more corals and stuff attached. That was like an old skeleton of a Primnoid coral. Pretty ragged looking. It looks like there's some evidence here for tissue loss by either primnoids or bamboo corals. But no evidence yet of predators. This one looks like a a stock that used to belong to Candidella, and it's completely denuded of polyps. Might have been a bamboo, actually. There's some nodes in there.
Interested in the background stuff?
That's the same as the one we imaged a few seconds ago, so we can move on from that. If we see any of those, though, with brittle stars or anything attached to that, it will be definitely a set-down
Is
imaging.
there a very huge star on the other side?
Yeah, I think it's a Brisingid sea star.
Okay.
A lot of urchins, these Aspidodiadematids, deep sea long spine urchins. I really like this boulder terrain. Usually you can find some corals on this, either on the tops or sides. And what do you know?
Oh.
So there's actually, I'm a little interested. So right below at the base of this Metallogorgia is a very small, what might be a juvenile Metallogorgia, a very early stage that I want to image. And then we'll look at the larger one later. But I want to get a good handle on that baby Metallogorgia because I've never seen one that small. It's kind of noteworthy. Wait, iris down when you have a chance to, or ISO down?
ISO down.
Can't get any closer with the top.
That's alright. I can zoom in once you're okay with me going in. So,
I tried to set up right.
so I think this is a small Metallogorgia, which is the larger colony next to it. I've never seen one this small, and can I have manual focus when you have a chance? Is it manual?
Auto focus. Oh, yes.
Thanks. Yeah, this I'm wondering if this is actually Metallogorgia, or if this could be a small Iridogorgia. It's not really easy to tell. They're in the same family. I'm gonna infer that it's Metallogorgia here because I've seen two of these small baby colonies on this rock next to the larger one. Interestingly, there is no associate, no ophiuroid brittle star associate that I can see in this one, which is interesting because usually from a very, very young age they don't have that associate, but there is a small worm tube about halfway up the colony. This is really interesting to note. I'm sure Tim will be excited to see that because usually even from a few centimeters you find that these ophiuroids are bonded with these corals for life. No, we're not going to get there. Alright, can we float up and take a look at the larger
Yeah.
colony?
Right.
Yep. I'm just going to want to look at the brittle star in the middle. So we're not going to need to focus on the coral, but just the brittle star on the inside of the colony.
Is this a Chrysogorgia?
This is a Chrysogorgia in the same family, but it's a genus called Metallogorgia. There it is. hold that for 2 seconds. Focus a little bit better. Alright, great, thanks. Can we go take a look at that coral in the background? The white fan?
The Crinoid.
Yeah, we're gonna take a look at it. You can throw it in auto when you have a chance. I think I lost the focus. If it's in a hole, I can try and zoom the best I can in here. Interesting, there might be some possible predation here. I see some zoanthids already on this colony, which are usually early colonizers of tissue. Early colonizers of skeleton that's been denuded of tissue. This is interesting. So it looks like we've got some
Crinoids.
Yeah,
It's
couple
a
of
star.
couple of crinoids, couple of brittle stars. Aplocophrin here, which seems to be doing most of the grazing. This is an interesting coral colony, just from a colony morphology perspective. Alright, I'm gonna go tight in on this predator here. This looks to be an Aplocophrin type of mollusk. Yeah. Yeah, so we have a couple different types of corals here. One is a zoanthid, which has the very large polyps there, and then the other is the octocoral, probably Neurella, by my approximation. Interestingly, this Aplocophrin is kind of on an area where we wouldn't expect it to be. We wouldn't really expect it to be with the zoanthids, but it seems like it is, and it seems like it may be eating zoanthid tissue, but that's not clear at the moment. And then these ophiuroids, I don't see any other Aplocophrins here. What is that? That looks like a jelly. That is kind of an interesting priority collection item. So while we've been trying to image, can't tell if it's actually just stuck on the coral or if it's actually one of these predatory jellyfish. While we've been trying to image and collect data about coral predators, we've always been on the lookout for these elusive jellies that are attached to these corals. It's not clear what they're doing, but if we could collect that jelly on the coral, that would be awesome.
In the coral with it.
Yeah, like grab a bunch of branches and maybe get that. I'm not sure how sticky it is. It's not something we collect very often, so I can't give any guidance. But maybe just grabbing enough of the branches, it will ensure that it'll stay on. All right, I think we've imaged this well enough, so we can start our collection protocol. I'm gonna go first do a specimen shot like this with laser. Perfect. All right, hit the specimen.
This one?
Yep. And then I'm gonna go tight in on the jelly here.
Okay, one more.
Yep, one more. All right. So I think we're gonna sample maybe somewhere around here. I'm not sure how stuck this thing is, so the jelly is right here roughly. So you know, maybe something like cutting it here.
Okay.
That's what I'm thinking. And then we can put it in a quiver.
Can you focus on zoom in on the jelly again?
Yep.
I got it.
This will be collection number 305. And we're going to aim for a quiver, but I don't know which one yet. And the text description is going to be Narella, Narella, with jelly.
Number 16? Or
So there are a number of ophiuroid brittle stars that we, those pink stars that are attached to the coral. It's pretty sure, I'm pretty sure that they're not feeding on the coral. I think we might have dislodged the jelly.
I feel
It's
like the jellyfish is
floating around.
Yeah.
Can we go up? I think it's behind the harm.
Which arm? Either one?
Yeah, I think it's gone. Which makes me think it probably wasn't stuck on there very well to begin with, but well, since we're already set up, yeah, can we grab a piece? I guess we'll grab the same area that we had planned.
about over here, Steve.
Yep, that sounds good. Yeah, something that'll definitely fit in the quiver.
How's that?
That looks great. They should break fairly easily. Yeah, perfect.
Is that quiver 9
This is gonna go, I think, in 16.
or 16?
Yeah. great, thanks. I'm gonna keep going onwards and upwards. Hop over back to the right for just a second. Right here there was a white piece of shell. Can I take a closer look at that? Is there a chitin? Yeah, I'd say that's what it is. It's a type of mollusk related to snails and clams. Neat. Usually, sometimes you can see trails associated with these things, but this one seems to have been in place for quite a while, but it's a neat little creature. There's basically armored plates on its back. Polypocophora. Very nice. Okay, thanks. Haven't seen one of those in a while. Certainly not at this dive. Can you add a comment? So go down and find the sample entry here.
This one?
That one, right there. Add a comment. Yeah, right there. And say like lost jellies, swim away. Just so they're not looking for the jelly later on.
Okay, lost cherries now.
Yeah, something like that. And tilt up just a touch. That's perfect. Seems like there's lots of rock candidates now. We're having trouble in the beginning. I'm not gonna pick any up right now. I'm gonna go maybe another 100 or so meters vertical before we try again. But it seems like there's plenty of pickings here. Anything interesting left or right, or is it pretty much just sediment out there?
So it's kind of sediment that
Yeah.
comes off from that side.
Yeah.
Come over lateral to coral.
I mean, this is still interesting. Just curious if there's anything we're not seeing in the distance.
Just zigzag up to it.
Yeah, a little bit of side to side, it's okay. We are expecting to get more biodiversity as we move shallower. It just, we've had some pretty amazing dives where we've had crazy corals at 2,000 meters plus, even 1,800. But I think that the steepness of the slope here, and possibly the current regime is resulting in fewer corals than we might expect. Namely, we're kind of diving on the inside of a little alcove between two ridges, and it seems like the water is a little stagnant in here, and it's not flowing as fast as perhaps over the top of a seamount or over the top of a ridge. That's my hypothesis, but it could be something else too. There could be something about the substrate requirements here, or depth alone could also be a factor. But we are seeing corals. It's just that it's the abundance that's not. That's kind of surprising, or the lack of abundance. Nice anthomastus. I do want to check the depth range on anthomastus that I have here, because that one looks a little bit out of its depth range, but I just want to double check. Oh, it seems right. 1,300 to 2,100. Again, these euplectylid glass sponges. Really liking the sediment at this site. Not understanding why. Look at that thing. It's a bit fuzzy. Can't make it out. It doesn't have a lot of contrast. If it turns out to just be sponge goo, I can move on. But just want to make sure it's not some sort of coral structure hidden. Nope. Yep, just a bunch of sponge fibers, glass fibers. Okay, thanks.
How's that right?
I don't have any more tablets. Tablets. I've just been
there's like sea cucumber loves to stay in this area.
There
Pretty
are a
much.
lot. Yeah, it's kind of their realm. They're mostly eating all of this organic material in the water column that settles onto the sediment. But if there's so much organic material, there's a lot of sea cucumbers here. I don't know why there aren't more suspension feeders. I'm guessing it has something to do with the supply. One of the things we are looking at and cross-comparing between sites is the effect of geomorphology on different types of deep water communities, like corals and sponges. So we have examined in the past the effect of different feature types and geomorphologies, like whether a seamount is a ridge shape or whether it's a conical shaped, or maybe it's a guyot, which is a flat-topped seamount, and or even if certain seamounts have access to shallower depths, in which case they identify as a bank, or in the most extreme case an island. There are some differences we've noted, but we're always trying to increase our sample size so we can make those patterns, make those tests a little bit more clear. But generally, if you were to go to, for example, compare this site with an island like Baker or Howland Island to the east, you would generally find pretty similar species of corals, although their abundances may vary, just because of different terrain variables like topography, but also possibly things like food inputs might be more substantial around islands than, say, in a seamount.
Oh, there's another sponge. Fish.
How fast are we moving roughly over ground now? 0.1? Okay, 0.2 is fine. Yeah.
So it's like between 0.1 and 0.2.
Yeah. I think we're doing pretty good. We'll be on the seafloor for at least another 10 hours or so, I think.
Just let me come ahead. We can run part of it around. It's not a problem. We do that.
I think in areas, if this sediment turns out to be more extensive, we can just kind of scoot right by it.
Okay.
We're not going to be looking at anything in the mud.
Just roll over here.
Another slime star. Pretty decrepit looking glass sponge. Probably not alive. There's some more, a little bit more relief. Still similar corals, Stichopathes, black coral. That's just a big boulder. Sometimes on the back sides of these large boulder falls, you can see corals growing where there's water moving pretty quickly around some of the contours of the boulder. Nothing really here though. About how far have we gone, distance, since the on bottom?
We have gone approximately 240 meters.
Okay. 100 meters an hour, something like that.
240 meters since we started.
Yeah.
Yeah, the track is,
Well,
if I just go as the crow flies, then yeah, it was like 215 up,
Yeah.
so.
we didn't get to see that.
I mean, we're on pace. Yeah.
I can show you, Steve, if you want. That's the houses.
Yeah. Yeah.
We're in a little bit, looks kind of a little bit like a hole here.
Yeah.
So
It's gonna get steeper.
it's shady, but yeah.
Yep. That looks good. No complaints there. I think you know, folks wanted to pick this track on purpose because of the steepness and the continuous nature of the slope. I'm imagining the shallower we get, we'll probably get some more interesting currents that wrap around the top of this ridge. That might lead to some more interesting corals. We are kind of moving into a depth range, though, or moving across a depth range that we are that we have not sampled well. We've been focusing mostly either on the very shallows or on the shallower than about 1,000 meters, or in the 1,800 to 2,500 meter depth range. We're going to be crossing that today, going up to about 1,200 meters. So we should be getting a good handle on the transitions that are occurring. Can we go look at that star on the left-hand right-hand side that we just passed? We might want to do a collection here for Tim, since this looks to be like a brittle star. Suction. Yeah. Yeah. So this is a brittle star that we typically find on coral associates, and it's not on one, which is why it's kind of interesting. And he's interested in a little bit about the biology of these coral associates when they're not on corals. But we'll. Oh, you know what? I think it tricked me. We'll do a quick zoom in anyway, because this is actually pretty interesting. But this actually might be a Brisingid. I thought it was an Asteroschema or something. In which case, it's a totally different group than brittle stars. So probably no sample, but just the imagery.
Okay, that's stable. Should I close out that window?
So sharp.
You know what? I don't know. You know what? This is I'm a little confused because it looks like a Brisingid, but it also looks like a brittle star, and it has the symmetry of a brittle star. I'm really confused about this one. But it has the spines of a Brisingid, so I'm... are lasers there somewhere? Yeah. Okay, lasers there, and then I'm going in to the disc. Looks like an Ophiacanthid, though. It's just weird enough. I changed my mind again. I think we want to collect it just to be safe.
It's pretty
Yeah,
flexible.
the body is. I just don't know how flexible the spines are. So that's my only thought.
Pick it up,
Yeah, you can try and pick it up. Yeah.
put in bio box.
You think it can go in a quiver? I don't know.
Tries to escape, but we're good at it, that's why.
Yeah. All right. Well, let's see what we can do.
Okay.
Do you need a sample?
Yeah. Can you hit this sample in situ?
Are you happy with the pictures?
Yep. Yeah. We got one there. Let me just get one more tight
Yeah,
end.
sure. I don't know if you want to adjust the light.
No, I think it looks great. Yeah. So this is
Okay.
the way it's moving is tells me this is an ophiuroid. Yeah. Hit that right there. Simple in situ one more time. Okay. Now we can go for collection. So this will be sample 306.
306.
And do you prefer if I pick this up at the end that tip of one of the tentacles?
You can try, but I think I think you're gonna have to kind of cradle it a little bit because because its arms might pop off. But you can try and grab it any which way first, and then if it falls off, we'll reevaluate.
Give her my fingers, you mean?
That's not good. Let's see if I can put the finger on it. That's right.
You zoom in a little bit. So it's stabilized.
We're gonna call this one.
So we haven't seen one like this before.
I've never seen something like this. I think
Such long spines.
it's interesting to me because there are some things called Brisingid sea stars, but there's also some ophiuroids that look. I mean, this is an ophiuroid. The way it's moving, that's pretty obvious, and it's an Ophiacanthid for sure. Which is a family of very spiny. He's not going very far, so yeah, you can chase him off. So definitely not a Brisingid, by the way it's moving. So it's an Ophiacanthid, and it's an unusual ophiocanthed because we often associate ophiocanthids with the coral colonies, like the one we sampled a while back, that Norella, and I part of me really wants to slurp it, but I know that if we slurp it, it's gonna get stuck. Yeah, so try and do what we can to cradle it and get it into a box or quiver.
Wait, that's the one, yeah?
Yeah, that's right. It fell down just a little bit.
I think it could just be one of his tentacles, so, like, that's all I see.
Yeah, if you can pick one up, I mean, the ultimate goal is to get at least an arm and the center disc. If it starts losing legs, the center disc is the most important thing. Yeah. Yeah.
Do
We-
you want this?
Yeah, we can take it. That's what I was afraid that's going to happen. Kind of, if it's possible to get under it and cradle it, it's moving. But I suppose if it loses enough arms, I suppose you could just slurp it.
Oh, it was like that.
So this is going to go in B. Let's see how much more we can get.
Okay.
Can I just type in the
Yeah,
name here?
sure.
It'll be easier to organize and settle it.
Okay. And it went in BioBox B, and you can trigger it now, and we'll see how much more we can get. At least we got something.
Maybe slurp it.
Yeah, let's slurp it.
And we can try and, it'll just go easier with some rather than drop it in biology.
Yeah, exactly. Yeah, if it doesn't go in the tube, we'll just, yeah, that sounds good, like a good idea. It's just weird enough that I can justify spending time on this.
Very nice, you can see how it goes.
The spines are really useful for wedging into the rock, I guess. Yeah, I think you got it.
Let's try and grab that.
Do you just go as a shake? So, yeah, that's it. Do you just the treasure? Yeah.
Whoa, sticky.
So hold it down. Yeah. Clean that.
It's climbing.
Voluntarily.
Trying to get more vertical, so I can take the rest of the day.
So sticky.
Yeah, it's very spiny. We're gonna have to be careful when we handle that one on deck. Very nice.
You probably do have a little bit in that section sample though, right? That little arm that fell off,
What jar was queued up?
Just gonna check.
just so you know.
Okay. Yeah, I
Jar
noticed that.
7.
Jar 7 might have a farm piece. Okay.
Okay, ready to submit 306, yeah?
Yeah, you can.
By a box B.
Right.
Yeah, jar 7's good. Here's the arm there.
Got it. Yeah. Can you put a comment on that entry and just say small piece of arm in jar 7?
Yeah.
Okay. Small piece of arm in jar 7.
Yeah. Excellent. Thank you.
Wait, let's do one that's it.
We'll talk about that a little bit, Steve.
Yeah. It's a common question is, do these things stay alive by the time they get to the surface? And the answer is it depends, and it depends a lot on the depth of their collection. For the most part, pressure has really no effect that we know of, or visible effect, on invertebrates. However, things like fishes that have gas bladders or air-filled spaces would probably experience some barotrauma associated with changes in pressure. But one of the things that does affect a lot of the collections down here are drastic changes in temperature. Corals, for example, if they're brought up into warmer temperatures, will start to produce profuse amounts of mucus, or
Sponges too will do that. But we were noticing on some of our collections from our shallow dives yesterday, they did just fine, and they actually, the polyps of the coral came out again. They were quite happy from our collections at around 300 meters. So generally, the shallower the collection, the more likely they are to be happy at what 1 atmosphere of pressure on the surface.
Tilt up a little bit. 19. Pretty steep stuff here. Not a ton of attached critters. So here, in the lab we set up, we have 3 different types of refrigeration. We have a refrigerator for keeping things cool and running some of our experiments. No different than the temperature of the refrigerators probably in your homes. But we also have 2 types of freezers. One is a minus 30 degrees C freezer, which is it's a pretty cold freezer. Definitely colder than what you probably have at your home. But that's kind of our standard freezer. And then we have a minus 80 degrees C freezer, which we use to store a lot of our cryogenic collections, which is usually like archive specimens. Can I look at this thing right here? And those are usually, that freezer is usually kept for small amounts of material that we need to keep ultra cold. For example, if we're looking at something that is time or temperature sensitive, like DNA, we might want to keep it at a colder temperature. But for the most part, the ophiuroid will probably survive just fine until we get to the surface, and we'll decide how we want to process it then, if we want to keep it alive, or if we want to preserve it right away. We'll let our associate expert, Tim Shank, is it an overhang there? All right, I'm going to push in and try and get some imagery of it. And it looks like a crinoid. I just want to, some kind of crinoid. That's perfect. All right, great, thanks. So it looks like this isn't even the steepest part of this dive track we drew today.
Seems like the steepest part's going to come between here and waypoint 3 or 4. So it's pretty impressive that we're seeing this kind of verticality now. Maybe the next watch might get the dramatic incline. Well, we made a few collections so far, mostly some interesting findings, some rocks. There's yep, stalked crinoids. We're coming into the depth of those stalked crinoids. We saw them at one site, and we haven't seen them again. And that's surprising because they, are sometimes as dominant as corals in certain parts of the seamount landscape. Wow, little caves. I love that. These look like collapsed pillows. Kind of has this dome. Collapsed pillow lavas or pillow basalts. I wonder how the sediment gets in there.
Yeah.
Don't you? It looks pretty uniform too. A lot of these stalks and things that we're seeing are probably old sponge stalks or maybe coral stalks that have zoanthids and things growing on them. I'm not seeing a lot of live, vibrant corals in this area. Stalked crinoids also could be the source. Take a closer look at the base of this colony here with this brittle star on the black coral. We do not have any liquid nitrogen producing capacity on the ship right now. We loaded up in Honolulu with a lot of liquid nitrogen to preserve things cold things very quickly, and then we keep them at minus 80 for longer if we need to. Thanks, going in. All right, nice Ophiocanthid there. No idea if it's similar or not to the one we previously collected. These ones are jumpers. They're not. I mean, they occasionally will attach here, but oftentimes you see them floating down the colony. They also have a tendency to fall off pretty readily. This one's pretty big, isn't it? Yeah. Is there anything else up here? Horrible to focus on these
thin ones. Yeah. I don't want to take the whole coral because it's like really wiry. But I was thinking of getting a sample of this, but I don't know a good way to get it without putting it in a bio box, and I don't want to use the box space right now.
Unless you just want to take a snap.
I would want to get the brittle star with it.
Right.
So I think we'll pass on this one.
Keep an eye out.
Yeah, we'll keep an eye out. It's just this was the same coral that we had lost in this slurp a few days ago, or last week or what.
You could get it at the base and get that small section in bio box.
Yeah, it doesn't want to use it.
Yeah. No, I don't want to use the bio box because that would be another. I can't put this collection in bio box B because it's the same type of brittle star as the one we just collected, and it might be confusing.
I guess it's gonna move pretty fast once we start moving, huh?
So he wants to do that section.
Yeah.
I know. I think if you snip it on top and snip it at the bottom, that you might fly away right.
It could.
Yeah.
Yeah. Yeah, I'm just trying to figure out a way how to best.
Put it in a quiver, you cut the top off, and then cut at the bottom, put it in a quiver.
Oh yeah, that could work.
Because it's not kill it.
Yeah, you'll lose a little bit of coral. If you think you can do it, yeah, let's try that. Obviously, it's not going to be easy to put the whole coral in there, so
We're gonna have to
get whatever we can. Yeah. Or you know what? How about we do this? Why don't we just put it in there, like that, with the brittle star down, and then just jam the stopper on top, and then whatever hangs out hangs out.
Say that again?
So put the coral in there, and then whatever hangs out of the quiver hangs out. You just put the stopper on top.
Yeah, but it's kind of long and then it might swim out. You could cut it halfway and then try to jam it so they don't do stay long. Just keep it long. How long is that? I think it's
Over a meter.
over a meter. That's a
Yeah,
long one.
yeah, I know.
We cut half of it maybe.
Yeah. I don't want to waste it, but
No. It's gonna be hard spiraling.
yeah, I know.
You can take a marker and then do a little scout around.
Yeah. Let's. I'll take a note of that.
Put a marker.
It's a little bit of a complicated collection, so I think I'm okay passing on this one for now.
Just ask him to climb up already.
Give it a nudge.
Please, sea star, climb up. Coral. So we can go at you.
Okay.
1351.
Go there just in case.
All right. Thanks. All right. Let's keep going. It's just that with these black corals, they sometimes have these associates, but not always. And I think we got a good enough imagery.
You already have a brittle star on the other one. You could put the top half of that coral in the brittle star.
Yeah, it's the same family, and trying to tell them apart could be problematic, especially if they start all hopping arms off,
Yeah.
because they get stressed out. Yeah, it was definitely a risky collection because the chances of that thing just bailing from that coral were very high. And that I didn't want to. If I can't get both, I'd rather have none. I guess that's the moral of that story. Yeah. I think that's going to be one of the most common corals we see on these steeps.
Is that a Crinoid?
What is that? Yeah, I think that's an Iridogorgia.
Okay.
We're just gonna do a quick scan of the colony, look for associates. We don't need to do a set down. Yep. Oh, yeah, there we go. Iridogorgia, Magnospiralis, and Metallogorgia. We may want to do a close-up of the brittle star on the Metallogorgia, though. Yeah, so we can move over that way when you're ready.
Which is the one with the stalk and then the fan at the end?
This one right here?
Yeah.
That's Metallogorgia. If you were to scrape away some of the tissue on the skeletal part, it has this metallic iridescence quality to it. Most of the Chrysogorgias do. They're called golden corals for a reason. Some are more golden than others. Metallogorgia is quite black and metallic. Yeah, I think we might need to come around a little bit more. Yep. It's one of those swinging in the breeze kind of corals. So I find it interesting, these guys typically orient such that the base is more up current and kind of this tuft of polyps is oriented down current. This suggests that, well, I mean, it's not wrong. The current is coming upslope. There we go. So I'm not going to be able to identify the ophiuroid as well as Tim can, but we'll hold this for about 10 seconds. Try and get at this. The center disc is a little hidden, but that's okay. We can do what we can.
Do you need a picture for that?
Yeah, you can write in coral, click the Metallogorgia button, and hit brittle star ophiuroid with the associate.
Is this one?
Yeah, Chrysogorgia, yeah, right, that one. And then
This one?
brittle star. Yeah.
Are you gonna sample this?
No, just imagery.
You might move, can you mute for a second?
Spiny sea star.
They have spines.
Yeah, a lot of them have spines or scaly
Mm-hmm.
plates. This deep. Alright. With lasers. There we go. Alright, and out. Great.
just curious, are there any ways to actually edit those reports or what goes into that afterwards, or is it kind of...
I can't remove it once that button's been pushed.
Yeah.
I mean, I can, well, that's not true. I can delete it. We could probably go back and delete it. I have, like, it's not ideal, but
Yeah.
I can. But what you can do right now is go back and add a comment and say this was not sampled.
Yeah.
So like if you scroll up in the events here, if you just scroll back to those sample in situs, which
Yep.
you can see, like you just click older events, or you can even filter by sample.
Yeah, so hit in there, type sample,
Then you could look at the latest sample, like so.
hit enter.
Um,
So this is right.
and then the little bubble
Just
next
put
to it.
a comment, yeah, put here, and here, and these two bubbles, and say not sampled, just so we have that on record.
I'm going to zoom in on the stalk. I don't think there's anything living there, but I just wanted to get a closer look on the flyby. It looks like zoanthids. Only zoanthids don't get that much love, unfortunately, since they're kind of invaders. All right. Good. Thanks.
Yeah.
Any animal coffee on those?
I'm gonna go down and eat, so I'm good now.
I'll take one, how's your bag?
Coffee?
I'll come through that.
How do you want?
Right.
Bridge or RV, I just moved with them.
It's pretty light. Yeah.
What, more rocks?
Yeah.
There's more rocks.
I was just thinking that actually.
Is the planes too small?
So the general criteria for the rock is we're looking for something that's looks like it's pretty much in place, but not
Yeah, it is.
too in place that it's not movable.
Yeah, I see.
Obviously, we don't want any of this like debris that ends up in a pile somewhere. But we're also looking for things that are bumpy and lumpy, not so much angular. The bumpy lumpy signifies it's like very crusty. But these are pretty deceptive too. You don't know if it's held together like concrete down below. Yeah, I can take a quick look. I think it's we're entering the Stichopathes zone again. I noticed this on one of the previous dives, is that with Stichopathes there's these two morphs that kind of co-occur. And I was wondering if they are just maybe older and younger. One is like has a green color, one is usually reddish bronze, something like that. But I think we're up to both color morphs now on this cruise, which is great because we didn't get any in 2017. Good. Let's keep going. Yeah. So for rocks, let me see if I can point out something that is resembling what we're kind of looking for. Yeah, I think something like this size, 10, 15 centimeters is good size rock. We don't want flat rocks, so something kind of blocky looking, but not too angular, if that makes sense.
Fly low, I'll push that and see what happens.
Chop the edges.
Fly low, I'll push that and see what happens.
What about
Walk around slats.
how about this one right here,
There you go.
Yep.
right under the coral there, where the lasers are?
He's a winner. I'll spec my interest in
Oh wait, hold on. Can we?
that one.
No, the one right there. It's got.
That it's got stuff on it?
Oh,
Yeah,
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 that was the
yeah.
bottom as well.
Yes, yes, good job. Force multiplier there. If that has some life on it, especially on the underside, that's a
It's like it's like that, like that.
interesting.
Like that's,
I'm just gonna take a look first.
Oh, how bad would you ever go flying away?
Oh yes, that is jackpot.
That is the one.
That is what I want.
Now I'm not stressed if I can move it. Alright, I'm gonna do this and do this.
I mean, yeah, I'm kind of torn because like I don't want it to go in the rock box, you know, with the biology, but I guess that's okay for
Put
this
it
time.
in.
Well, let's see how big it is, folks, 'cause it's
Yeah.
a little bit bigger than I think it is.
Zoom out for it real quick.
Yeah, it's big. If you can break it, I mean that's great too. Try and avoid the soft corals.
We
But-
avoid this off.
As well, like avoid like squishing them as best as possible. I know it's
Try and scoop it
big
out.
hydraulic arm versus soft-bodied, you know, 2 centimeter
That's
tall
what-
coral.
Break it off. Pick it up at the edge and try to plug it out.
Let's think of the sand is the biggest one.
Hold on.
If it is loose, I will image it too.
Okay. Zoom in a little bit further.
Look down to it. Just a touch. Yep. Move.
Do you think that'll fit in the bio box? Or no, not bio box, rock box?
Yeah, fit the rock box.
Okay, let's image it then.
Campus free.
All right. So,
That's watching
yep.
those.
Now we can hit this sample in situ button.
Okay.
What's up?
All right, I'm going back in, and we're gonna do a tight zoom on the coral.
Is it recorded? The picture? I have no idea what
It is, yeah, it is. Tight zoom, sample.
Another sample.
Yep. And now, yeah. I mean, I'm not going to stress about this because I think we have this stoloniferin from another sample. So I'm fine with just putting it in the rock box and then, yeah, whatever we get from it, we get from it. So ready when you are. So I hit sample and then the sample number is going to be FK21 dash.
307.
All right. Rock box is going to mean the main sample, and then the coral is going to be the associate. But just write large 30 centimeter rock.
So
Yeah.
the best way for me to be able to get this,
Yeah.
like, you know, top maybe, would be to crush all those associates on the bottom of it. So I'm gonna try to get it to the top.
Yeah, I mean, there's lots of them around the rock, but you know, anything you can do to reduce the
Yeah, damage.
damage is helpful.
Okay, large, 30 centimeter.
Yep, with attached coral polyps. I mean, it might even be bigger than 30 centimeters, so we'll adjust that, but just leave that there for now.
Don't give me slab.
There might be some bonus biology on this.
Tourist.
Yeah.
That's a rock sample.
That is a workout
That's
there.
gonna definitely affect our buoyancy.
Well, you
I wouldn't go
asked
that
for it.
Down now, anyway. That's for sure.
Alright. Workout phone?
I'm just gonna bring the racket back. Towards me.
Wanna take a look at it first?
Yeah, let's go in and image.
Alright.
I mean, this might not be the best rock for crust, but it also checks a bunch of boxes at the same time. So I'm gonna call this one a little bit of everything for everyone. But yeah, I really like these stoloniferins. It's really hard to get these.
Go break it, go home.
Yep. And love when I can do this. All right,
Alright.
all set, ready to go.
Right, watch out.
It depends. If it's mostly crust, it might not be as heavy as you think.
Oh, I'm gonna find it.
Yeah, the way that I'm folding it, it appears it's not that heavy.
Yeah. And it's certainly not brittle, but it is a little altered.
Let go of it.
Okay.
Alright, I'll move that. Well, alright, that's it.
Perfect. Did it.
Not bad.
Very nice.
Plenty of room for another
We do have to keep a little bit of space for one more rock somewhere up in the 1,000, towards the end of the dive.
Go off it or rock.
one. Oh, you know, there's loads of room there.
Yeah. At least one or two. I think we'll do one or two in the beginning and then maybe one or two at the end. Alright, let's move a bit.
Second.
That's by myself. Yeah, this is.
Right.
Yep. That is
Mushroom.
yeah, it's a mushroom coral.
And the Mestis.
Yes. Just trying to figure out if there's anything attached to it. Doesn't look like it. There might be something attached to the back. Something in the background.
That was smart, right?
Yeah, I don't know what that is. Worth a look.
I think it's a different.
I think so too, yeah.
Are these sponges?
Yep, there's a little bit of sponge. You can see the difference between the live and the dead sponge. The live sponge is kind of pearlescent white. Alright, so we have here, going the wrong way. Is it an autofocus or a manual?
Never mind.
Okay. I'm having trouble focusing.
Hold on one second.
Try it out.
Try it.
It looks like it's coming around slowly.
Use the one back.
Yeah, there it is.
One is off.
Great. It's kind of a weird angle.
Yeah.
Go a little bit more.
She really got
Yeah, I mean, the interesting thing about this coral is that the ophiuroid is so disproportionate in size to the coral. And there is actually an apelocophoran at the base, which itself looks partially denuded of polyps. Can we move around and look at this on a different? Yeah.
I'll try and get around the back.
Yeah.
That's right.
It may be a collection target, but if that helps. decide which side you want to be on.
So I think probably sun is a second to
Sure.
says just think that
Going in. I think we got it, yeah. Okay, so this is some kind of aplicophoran mollusk thing. Aplicophoran. So this looks like maybe 2 colonies, but one on brittle star, which is not unusual for the coral. They kind of grow like this. This is pretty small. Do you think we could suction up everything here? Like the worm, the mollusk, the coral, like scrape it off the rock, and then the brittle star?
I don't see what it is.
That would be, yeah, my preference, just to get it all in one
Alright.
jar. Let me image it right here first. This in situ image there. And
I'm surprised that it doesn't show over here the image.
it does. Sometimes it's just a little delayed.
Oh, okay.
One more.
Okay.
All right. And bonus points if you can grab that little cup coral
Okay.
Was
In-
that in the,
in the same section.
okay.
Yeah.
No problem. This is on it. Right. Can I bring the camera back to this so you can see it there?
Is it number 308?
That is correct. And it will go into one of the jars, I think.
Okay. Any additional tips?
Plexaurid, Plexaurd, with brittle star,
Twitter star.
Aplicophoran, APLAC, we'll just call it aplac, and cup coral. And hopefully we'll get all of those, but if we miss one of them, we'll just take it out of the text there.
Okay.
Is it on? Okay, pumps are on.
Okay. The coral should come up. You know, it's not really super well calcified. It's a little stringy, but looks like you got half of it. The other one is just the base part of it.
Okay. Nope. Zoom in a little bit first so you can see.
There, good, perfect.
And then the cup coral.
It's in right under the sponge.
Yep, I just made sure it's all in there first. So that's the colonies in there, right?
Oh, the one up top, gotcha.
So you
Yep, got it.
that little black guy there.
We don't want the,
Don't want the sponge.
We don't want the sponge, but yeah, if some comes along, we won't complain. I think you got it.
You get the base.
Don't worry about the base, yeah.
Okay.
Just the top part's fine. And there it
That's
is. Yep, got it all. So that's jar
That is jar 6, yeah.
jar 6.
That's
Good to go.
Good to go.
okay.
Thanks.
It's done.
This is another one of those brittle stars that we spend all the time chasing down. It's good to know that. Yeah, can I just image it while we're here?
Yeah, yeah.
No, I don't want to get it. We have one. But it's unusual enough that this one is a bit darker coloration, just unusual enough to warrant a second look. Can you autofocus me? I feel like I'm lost it a bit.
Let's try it manually. See if we can.
The,
Not getting anything.
I'll put this out and I'll see that helps. Okay, some tips.
Yeah, thank you. Perfect. Yeah, looks like the same one, just a little bit darker pigmentation.
So that was 6. So with these, see the arm there, those small sections, white. I'm
Yep.
assuming it's lost a bit, and that's the regrowth.
Exactly. Yeah, that's the interpretation that I'm going with. Yep. All right, lovely.
When I come off here, I need to just take my turn.
Yep. Sure.
So I'll just be quick.
The question is why we're interested in crossing the rocks.
So for the crusty rocks, we are partnering with some scientists at the U.S. Geological Survey who are interested in trying to identify just patterns of iron manganese crust in particularly U.S. waters. We are interested in that data because we're interested in the settling preferences of corals and sponges on those crusts, and if they like to settle on a certain type of crust or a certain composition, or if there's some sort of relationship between crust thickness and coral communities. So it's really just a multi-dimensional collaboration we have with our colleagues at USGS. There's also interest not just in the
crust themselves, but the rocks that are within the crust. And so a lot of the rocks that that these crusts are precipitating on are old basalt, cooled lava, basically, rocks from the volcanism that created this seamount. And that basalt can be dated, which gives us an idea of how old the seamount might be. And how old the seamount might be tells us a little bit of something about the motion of tectonic plates in the Pacific and hotspot trajectories across the Pacific over millions and millions of years. Can't be vertical all the time, right? Sea cucumbers gotta eat too. Yeah. So we're about half an hour left on our shift here before the next team takes over. Try and set them up in a good place so that they can keep trekking up the wall here. We've covered about 200 meters of vertical and maybe a couple hundred, maybe 300 meters so far, or approaching that, of horizontal distance. We can scoot faster over the soft sediment, whatever you're comfortable with. We're not going to be zooming on stuff here. Standing orders are to move quickly across soft stuff. On the very rare chance we see a sea pen, I might say something, but they're pretty rare in this type of environment. I've not been seeing very many sea pens as I thought I would be seeing. They are here. They're usually just very small, but most of what we're seeing right now are xenophyophores and phytodetritus on the seafloor, just bits of clumps of marine snow, a couple of sea stars here and again. Quite a bit of bioturbation too. This is something we've been noticing for pretty much since the start of the dive. These small white mounds. Speaking of white, can we do a white balance when you have a chance? And the small white mounds are bioturbation, which just a fancy word that means there's animals in the sediment that are making holes and burrows, and they're sending up They're extracting all the sediment from the seafloor and creating these small little mounds or burrows of fresh sediment on top, so it looks. Oh, halosaur. Yep. Yeah, let's take a look at these because we haven't seen very many fishes, and while this is probably one of the more common deep water fishes at this depth, we're gonna go in quick before the mud. All right. I kind of saw what I wanted to see. So there's a few different genera within this family of fishes. Oh, there you go. All right, well, coming back out. So halosaurs is means salt lizards, or salt animal. Interestingly, I'm not quite sure why they're named that. It must have something to do with just the characteristics of their body shape and morphology. It makes them look lizard-like, but for the most part, there's two species or two different genera that can be easily differentiated on camera. A couple of the characteristics being the scaliness of certain parts of their body, and namely their head. That one seemed to. Oh, nice sponge. Yep, no. That's alright. We imaged it a bit deeper, but yeah, still that's nice. There's a couple of polychaete worms in there too.
Nice eublectellid. Now, oops. I didn't know they were so loosely held in there. There are actually some pretty fine spines on this thing. I haven't seen a eublectellid quite like that. Alright. Since I don't have many more opportunities, do you think we can collect this sponge and put it in a quiver? Looks to be about 12 centimeters tall and maybe 5-6 centimeters wide. We've seen enough of them, so one of the things that we're collecting, as well as like the rare and unusual, we're collecting characteristic species of the environment that we're in, and this sponge we've seen several times so far, and I would describe it as characteristic of this depth range. And before we move out of this depth range, I want to make sure we have a sponge sample, since that is useful to several members of our team, in that we're interested in the diversity of the sponges, as well as can I go in for one more image?
One more sample instance.
Yeah, let me frame it up first. Wait a second for some of the mud to clear. We're interested in the diversity of the sponges, of course, but we are also interested in the microbial communities that live on and around the sponge. And so we're gonna try and culture some microbes from some of these sponges as well when we get them back up on deck.
Hit it? No, yeah.
Hold on. I mean, the currents feels like it's a little bit stronger here. It should take just a couple seconds to clear.
clearing now. You can hit it. Hit the in situ button.
Let's get a boot jet first.
Alright.
So about your camera, be safe.
Hit it one more time. All right.
We have some good images from before the snowstorm, so we'll just replace them with that stuff. So this is FK21. So I think it's not held in by anything. It's just a bunch of glass fibers. So maybe just like plucking it.
Yeah.
Yeah. 21-309.
Should be fine. You hear how it goes into the
Nope. Upside, downside, any side. Yep. So quiver 15, and then the family, I'll type it in.
That's good. See it if we get some image.
And you can submit it.
Really good. That's how you were playing?
It's going. Nice collection. Excellent. Tilt up just a little bit.
Another rock.
Nope. Good on rocks for a while.
Won't get rocks.
No, it's one of my favorite things.
Yeah. You just like manhandle
recommendations we have were for maybe 1 or 2 rocks per dive at 2,000 plus, and then 1 or 2,000, or 1 or 2 rocks at somewhere around 1,000 meters, 1,500. We want to get a good depth range just so we can see like how crust rates are accumulating across different depth zones.
I think we should have a dive and put rocks oxygen on the entire portion of rocks.
That's why we did kangaroos.
Yeah.
I think you might have some takers in the geology community, but
They're underrepresented out here, I believe.
I think some of the best cruises I've been on have been with partnering with geologists. You get so much more of a holistic idea of like what the heck's going on down here, because they're able to tell you exactly what kind of rock this is, how it was formed, why you have certain types of sedimentation going on in certain areas and not in others. They're really interested in the rocks. They call it my biology contamination, but that's okay. The only downside about working with geologists is that bio boxes are kind of useless. I mean, they can hold rocks, but they don't have any need to keep things at temperature.
Yeah. We have plenty of milk crates.
Yeah, milk crates. That's the thing.
The entire thing freezes away. Ice melt. Fucking grab that rock.
Tilt up it just a tiny bit more. Yep. Yeah, I think we moved up into a bamboo zone here, because that's most of what I'm seeing. I mean, we still can see a lot of the corals. We're not out of the zone where we have a lot of corals that are dissimilar from what we saw down below. Around 1,500 meters, there seems to be a bit of a transition that's associated with the oceanography, some of the water mass boundaries in the area. But it's kind of a blurry boundary. There's a lot of species that cross that transition pretty readily. I'm seeing a few small bamboo corals, but nothing that we haven't seen so far. So, you notice like how this like crusty stuff is layered there.
Yeah.
I think that's a lot of the rock that we got. Is just like these terraces that break apart. I don't know if that's good or not, but it's something at least. Just past 1,800 meters, so we're now in the 1,700s. Still coming up through, well, somewhere around 1,200 meters we'll end our dive today. Trying to cruise as quickly as possible through this soft sediment, so we can get to some of
steeper slopes, but even when you're planning a dive on 40 to 60 degree slopes, there's no real way of being absolutely certain what kind of substrate you're going to be looking at. It's totally possible to have sedimented slopes in the 40 to 50 degree range, which are pretty impressive in their own right, but just not always what we're looking for. Can I zoom on that little stem there, the red stem?
Yeah.
And yes, it will be a collection target. It's a sea pen. I'm not sure if this is going to be like slurpable, but probably grab it, quiver near the base. Yeah, somewhere, even getting in the mud. Yeah.
Yeah, well, I'm saying, here's what we have.
So this I have.
14. Real soft.
It's okay. Take your time. This will bring us, probably pretty close to the watch change.
Yeah, it's okay. Just,
So this is going to be a sea pen called Predation. It's not one we have very many collections for. It's kind of rare, usual, and I'm very, very happy to see it.
Is it another sample?
Yes, we're going to do some in situ photos the first one
Okay.
sediment clears, so hold off on that.
Down with the camera. You got the yearly chance to sit down with the
Something.
Yeah,
I'll take about 2 minutes, just sucking through it. I'm just
Yep.
using the afterwards.
Sure. Yeah, I don't mind. Good pictures of these things are useful.
Particular area, unfortunately. Sometimes it's useful.
Yeah.
Little current, very soft.
I'm gonna get the camera in position for when it clears. Can you do one in situ photo?
Sure.
And let me frame it up. All right, do a second one. Lovely. And
Can I drop my ISO again just to see how sharp
Yeah, you can try it.
Yeah, it's not really much.
It's tough. Yeah, it's not a lot of contrast.
Yeah.
Instead of
I like it a little bit brighter. I think that's
Yeah, I
okay.
think that's
Yeah.
usually good.
All right, and one more in situ photo. All right.
I hope it's already.
It is taking them. Yeah, it doesn't always update, but for example, I can see it. If you reload the page, you'll see it. Yeah. Yeah, I see it here.
Okay, cool.
Yep.
I'm just worried, like.
No, it's pretty reliable, but sometimes it hangs up. So FK21-310,
Okay.
and it's gonna go in quiver 1-4.
Let's see. Do you have any additional text?
Yeah, it's a Protoptilum. I will write it in here. Haven't done any eDNA yet, but I think maybe I don't want to.
Morning.
I don't want to use the eDNA bottles yet. Oh yeah, I think we'll take a water sample right after this.
Alright.
So, Niskin bottles 4, is that a commission still?
It's fixed.
Okay, perfect. I think it might go into the sediment something like maybe 2-3 inches maximum.
Alright.
But we would like to get that
Oh,
intact. We don't want to cut it, yeah.
Yep.
Just
Alright.
gonna come forward a bit.
Yep, that's fine.
Whoa, whoa. Good catch. I've never seen them do that so fast.
I'm gonna dig to get it. Sorry. I think I got him. No. That thing took off.
Wow. That was fast. I didn't think it would do that.
It's gone?
Can
It,
we dig around a little bit?
yeah.
I mean, it's not going to keep going. It's going to stop eventually. I hope. Should have used the push cord, maybe.
That thing was tricky. I think it actually went down into the hard substrate underneath. Which I can't get into. It pushes us off.
You think,
Forced up the wall.
I don't know.
That's it. Let's see if we can look.
Yeah, back up a bit. Is that part of it, or is that something? I think.
Were you looking?
No, it was just a shadow. I thought it was part of the underside of it sticking out.
Try to zoom that way, just once, see.
If this doesn't work, I don't want to waste too much time. We could scan around. Maybe there's one nearby.
Yeah.
They're usually pretty. They're like that, where if you find one, you might find two.
Just got beaten by a coral.
Oh, that's a bummer. I learned something about that sea pen though.
Yeah.
That they are fast. Never thought I would say that.
Good to know.
Do I just cancel this?
Yeah, cancel that one. And on the pre-sample photos, so
There's
here, add a where the little comment bubble is. In these two photos, just say no collection.
Well, if you see any more red stick sea pens while you're in the mud, they're really obvious. You can't miss them.
Can you give us a rundown of what's
Yeah. we've seen a lot of these Sticopathes. We've seen a lot of Tanthomastus.
Okay.
We've seen bamboos, and Crinoids are pretty rare, but they're there too. Narella mostly.
Okie dokie. Thanks. Yeah, we can just
Yeah.
keep going, yeah.
Yeah, I was just giving you a little view of that.
Yeah, that's great. That's perfect.
It was the only thing I haven't seen in for a while.
Okay.
The sand.
That's a good, oh, these are good.
Yeah, dude.
I added a checkbox for peninsulas.
yeah.
So if you do see any, you know,
Oh, there you go.
Go for it, especially since we're in the sediment. Might as
There's
well
a
get
sea
something.
cucumber. Was that the first red? That little red one? Was that the first one you'd
Yeah,
seen?
yeah, we saw it in 2017, and it was never collected. So, pretty much any sea pens you find are of interest, since they were really poorly represented last time.
Okay.
With the exception of Anthoptylan, we don't need that much more Anthoptylan.
Okay. I'm gonna just tilt up a little, Russ. Is that okay? Xenophyophores.
See you in the morning.
Okay, step up.
Later on.
There's a halosaur. Sounds good.
No predation so far. Just some apocophorans, but really no like clear evidence of predation by anything on an octocoral.
Okay. Let's see. So we've just had a science watch change. My name is Alexis Weinig. I am a research scientist with the USGS.
And good morning, everyone. I'm Brian Kennedy. I'm a marine ecologist with Boston University.
And we'll be on with you guys for the next 4 hours or so. It's 4 a.m. here. And we started this dive. We got in the water around 10:45 yesterday for us, local time. And we've been diving on this previously unmapped, unexplored seamount in the U.S. EEZ around Howland and Baker. We spent the last couple days at Howland and Baker proper, doing multiple dives on both islands, and then we transitioned. We transited to out west, so we're kind of on the western edge of the U.S. EEZ, but out of the PRIMNM. Here's some rocks coming up.
Chasing the rocks.
Chasing the rocks.
we take just a quick zoom? We don't need to land.
Yeah, I'll just hold it again.
Great. Just let me know if I can zoom in.
Whenever you're ready.
Okay. Looks like a bamboo coral. Just a small bamboo. That's good. Thanks, Russ. Snap a blanket.
Probably, yeah.
Tripod fish.
This is kind of the most bioturbation we've seen in settlements, I think, most time.
Do you want to explain what
Easy.
bioturbation means?
Sure, yeah. So what we're looking at is the different color changes and, you know, the shape of the sediment here from different animals moving through the sediment, eating it, turning it over, which is a super important process in the deep sea. Basically, the same thing earthworms do in your garden or your backyard, where they go through and eat up and turn the soil and aerate it. The same thing happens in the deep sea with all kinds of different creatures that live both on the surface of the sediment and deeper in the sediment. And we've actually not spent a lot of time over this kind of deeper deep sea mud most of this expedition yet. What I find interesting is we're not seeing a lot of obvious bioturbators like the sea cucumbers here, so a lot of them must be buried into the sediment,
Sounds like it'd be rocks.
or
Yeah.
We can just kind of
or might be
sonar-
old.
Yeah,
And
we can
these
just, like,
areas are
pump
so
the rocks by
stable that
sonar.
the effects of this feeding trace of one sea cucumber could probably still be visible for, honestly, decades to hundreds of years potentially.
And at the start of the dive, we were about probably around 2,050 meters in water depth, and we're currently at 1,730 meters. We're working our way up to, we'll look at this sponge real quick. A small little glass sponge. No, that's fine. Small little stalked glass sponge. How's that, Brian?
Yep, that's all I need.
Okay.
Thanks.
Sponge dead ahead as well.
Oh yeah,
Dead.
that's okay. We'll take a look at it. Yeah, it looks like another glass sponge, but this one looks like it's dead. Has some associates living on it, a couple crinoids. And they have that really dark, sandy color. That's what we consider a dead sponge. Probably has just been covered in sediment over time. Oh, I got some bigger rocks coming up.
Well, I'll tell you, gotta trim it down. Just to keep us safe. Try and easy. So I've just been adjusting that so to the lowest they say that feel reasonable rate. So I have to just chase it on to it.
Good morning, Ben.
Just watch the deviation from the ship. I find it interesting, yeah, right?
Couple more of those stickopathes. Urchin. Yeah, we're just kind of going along, but we can use the sonar to look for rocks. And, you know, as long as we're not too far off our track, we're good to do that. Come up a bit. Got any interesting facts for us, Brian?
Sure I do, but nothing comes to mind right at the top of my head. I have to admit I'm still waking up a bit.
Yeah, fair enough. Lots of xenophyophores in this
Yeah.
sediment.
You want to explain what a xenophyophore is?
You can't.
So xenophyophores are really cool. They're one of the largest single-celled organisms we know of. And so when you point, they're about, an inch, maybe 2 inches across, but they're all one cell in these really complex shapes sometimes, these honeycomb patterns. And so they're kind of an amazing, strange piece of biology that normally we think of cells being microscopic
Take a quick
and
zoom on this
hard to
brittle star on that
very
rock.
small, and that's governed by a whole bunch of types of cellular mechanisms, gas diffusion, stuff like that generally limit the size of an individual cell, but xenophyophores have kind of found a way around a lot of those things to the point that they can grow so large without any kind of cellular cooperation like you would expect in
Okay.
almost every other organism.
That's great.
Hey, people.
Deb!
Welcome to the science control.
Thank you. How's it been going?
Oh, lots of rocks. We love rocks, right, Chris?
I love
Did anybody
rocks.
get Ben's pilot change?
Nope.
You were wrong.
That brings the morning energy.
Yes, thank you, Deb.
Huh?
So we said you bring the morning energy.
Well, it's like my lunchtime.
Well, I know.
Okay, pilot change. How long have you been there, Ben? 30 seconds.
No.
Dang it.
I already admitted it was too hard for you.
You didn't say it was hard.
You said I would love to fly, but it's way too hard on
It's
how you made you guys do it. I couldn't
We
possibly
can
do
keep
it.
going.
Right
That's
here.
a just it looks like a dead coral stock. Actually, to the
Only
left
time I've
here,
ever flown
then
over the
looks
bottom
like
was
something
flat, so
we'll take a look at.
it wasn't vertical.
Yeah.
Our first big coral, big-ish coral for the day, or for our watch. It's okay.
Coral. Some kind of color.
It's an interesting perspective, how it's like in a valley.
Channel, yeah.
About the rocks that we got, now I have Jennifer Lopez on
Oh,
some.
You gonna sing for us, Chris?
I'm trying to get rid of it, try to spread it.
Can't sing copyright, we get thrown off.
Oh,
Yep, that's the reason. we're not
Shazam is
singing.
That's the
definitely
reason.
gonna.
Yeah, that's the reason.
It's gonna match Chris's voice exactly.
it's good.
I feel like it's Jennifer Lopez, but like angelic. Oh. It's on right now, but it's on right now.
So it's got a fair amount of movement.
Yeah, so that's actually helpful for us to see. It looks like it's a Paragorgia based on the way the skeleton is swaying in the breeze, if you will. So there's two corals that can have very similar coloration, or two families of corals that can have similar coloration. But the skeletons have very different structure, and one way we can tell the difference is if the skeleton starts to sway. That means that it's from the family Paragorgidae versus from the family Corallidae. And you can see also these polyps have started to retract, and at the tips of each branch there's like a ball, and it looks like bubblegum. So the common name for this coral is the bubblegum coral. And it has a Astroschema sea star, which we've pretty commonly
Brittle star.
seen. I'm sorry, brittle star. Well, the pink helps with the bubblegum, really. But yeah, it looks like bubblegum. And there's another one of these same rock inhabiting ophroids, which are pretty interesting. I wonder if that's what Steve sampled.
No, we have one, it had the super spines on it, like spiny
Oh, that's
spinies.
yeah. Spiny spinies, okay.
Instead of these little ridges, there
Yeah.
were long spines sticking out of the.
We'll just take a really good look at the oral disc. And then looks like there's a sea star hanging out at the underside of this rock. See anybody else around here that we can look at? Nope. I think that's good. Thanks. Ooh, can I take a look at what's on the seafloor right here? I can just snap zoom. We probably don't have to land. Sounds good. Okay.
It is a crinoid.
I think it's a sad crinoid.
I'm not used to seeing the unstalked crinoids sitting in the sediment like that.
No, not at all. And all of
Sitting on
its
that itty bitty tiny
strange looking.
It looks like a crinoid from the structure of, or maybe it's stalked.
Is it a
Oh.
stalk that got buried?
Oh, no, maybe. It's not like lifting on. It does kind of, it has the structure of a stalked.
It does.
So maybe it has a very short stalk. Actually, we don't know how deep the sediment is. Oh,
No, it's been
not stalked.
severed.
Yeah, it's been. And also the branches look like they have been predated. They
Yeah,
don't look.
It looks like something might have bitten off the top of a stalked
There's a
crinoid
little-
and dropped it.
What would do that, Brian?
No idea, actually.
I want to look at what this little rock up here. We have a little baby Anthomastus, mushroom coral. Very like sticky sediment. You can see when it is, the particulate, some of the particulate in the water is from the ROV setting down. That's good. We can carry on. And when it gets kicked up, you can see it's like in big chunks, not like very fine, silty sediment. Lots of kind of rocky outcrops here. It looks like maybe some of these have kind of tumbled down over probably hundreds of thousands of years. Looks like to the right we might have some more rocks coming up.
So corals in the deep sea really
Wow, that's
require
big.
rocks to live on. So one of the things you'll see us doing in terrain like this is really trying to find the rocks.
Stalked
So
crinoid.
we're looking out in the distance and using the vehicle's forward-looking sonar, looking for these little rock outcroppings, because that's where we're most likely to find deep sea
Looks
corals
like
and
there's a
sponges.
coral right. If we can take a look at that rock. Where's my pointy stick? Thanks. Yeah, looks like we have a Primnoid octocoral, which Steve was saying we've seen a couple of on this dive. There's something on the one of those lower branches. Very small brittle star.
A couple crinoids as well.
Yes. Very interesting growth where it's like split off and symmetrical on both sides. Yeah, yeah. It's not a very strong current, huh?
No, it's not.
Just spread the food around. Just spread
Can I have manual zoom for?
There you go.
Thanks. Let's see if we can come back.
Don't worry, I don't want this, but just check Slack.
Oh. I got it. I got it.
It's
That's what I was It's a silliness. Meaning green is not checked.
meaning that I think that if you could hit it there, it then changes how
So this is Calyptrophora antilla.
switches. Yeah.
Polyps are all facing up. Okay, that's great. Thanks. You can carry on.
So still work over there on that control.
Must that make sense?
How many cruises ago?
Summer 2020.
Oh, I haven't read this. Okay. I would think if we saw this again, that's what he tried to sample,
Actually,
that we would slurp it.
camera down. There it goes. That's some of the problems of brightness of the lights.
Do you want to fire this one?
Yeah, we can. But as a, yeah. Let's take a look at that coral up there. To the right, yes. I'm just gonna come in. We don't need to land.
You guys underneath? That's fine.
Looks like a bamboo coral. Has two branches. That's great. We can keep on.
There's another
Oh, there's
one off
another
the
one
top
over here.
left.
You thinking about meat? What? You should just
Looks like another bamboo, but all the polyps are on one side. It's very fleshy. Okay. That's good. Thanks, Ben.
like you had something in the galley, but did they like
Now last
move it?
July,
Yeah.
last July.
It's like a stalked crinoid.
In
Definitely a.
Steve is saying something about collecting them.
Oh, but we'll keep an eye out for the next if we see it again. Yeah, go ahead and keep going. Thanks.
I did Jared, it seemed to do something in storage. Hi.
Our smoke corner, the van.
you part of the meat smoking crew, Chris?
I'm the barbecue babysitter when J-Rod used to go to bed.
Okay. I noticed the
He cooks well.
he what?
He cooks well.
He cooks well. I noticed those like grilling gloves in the RV room, and I put them on, and I was like, Cody, what do you guys use these for? And he was like, grilling. I was like, cool. I was just like, I just figured it was like
For
some
the tool, use as the tool.
yeah. It's like-
No, we didn't. Yeah, we.
Not like the kitchen brush of science.
right.
Or the bathroom brush.
Toilet brush. Yeah. I just assumed there was like an ROV use for them.
Yeah. Yeah, we need to eat. We need to eat.
That's how they feed Sebastian. He takes big chunks. It's like feeding a lion.
He's hungry after a 12-hour dive.
Yeah, I wish. This is the part of the movie with the giant sea creature appears. Let's have it.
Oh, actually, right here. Yeah. Sorry. Kind of blends in with the sediment. I'm gonna try and zoom in without you having to sit down all the way, just because of the sediment. It seems to be kicking up.
Okay.
Oh, goodness, I didn't see that one. I was
Right.
going past it.
I would say plexaurid. It's very light colored with a big astroschema. Oh, Steve says this is the one they collected earlier. Okay, great. So now we know. Oh, some slime. Steve, shouldn't you be back in bed?
We noticed yesterday we were having a science meeting on the ship that the parents on board seem to need a substantially less sleep than those of us who are not parents.
That's not true. Speak for yourself.
I don't know. I think it's true.
I don't need more sleep. I'm a robot. Whoa, very wiggly armed crinoid.
Is it? Look at that stalk.
What do you mean?
Look at the base. It doesn't. I mean, it's crinoid arms, but
I think it's a crinoid. It's just very flex.
it's a very different crinoid.
It is very flexy.
Keep going.
Keep going, yeah. I'll try and get out of our own cloud. That is strange.
Yeah, that was a very
Like-
different looking crinoid.
Crinoids are very hard to preserve, so we try to
My other guess would have been.
not collect them as much as possible.
Okay, well.
They fall apart once they come to the surface pretty quickly.
I think that worked. It was looking like somebody like built a giant guitar or something. I'm trying to move to that. Or just
This one?
up right now. No, the wind's not blowing.
I keep moving. Or her. How are we up there right now?
Tony.
Tony?
Tony.
Yeah, okay.
Huh?
Yeah, there's a little whip coral right there. Wow, there really is not much current, is there? It just sits, that sediment, once you kick it up.
Plus the sediment is very sticky. So
Yes.
It sticks in the bottom of the ROV, so
Right.
it floats around. Comes off. There's fishy easily get to the bottom left.
That's good. Eel. Coming up. Yeah, I see it in yours.
I can't hear you.
It's okay. We can keep going. I think he's gonna elude us. It's interesting, all the rubble, like all the rock is right on the, we're right on the track.
Well, means we picked a good dive track. I guess it is Father's Day, right? I guess
Oh,
we should
yeah.
say Happy Father's Day to everyone
Happy
on shore.
Father's Day, everybody. Oh, I saw her doing this last night.
Reaching out.
Porching.
We're sleeping. Reaching out.
Another one of those strange crinoids.
Oh, yeah. Yay! Happy Father's Day.
And note from Sebastian to everyone.
Happy Father's Day to all the dads and dad figures. Guess when they were writing it, Tim was like, Happy Fat Hers Day. Thanks.
Happy Fat Thursday.
Hopefully everybody's having a good Sunday. Whatever that looks like for you. Or Monday if you're already into Monday.
This is our Monday on the other side of the dateline.
Yeah.
Not by much.
We're very close to the international dateline. We thought about what it would take to run over there and cross the equator at the international dateline. Huh? Oops. But we calculated what it was to go down there and back. It's too much.
Yeah, the dateline and the 180th
Okay, can we take a look at
meridian
this
are different
crinoid?
here.
This is a similar to what we've been seeing.
That's really different. I've never seen one quite like that.
Very long arms. And like only five of them.
Which artist or you
Gullercrinus.
Steve's saying that's Gillercrinus.
Okay, that's good. Thanks, Ben.
Swan dress was
Keep going.
Huber, the Icelandic pop star.
Well, according to Steve, I have seen this one before on another expedition that he and I were on together, but I don't remember it.
They all run together.
Steve has this ridiculous photographic memory for deep sea organisms.
Just gonna take a quick zoom at this sea star that's right in front of us. Just hanging out on the sediment. Steve thinking. That's good. Thanks, Ben. Yeah, looks like another crinoid. This looks like the one that we just saw that was
Yeah, that was
detached.
had a Crinoidevry? Crinoidevry?
Yes. Looks like a Walteria sponge on the left. Right here. It's like translucent. A lot of little ophroids living in there, which is pretty common for us to see. That's good. We can ID that. Is this pretty vertical? Yeah. No?
You can sense that looking at that camera right there.
Oh, okay.
Straight down. So.
Yeah. Yeah. Getting two different angles over there. Do you want to zoom on? Can we zoom on this little pink thing?
It is.
I know that's right. Let's take a quick look. It's probably a sea cucumber.
Yeah, but it's on a rock, which is a little unusual.
There's still sediment on the rocks here.
Fair.
Oops, it's a little too fast, sorry.
Yeah. So I can see.
Clinging on, sea cucumber. Okay, that's good. I think that sea cucumber is like, whoa, the current really picked up.
Yeah. It's like a
Predatory
Predatory tunicate.
tunicate. Right here. just take a quick. These are. Yep, you can see the gut inside. There we go. That's good.
If you were gonna get last week, I thought it'd take it. Dad, but a great uncle.
I'm a great aunt too. I get aunt cards for Mother's Day. So you get an uncle card for Father's Day.
Dad, happy Father's Day tomorrow.
Oh, good.
Do you think we'll get a Father's Day cake?
No.
Dang it.
No, we get cologne and neckties. Come on.
Oh.
It's not
I got two
everywhere
different.
in the world.
It's Father. Oh, really? Okay. I should know that.
That's why I always drink it.
Thanks.
Well, that is a tall.
That is a tall crinoid. It's beautiful. They're all facing, have you noticed that? They're all facing the other direction. When we come up to them, oh, we got a
Big sea cucumber.
big boy sea cucumber over here.
What's the secret?
Yeah, let's just take, because that's pretty large. It does look like an armadillo. Where's our lasers? There we go. 10 centimeters. It's like a 20 centimeter. He has no shortage of sand to graze on. There we go. I'm not sure. Is that one of the swimming ones?
I mean, I think it will swim from perturb, but that's definitely a big one that hangs out.
Well, yeah. It's not a.
But yeah, that species will definitely jump up and swim sometimes. Benthatides.
Benthatides. That's good. We can keep going. Let him float on his way.
People call it Falkor with a C too, and it's not.
I told Wendy that she named it after the Little Mermaid monster thing, or whatever it was.
Oh.
She didn't trust it.
Clearly you were new here.
This is my very first. Yeah, it was 4 years ago.
That's your first
It was
trip.
Pippa in 2006.
Yeah.
Really?
Yeah.
That was when she was
You can take a look at this urchin.
Yeah, she wanted us to write a check. We actually did watch it, but Darren did.
Oh, it's floating away. Never mind.
Oh, it's one of these little booted urchins. They have little
Yeah.
white
White
things,
booty.
little white boots.
White booties. Let's just see.
Hey, Tony, you're gonna have to bring the heading around the south of Maypan, or you just keep on it?
It's like a saucer.
I don't think I've ever seen one get blown off.
That's
quite
a
that
first
easily
for me,
an urchin.
yeah.
We're gonna start heading more like due west in a little bit, so if you need to move around, just let me know, and I'll try to position you what's best for everybody.
Wow, the sediment is really so light.
Yeah.
You can't even.
Yeah.
Okay, cool, man.
the seamount we're exploring today has never been explored before. It had one kind of rough mapping line, transit line from a ship transiting through the area previously, but before we got here. But we're exploring a similar depth range to what we were exploring around Howland, because we're more or less the same latitude, the same distance away from the equator, but 190 nautical miles more or less due west. So this will give us an interesting comparison of what this feature looks like
There's a
on
house
what
there.
honestly is somewhat a similar track line, similar seafloor topography, but further west and on a seamount as opposed to an island.
Sounds interesting.
Yeah, probably. Yeah, sounds good. Even though I feel like now I'm going to be tricked forever by the sea cucumbers that aren't sea cucumbers, or the tubes that look like sponges
The
that look like
parchment
sea.
worm, the parchment
Is that what
worms.
it's called?
I'm pretty sure it's some kind
Oh,
of
there's an
parchment
old.
worm at this point.
You can see a rock tumbled down, and the coral was covered in sediment.
That right there.
Yeah, we don't need to spend time looking at it, but that's what that
Yeah,
looks
I
like.
never noticed it before, and then rocky and I see
Yeah, yeah. It's like when we talk about there being hard bottom, you can have hard bottom that's not actually attached, and then so when the coral becomes too heavy in the current, it just falls down, and eventually the coral will die if it can't get upright again. Another halosaur. Those are pretty common down at these depths. We don't observe a big fish diversity, but those halosaurs and the eels are pretty common. There's another snailfish, maybe.
Probably a cusk eel.
Oh, okay.
But
There's a Brisingid sea star, a couple of them. One looks pretty small. They're attached pretty strongly to the rock.
now even here on the steep, really
Oh, here's a
like near
nice
vertical part, the sediment drape is
Glass
still
sponge.
pretty serious.
It's beautiful. Take a quick look at it as we pass. That's good. We can keep going. It looks very healthy.
It must not ever be occurring here for something this steep to be this sediment draped.
Yeah. I feel like we've seen that. There's another one of those urchins with the little boots on. That's good. We can keep going. We're at about 1,600 meters right now. So since we came in the room, we've climbed about 100 meters. Another one of those big sea cucumbers. I think there's very little known about sea cucumber reproduction, especially in the deep sea, but I'd be curious why they don't like dominate these habitats. There's
Yeah.
so much sediment that they could just, you know, and nothing, as far as I know, I don't know if they have natural predators. What eat the sea cucumber?
I know very little about their natural history.
We haven't picked up anything else since I was humiliated by the sea turn.
Thought we had something in 14 as well.
Let me see.
Yeah, maybe not yet.
Yeah, no, we don't.
How's that sea drainage?
Some urchins and crinoids. A lot of what we see kind of as the non-associated or inverse that are not associated with the corals tend to be echinoderms. So the urchins, the sea cucumbers, the crinoids are all within the same phylum. And seem to be pretty well adapted to this space in the deep sea as well as in the shallow water. They're ubiquitous across all depths. Just different variants of them. Crinoids are particularly interesting. They were thought to be extinct for a long time until they started. Were they first when they were imaged again? Because
I don't know exactly when it was, if it was trawls or at what point, but the stalked crinoids specifically are well known in the fossil record, but were thought to be a prehistoric group until we really started exploring the deep sea and found them in quite high abundance in some places. The shallow water the comatula crinoids, the unstalked crinoids you can find in shallow water. I see them scuba diving and stuff like that as well.
That's a shrimp. And it's not that surprising. Oh, here's a big outcrop. It's not that surprising that even probably from trawling it was difficult to ID a stalked crinoid because, I mean, we bring them to the surface and you kind of barely touch them and they start to fall apart. They're very delicate. So from a trawling perspective, if you were being caught up in a large net, I can't imagine that they would stay intact very well on their way to the surface.
Yeah, so much of what we know about the ocean in general, both the mid-water, shallow water, and the deep sea is by net trawls and stuff like that. So the more fragile organisms really are much less studied just by the nature of was up until the invention of these kind of deep submergence robotic technologies and submarines that we were able to see these things in their natural habitats without having them just absolutely mangled by a net or a dredge or something like that.
Yeah, or even coming up and dealing with the pressure change. A lot of the inverts are able to actually come up in good condition, but fishes have swim bladders, so they are often distorted by the time they come up to the surface because of the large pressure change. So seeing them in their natural habitat is not something to be, it is actually a pretty big deal for us to be able to observe them. And there was probably a lot of species that weren't able to be accurately described until there was footage of them.
And frankly, we still haven't caught up on that backlog, for lack of
Yeah,
a better word.
yeah.
I mean, there's many things that are undescribed species in the
Here's
deep
a
sea.
coral coming up on the
We
left.
often see
For a stalk.
new species. And even though we kind of know they're new species, people haven't had the time or the funding to take the time to scientifically describe the species.
Yeah, we talk about, finding new species, and people often ask if we find new species, and the answer is pretty much always that we're finding something new. But like Brian said, there are very few people in the world that actually focus on describing new species. It's not a trivial thing to determine if something is a new species versus just a different variant of a species that's already described. And there's many different rules, if you will, for when something is considered a new species or just a. This is a very long bamboo. Let's see what we got on here. Is it okay if I come in, Ben?
Abandoned ship, off jump.
Yep, off jump. We've got a lot of crinoids at the base. Very sparse polyps at the lower end of this. Some ophiocanthids, those spiny-armed sea stars. Let's see what's up towards the long. It's probably a couple meters, I would reckon. Yeah, if we can kind of just fly along the length of it, that would be great. Doesn't look like any predation.
No, I don't see.
Very
any
thin
signs
at
of
the
predation.
tips. It's just hanging out there on its own. That's good. We can keep going up. Another couple big stalked crinoids. Yeah, all facing uphill. Yeah, that's good. Yeah. Little crinoid.
Yeah, Bush project.
Big rock. A little small plexaria. Looks like another little whip coral, or a couple of them coming up. Oh, here we go. We got a big. Can we?
One on the right.
Or there's one up
Oh, with
front.
the whole top
Oh,
looking
yeah.
over.
It's
That's the one
actually
on top.
sure. I'll actually take a look at this yellow one first. There's no coffee in there. Don't worry. It's definitely seems to be a nice little spot where they have a lot of corals have decided to settle. I shouldn't say decided. They don't actually. We have a nice bamboo, and then behind it looks like a yellow plexaurid. There's a couple, it's like 3, 4 of those five
Look at
plexaurids.
that. I can't tell if there's a
Okay.
scale worm or something right next to it.
Ben, I'm gonna look right over here.
And Mark. Can you zoom out?
Oh, zoom out, yeah. Looks like a chrysogorgia.
I agree that I agree it's Chrysogorgia, but there's something on the seafloor right next to
Okay.
it. I think. No, it's Victor Gorgia as well.
Yeah. Okay. There was one just right down here that we were interested in seeing. We can come back a little bit. Yeah, there we go. Right here.
Zoom in.
Yeah, you're right. I think, Brian, it's a polychaete of some kind. So Chrysogorgia, the little octocoral, and then next to it, right behind it, is that very prickly looking polychaete sitting on the rock. I don't see any crabs in the chirostyle or in the Chrysogorgia, but could be hiding. And then up here, I'm gonna take a look at this one.
Another side.
Yeah. Oh, there's one of those big crinoids on the other side
There's
too.
3 different species of stalked crinoid.
I might just land on top.
Okay. Okay. Yeah, it looks like a Swiftia to me.
It's a plexaurid, or that's its own?
It's a plexaurid, yeah. It's very, very large polyps for a Swiftia, though. Could I have manual zoom, Russ?
You should have,
Or focus, I'm sorry, manual focus.
you should try again.
Okay. Yeah, there we go. It's very weird. Some very strange, like elongated polyps down here.
Yeah.
Might be growing over something else. If we can, I'd actually like to take a sample of this.
Right. I just wondering, there's a couple of pieces that come off that other coral too, so I'm not
Oh
wrong.
yeah, we could just, think you could just pick them up?
Does that seem
Get the red, the orange one first.
Okay.
Right, let's get a trio of them there. Fourteen is open, let's use 14, it's fine. And trade back in. How much do you like?
Let's see. Did you get a pre-photo?
We got a wide one and we got a gray one.
Okay, we'll come in real quick. Take a closer look at it. If we could take a snip with the ophoroid arm, so this kind of branch in here where we can take that arm with it,
Yeah.
that would be good. We can pop it in a quiver. Yep, absolutely. This will be sample 310.
Just there, right? You get a bit closer?
It's a nice view of that plexaurid. With a barnacle on it and a astroschema.
That good?
Yeah, that looks great. Perfect.
.310, right?
Correct.
I know, but I've got some of it now. Trying to get this all out.
Nice, perfect.
The other part, the other piece, would you want just another quiver?
We could just put it in the same quiver.
Right.
You want to call it the same sample number then?
No, call it a different sample number.
Each range, you know?
I don't think it's worth opening a new quiver for it though.
Alright, so this is 311.
Yes.
So I'm just gonna put a lid on, you can have a little bit sticking out, the rest is in.
Yeah, that sounds good. Or if you want to snip that, you could also do that. It might.
It's difficult for me to get in there.
Okay,
without
no
that,
worries.
just something,
Yep, just.
you know.
Great.
Yeah.
I think we are. Let me pan around a little bit, but we have that big crinoid, Chrysogorgia. Let's see if we can. Can we actually come up and look at this coral up here? Thanks.
Just one, right?
Just down, just below where we are. Oh, okay. Okay. It's this coral right here that we want to look at.
Look at all the mucus nets and everything
Yeah.
on that rock.
It's a very planar Chrysogorgia, it looks like. Oh. Okay, let's take a look. I need to let that clear.
Let me,
Okay.
get a better views on beautiful photo. That's the one. It's a little over. I'm just gonna suck it out.
Can we like ISO down or iris down a little bit? Thank you. Yeah, definitely a I keep saying chirostylid, a Chrysogorgia, and there is a chirostylid inside, it looks like. Let me see. I wonder if that's the same one we saw earlier. Okay, that's good. We can keep going up. Yep, another plexaurid. Yeah, go ahead. Keep going. It's an Gorgia around the corner. And looks like one live bamboo and then a couple bamboo bases. Looks like 3 more of those similar plexaurids. Another one of these little Chrysogorgias on the right. Another yellow plexaurid. Looks like we have some over here. If we can take a look over there. Some nice big colonies.
The one on the far right.
Yep, far right. Or sorry, left.
Other right.
Oh, look at that one growing between those rocks right there.
Oh yeah, that's cool.
I don't know what. Wow, yeah, these are nice. They found.
Probably gonna want to look at the dead tissue on that middle one.
Okay.
Probably want to take some good time.
So we'll
looking
take some
for-
time looking at this one first. It's probably.
Very round as well. Yeah,
these are going to be pretty delicate, so it's going to be just probably not going to sample them, but just take some imagery. This is a good example of the difference between the,
Zoom out a bit.
I think I'm out all the way, of Paragorgia versus Corallidae. So these are Hemicorallium, which is from the family Corallidae, and they have a very stiff, more fragile skeleton than some of the Paragorgias that we saw earlier, where they're really flexible. So these are really perched on the side of this rock. So we aren't going to set up to sample, but we are going to take some imagery because it looks like we have quite a lot going on here. Good? Cool. There one that looks like at the base we have a clam. Bivalve of some kind. Which is pretty deep. What do we got? 1534. Yeah, we have a couple branches that have lost the hemicorallium tissue that have hydroids and zoanthids growing over it. So the lighter pink polyps are another type of coral that's settled on the dead skeleton. And then, interestingly enough, the atroschema is on the zoanthids, the brittle star, with a couple large crinoids and a shrimp hanging out, or a couple shrimp. And then I believe we also have, let's look down here. Yes, a squat lobster. Kind of hard to see. It's on the other side, but there's
Chrysostylid or? Europtychus to me.
It might be Europtychus, right?
That's what I-
Which is a type of chirostylid, so you can kind of see the horn, the rostrum through there, which is helpful for Tim, who studies the associates that live on the corals. While we're here, we'll just pan over to this other coral and take a quick look. Huh?
I know it's such a thing.
Seems to be quite the hotspot for these hemicorallium right here. So the other thing that happens with this coral is there is a polychaete worm, which you can actually see right here, that burrows into the skeleton. So if you look right and left of middle of the screen, there's a small worm coming out of the skeleton, and if you look closely, there's a bunch of holes along the skeleton, and so these polychaetes live amongst, there's a lot of them that live within the colony that do this, and it doesn't seem to affect the health of the coral. It continues to grow, and the polyps are still all there, but the skeleton does become a lot more brittle over time. There's another one of them. And there's a
Little tiny ophiuroid next to the mysid.
yeah. There's some galls on here too, which we never really know what's in them unless we bring them up. Sometimes it's eggs, sometimes it's a grouping of polyps. something that the coral has grown over. We've seen a barnacle inside of a gall before. Not seeing any predation on these though.
No, no, am I. The first one we looked at definitely had, you know, big swaths of kind of dead tissue and overgrown, but I didn't see any active predation.
No. Here's one with all the polyps out versus the one we were just looking at had most of the polyps retracted.
Another piece of dead tissue on that one.
These are in a family that's known as the precious corals because of that skeleton. It's very brittle, but it's also been used. Certain species have been harvested for things like jewelry, but now they're a protected family of corals, so hopefully that's happening less because they are very long-lived and slow-growing, so it's not trivial for them to build that that hard skeleton. Look at that gall at the base of that. It's huge. I wonder if that's one of those. I would be curious if that's a clam inside there like we saw on the other. Okay, let's see. One more colony here. Also looks very healthy. Lots of large crinoid on it.
What's that?
Oh, and then one kind of growing in this underneath the rock back there. That's a really cool orientation. We see that more often with hemicorallium than some of the other corals where they really will wedge themselves and grow in between two rocks. Okay, I think that's good. We can pull off the wall. Yeah, they'll wedge themselves between, or it'll start growing between two rocks, likely because there's nice flow in that space, and then they will keep growing and they just kind of mold into the rock space.
It just continues
Oh,
to
an anemone.
pick up.
Oh yeah, there's some more hemicorallium, a nice whip coral.
It's like a big Chrysogorgia in the corner. Wow, what
Yes.
is, wait a second, what is that?
What's what?
That stalk, can you
You
guys
want-
come up a little bit in altitude?
Just look at this, right? Get to the top of this. Another type of
Oh, it's a
crinoid.
crinoid, okay.
Yeah. No, I think we can keep kind of carrying up slope. We are getting a higher abundance of corals as we're climbing up. We're now at about 1,530 meters. I would say over the last like 20 meters or so we saw.
Yeah, once we kind of got a good little ways up that kind of sheer rock face, we started picking up the,
Here's a big bamboo coral. Wow. Can I take, yeah, let's take some time to look at this. We've been seeing the stalked, or just the whip bamboo corals, but I think this is a different one that we've definitely seen at other sites before, but we haven't taken a look at one today.
then have you noticed an increase in current up here? or is it still about the same, pretty still?
There's some current here now.
Okay, so there's some pickup in current.
It's coming from the west.
Lots of crinoids on this. It seems to be a very crinoid heavy dive. Not many other associates, just the crinoids. I don't see any squat lobsters. That's good. We can.
We'll see.
Yep. It's very large.
Yeah.
Remind me when we see another patch of corals to take a Niskin.
I had that thought when we were parked on the hammock rally, and I forgot when we were done looking at it. It was a black.
Oh yes, looks like a black coral. Yeah, that's one of the big questions in deep sea is why are certain areas kind of filled with corals or more there's a higher density of them versus other areas, and that's a good example where we've been moving up this slope pretty consistently, and we've seen rock outcrops all along the way, but we came across an area that just seemed to be, higher density. There's something about that space that facilitated coral settlement, and then they just did really well. They were able to grow to pretty large sizes. We've had relatively decent diversity. We haven't been seeing necessarily the same one species the whole time. We've been seeing some plexaurids, which we're not always sure on this genus or species ID for those because that's a tricky group, but they look to be relatively similar as we're going through. Here's an Chrysogorgia. And then a number of bamboo corals, these Chrysogorgids like Iridogorgia and the little plainer one that we've been seeing. Victorgorgia, which is the purple octocoral. What else? Hemicorallium.
Swiftia.
Yeah, one Swiftia. Victor
Gorgia there.
Gorgia. We saw Paragorgid earlier. There's a Umbellula, the sea pen.
It's been a pretty, I mean, it's been light overall, but we've seen a lot more corals and sponges here.
Yeah.
which is-
Oh, actually, can I take a look at this coral right here, Ben? Sorry to slow you down. Yeah. I think it's a cup coral. But yeah, we're getting to be in about the range for those. This right here. Okay. Oh, okay. This is a cup coral, so a solitary scleractinian, which is pretty deep for other types of scleractinians, the colonial ones, but we do see these cup corals down to about 2,000 meters. Oops. You can see all the tentacles are out, but underneath is that white oval is the skeleton of the coral. And now it disappears by smoke cloud.
Vanishes.
Actually, I'd like to sample that if we can. If we can slurp it, or you can grab it, Russ, whatever.
It's up to you. Slurp it would really be a good idea.
Yeah.
Actually. I'm gonna reach it from the other way.
Yeah. Settle down.
And then you got minus 19 minutes. Maybe it all eight or just keep turbulating.
Okay. Let's see.
But I go a bit, don't you think?
Thanks.
Do you want to pull on, or are you gonna slip it? I need pork chop, Russ.
Oh, yeah.
Go ahead. This is gonna be in seven?
Okay. I think
Five.
something's in 7. Yeah. 5.
I don't have to. Let's jump up. One more. Grab one more here. Just tight stick it. Now you see brown style on the other side. This is good.
It's underneath, isn't it? I see, I see. There it is. Oh, it's in the wrong one.
If you're just tuning in with us, we're diving on a previously unmapped and unexplored seamount on the western edge of the US EEZ around Hallen and Baker Island. It is a lovely Sunday morning for us, and happy Father's Day to all the dads on board and the dads at home and the father figures. We have been diving for a couple hours on this seamount. Started at around 2,000 meters and are currently at about 1,500 meters.
Do I zoom in and get a shot first?
Yes,
Sure.
please.
There you go. All right, we are good to go. Oh, we can just.
You leave the suction on, man, just on low, man.
Yeah, I'm back to
Right, well, put it in and see what you can.
Hold on.
Just leave on low, man.
We'll put it in the river, right?
Big cup coral.
That is a big cup coral.
Bigger than expected.
Yeah, me too, actually.
Poaching out.
It's okay.
Looks like Javania, which we've seen at other sites. But that really thin base is kind of how you.
Yeah, no,
Stop.
stop.
See, right camera back as far as you can.
That's it.
Zoom right there. Suction on.
It's on. Forward. You ready?
There you go.
312, right?
Yeah.
It's off.
Whee! Great job. Thanks, guys.
Urchins covering
That was 312, right?
Yep. That's what I put in here.
Please see if I can zoom out.
Okay. Give me a second.
So a question about the effective pressure on the samples we take. Generally, the invertebrates that we sample down here are fairly pressure tolerant. They don't have any gas swim bladders or anything like that. So they handle the pressure changes up to the surface reasonably well. It's actually the temperature that they seem to be more affected by. It's pretty cold down here. It's 3.1 degrees Celsius, so that's 36, 39-ish degrees Fahrenheit. And so we're really quick to get the samples onto ice or in the refrigerator when we get up to the surface, so we have time to process them and get pictures of them and get them into a preservative to try and before they start to degrade. If you start bringing up fish and things like that, the pressure becomes more of an issue. And for long-term health, keeping them on the surface, the pressure is an issue. But for just the ride up in the vehicle until we can get them in the lab, it's temperature we worry about, not the pressure.
Yeah, and especially like the corals that we sample, some of them we're able to keep alive longer than others, and it's not fully understood why that is, but especially there seems to be something about being shallower than 1,000 meters. They tend to be able to stay alive longer in an aquarium, as long as the temperature is kept cold. But some of the octocorals especially really don't do well with trying to keep them long term. So we just collect those in order to have identification and be able to run different analysis, like figuring out running DNA barcodes so we know what individual it is, and then running isotopes so we can have a better understanding of what their nutrient source is. But Randy Rogen's the chief scientist on board, and she is running a set of experiments while we're out here. So instead of trying to take corals back to a lab, we've actually brought the lab on board with us. So that makes it, being able to put the corals into the chambers relatively quickly makes it possible to do those experiments, because the corals don't have to stay alive for a long time in order to complete the experiment.
And when Alex says chambers, we're using these little respirometer chambers, so we're measuring the oxygen consumption of the corals as a proxy for their metabolic response to different stimuli, investigating kind of how their immune systems work.
And even just basal respirometry, so like what is the sitting respiration rate of some of these corals, because that's, for most of these species, that's never been measured before. So gives us a little insight into how quickly they consume oxygen. And it's a very small amount of coral that goes into each little chamber. So we're able to replicate that at a pretty decent replication rate in order to be able to run statistics on it, to know if we're within a margin of error or if what we're seeing is a true rate of respiration. Is this coming off of us, or is this?
No, this is coming.
Oh wow, that was very stark. Okay, we very quickly have entered into kind of a sediment fog.
Yeah.
Where was that? Right at 1,500 meters. Looks like a anemone. Yeah. You can take a quick look at it. Just, I don't think we need to set down. Yeah, it looks like an anemone. That's a good question. Do the corals at this depth have a slow metabolic system? And that's that's something that this experiment is trying to answer. So they do, it is thought, and from some previous experiments known, that they do respire at a relatively slow rate compared to especially shallow water corals. But we also just don't, there isn't that information yet. So that's why we're running some of those experiments, is to try and get a better sense of just how slowly they respire, and if the different families of corals or the different types of corals respire at similar rates. Can we head over here and look at this coral? But because it is so cold and they are so food limited, they don't have a constant supply of food like shallow water corals do from their algal symbionts. It is understood and thought that they do grow relatively slowly, and they can be really long lived, so they would be conserving that energy over time. And they might also, there's some theories about corals receiving like pulses of nutrients, so when there's more surface productivity in an area, they will be more productive. Just this one down here. So they might not have a constant supply of food, but they might have like a really high amount of food at a certain time of the year, and then at other times of the year they just kind of sustain themselves until they have that higher input of nutrients again. Oh, there's a couple. There's one down there too. Great. Looks like a Crinoid. With a Astroschema sea star. Very dense polyps in each whorl. I'm gonna say Narella, but I'm not really sure. I think they sampled a Narella earlier on the dive, so we're not gonna sample, but it looked like there were two of these. There's one right below us too. We don't need to move, I'll just pan. Sea star on the rock right there. We have a crinoid right next to it. Their little stalks are right next to each other.
I think that it's beyond me. What are you trying to do? Chop that one. That's lower. It did lower, it didn't go away.
So one of the other things we are sampling when we get these organisms up is where we are taking some samples to look at their microbiome, so the bacteria that live with them, and and we'll be filtering some of the water samples we collect with the ROV to get a better sense of the bacteria in these communities as well, because that's an important thing to understand when trying to better understand how the immune systems of these organisms work. We need to understand the underlying bacterial community around them, and it's also at the same time a useful way to get a better sense of the microbial community broadly in the deep sea.
Yeah, so we're not keeping them under pressure to bring them to the surface, but there is a microbiologist on board, and she is taking samples from each sample that we take, so coral or sponge, and she is culturing those samples to be able to see what she can grow on media and take back to the lab. And then we're also have plans to preserve samples for microbiome analysis, so that's where you are just sequencing that information versus trying to grow those bacteria. Any particular good resources for identifying known animals in the deep sea? I think I called you Ben. I think Brian definitely has some experience with this.
I mean, there's a couple. I think my personal favorite is probably the Oceanus Explorer benthic animal ID guide that NOAA maintains. I've seen a couple other resources. MBARI has Monterey Bay Research Institute has a decent one, but it's certainly something that it's hard to do, and a lot of these things can't be identified without microscopy work, or at least getting looking at under a microscope. So that's something that's frustrating to do by video alone. A couple sea cucumbers here in the sediment. Looks
And
like coming up on a sea pen.
Umbellula.
Trying to catch your sea pen?
No, not this
Not
one.
this one.
Okay.
Wrong seat. Maybe you'll get your revenge.
So we'll grab a sea pen now. That one like this. Disappeared.
I distinctly remember that happening in 2017 too. That same. So some of them can retract.
Oh.
Yeah.
You should have reminded me about that.
Well, sorry, I wasn't on watch yet. I was in the galley getting coffee.
I just sat there like, whoa.
I remember trying to sample. I hadn't thought about it being that same one.
It's like that game when you put the $1 bill on or $20 bill on the ground, but you have it taped to something, and when they go to grab it, you pull it out of the way. It's a little less.
Remember in Honduras, there's kids that tied a string to a wallet and was walking across the road, and it was like, oh, wallet, and they reached out, and they thought it was a fucking scam.
If I'd have seen it, I probably would have as well.
I was laughing.
Let's see. There's another Crinoid.
We'll look at it.
No, we can keep going.
Yeah, it's not my
That's a big Crinoidea.
Oh wow, that is a very big Rotogorgia. We can look at that
Run
just for.
around and get a other side view. So we're gonna go all the way around to another. Looking at that, see right there.
Oh yeah. Hey Dad.
Yeah.
Could you could you type in the name of that into the chat?
Yeah. Spell it for me. What am I typing?
It's just the NOAA Oceanus Explorer animal ID guide.
Yeah, it's wrong.
EANOS.
I always spell it wrong. I don't know why.
Here's a nice Chrysogorgia, which we've seen a couple of today. A couple different sizes too. This one, similar to this.
I think
Animal.
instead of creature.
Yeah. Just take creature
take
out.
both.
You like those creature back?
There is somebody,
There's a little
somebody
shrimp.
about that.
I'm just asking.
There's a shrimp that seems to be hanging out around this coral. There's also, I'm gonna zoom in a little, Chris.
Far side or something.
Yeah, there's a, looks like a squat lobster in there.
It's the latest version, 3.
Yeah.
Try to line out really good.
No, I just wanna try and look at that crab over
Okay.
here.
That's what we're gonna do that.
Cool.
Going in a snap zoom?
Snap zoom. Was there a terminology conversation?
Yeah.
Okay.
Randy told us last night.
Okay.
Snap zoom.
I mean, I feel like we've been doing real good.
But we can call something cooler.
Like what?
Like a, I don't know, solution douche unit. I don't know.
Shit.
What do you call it? Well, it's not a reverse Chesky. A reverse Chesky. Yeah, you're gonna leave back somewhere.
It's definitely not that.
Oh, that's a great shot, Chris. Nice.
Well, be back here.
Alright, no,
That's okay. I'll come out a little bit, yeah. I mean, that's good. I think we were able to get it. We can keep going on. Another place to look for information about a lot of different animals is a database called Worms. Which, do you know what worms do?
World Order of Marine Species.
There you go. And you can find some information about the depth ranges of certain species and when they were first described and who described them. There's not always images, so it isn't always helpful for trying to ID something based on images, but if you know what you're looking for, then there's a lot of information there.
the current change moving around this thing.
Oh, really?
Yeah. Can you look at any of these close up? Look at one right cool.
Mm.
Here.
Yeah, we'll take a look at this one real quick. On the bottom of the screen. We haven't been zooming too much on these, but we should probably get a couple close-ups.
There you go. Ah, there you go. Yes. 1 in 4. No, I don't know. I didn't really give it a chance. We're playing that. We'll try to get into it.
Really does look like snow down here.
There it goes.
Part of me wants to say Acanthogorgia, but it's hard. Flexarid or Acanthogorgia. Okay, that's good. Thank you.
Then we'll just upload it.
Nah, we can
No?
keep going. Those are crinoids.
So sometimes we do see fairly extreme currents. The other day we hit a set of currents in the shallow that made it very hard to drive the ROV. I've seen 1 or 2 times I've been in situations where the ROV just could do nothing but sit on the bottom for a few minutes until the current changed or subsided. They're not necessarily associated with Ekman spirals. Those are, by our definition, shallow water features, and they kind of taper out with depth. But you will certainly see some strong and sometimes sudden shifts in currents in the deep sea, especially around steep topography changes.
Can we take a look at this, Chris? This little, it looks like a sea star. I can just do a snap zoom, if you will. Just kind of slowly come in. It's a different looking star than we've been seeing. Oh.
Whoops, oh no.
It's okay. He'll right himself. Or herself.
Oh, wait, the most fun is out there.
Some Victorgorgia, the purple ones. We can just keep going. Here's a different sea pen we can take a look at. We might actually sample this one. Yeah, why don't we sit down to sample that?
I don't think that. I think that the it's just
That's a little like stuck on that.
it's pulling me forward. Yeah.
Just the display of the still capture.
Okay.
So when I just bump the space bar and pause the display.
Oh. It's just like we saw that many hours ago. Or not many, but
It went, dude, it did quick.
so it retracted most of its polyps in our approach.
Try that.
Yeah.
That's pretty good.
These are very beautiful. So it will have a base underneath, probably not embedded, but using to anchor itself into the sediment. I don't think this one will retract on you. So I would go for it with the claw.
Okay.
The arm, not the claw. And then I think we can just put it in a quiver.
Got pictures?
Got pictures?
Got pictures.
Hold on. What's that?
So there's a question in the chat about emerging technologies looking forward to, and I think
14
there's
and
a
12
couple,
are full.
that I'm
Yes.
looking forward to. One is just the increasing amount of autonomy and the persistence of these vehicles. We come down and make kind of single time point measurements, and it'll be easier and to understand the deep sea when we can deploy assets for longer and a much higher density. So having systems that are more autonomous and can stay deployed longer will go a long way in helping us better understand the ocean.
Yeah, I agree with Brian. Pretty spot on with that.
What kind of you do?
we really appreciate how far the technology for this type of ocean exploration has come. But now that we are pretty consistently doing like this type of ROV operation and exploring a relatively small track on a feature, I think the next real step forward is to be able to really survey a whole feature, a whole seamount through autonomy, and then be able to return with an ROV or a human-occupied submersible. I would just try and not.
Oh, we're just worried about the rock on the side.
Oh, sorry. Just don't. Grab it with the rubber bits, and don't cut it, and it should come up all together.
Okay.
The other thing I think I'm looking forward to in the very near future is the increase in bandwidth for satellite telecommunications on ship, which will allow a lot more collaboration and remote operation of systems as well. Being able to, at a much cheaper price point, be able to do a lot more bandwidth will increase some efficiency as well.
Great. Whoa. Whoa.
That's super cool.
Is that gonna fit in the quiver? You wanna-
Yeah, I think if I turn it upside down, I could snip it too if you want
It might be better to put it in a bio box because it would be a lot more useful as a whole specimen.
Sure. Alright, well, some more? Yeah.
We can put it in bio box B, the middle bio box.
The one with the sea star in it?
Yeah, because we know that they're
We
separate.
can tell them apart.
Yeah, exactly. Yeah, sea star could probably use a friend what happened to it. So.
It's a snack.
There's also a question about what kind of media we're using to grow the bacteria, and frankly, I don't know.
Yeah, I don't either. Sorry. Maybe another time somebody can answer that
Yeah,
when there's more
when-
people
when-
awake.
When Anna's on watch, you can ask her.
I do think it is a consistent media across the board. That's what she's using the same media for all the
Yes.
samples.
That is, yes, I do know that. There's only one type of plate.
And she did this in 2017 on our first trip with the Falkor to the Phoenix Islands protected area. And she published a paper about some of the characteristics of the microbes that she was able to culture from that trip. It's published in Science and Immunology. I don't know the title of the paper.
Not top matter, any other.
Perfect. Thanks, Adam.
So this is sample 313 in BioBox B.
Yes.
Moving anything else while we're sitting here? We have
Do we have anything else around us? We got a sponge. We'll take a look at that sponge before we lift off. Oh, sounds good.
There's also Chrysogorgia up on the left with the master schema, if that's at all interesting while we're here.
Oh, okay. This looks like an encrusting sponge, where it's growing on and around the rock versus having a base and growing. Oh, this one over here. Let's see. It's getting a little bit out of our light field, but we should probably be able to get a little look at it. Oh, those look weird. thank you. That's great. Victorgorgia with the astroschema. Actually, I'd like to go look at what this is over here. Yeah. Those polyps are gigantic. Yeah.
I don't know. I don't think so. It's just, I'm gonna try for it to.
There's a question from on Facebook. Do we sample the rocks or the sediment? We do take samples of, especially the rocks. We have geologists that are not on board but are partnered with us, who will be able to age date the rocks, look at the thickness of the crust that's formed on the rocks, and do some other things. Maybe Brian.
Do what geologists do with rocks.
Do what geologists do with rocks. We're not taking quantitative samples of sediment, but we do bring sediment up. Sometimes we use the suction sampler to sample the sediment because there is a really high diversity of microbes in the sediment as well. So that's of interest from a microbiology standpoint.
Getting to places like this that are so remote. We're nearly 2,000 nautical miles from Honolulu, and I don't know, 600, 700, 800 miles from Samoa, which is probably the closest port of any real size in Apia. So getting to these super remote locations, we really put a lot of effort in partnering with other people and other scientists and try and do as interdisciplinary of an expedition we can. We only have biologists on board, but we have geologists and oceanographers that are kind of on speed dial, that are sending us requests for data or that we are pinging with questions. So the power of the internet that allows you to watch this video also allows us to collaborate in real time with other people.
It's this coral.
So
Yeah, it was
we try
something
and
that
collect
had
as much
much
data as we
different
can.
morphology, maybe more to the right.
Yeah, let's not run. See anything over there.
Okay, let's just take a look.
Wasn't that, was it?
No, that's Victorgorgia, right? I mean, it's way far out there, so. That's what. Sorry?
Just look down.
Yeah.
Yeah, 'cause it was kind of purpley in color, wasn't it?
Maybe I just.
No,
I didn't think it was.
no, it didn't
looked
look like
it had
Victoria. It had big fleshy
really
polyps.
big polyps
Yeah.
on it.
Something's right there.
Yeah, I think that is like a Victorgorgia, but either it was like the current water playing tricks on us,
No,
or
I don't know. I definitely saw what you're
Okay.
talking about.
I saw you guys were talking about.
okay.
But
I think it is that purple one right there. Left of the screen there.
Oh,
'Cause
Well, we were
we
looking
were
at
sitting
looking
here.
at that one.
Okay, right. We're
Yeah, we're
yes.
sitting there and then there's
Oh yeah, there's behind the
So-
rock.
right there, I see it now.
You got it.
Yep, right next to that big rock sticking up.
That's what this looks like Victorgorgia from now.
Well, but that's. Wait, let's see. It was like we
That
were
was
here.
right here.
Yeah,
It sits down, yeah, then it'll be right there. Yeah, 'cause we looked at that one on the left,
It's either
and then it was this one on the front, or this rock right here.
yeah, it's either one of those three. Take a pick.
Well,
must
no,
have.
it was in the distance.
Yeah, it was pretty far out.
But where he was sitting,
Yeah.
those are the only ones you could have seen because everything else would have been sitting here,
Yeah, we
and
were
you look
looking
over
at-
to the left arm, so it's the one to the left of the big rock clear.
Oh,
I was sitting right.
right, yeah.
Right. I agree.
I get it.
I think we must have been looking at that
I think it was
one.
that one right there.
That one.
Oh, it was just a weird.
It was-
Okay.
Just looks pillowy.
Dang it. It's fine.
Yeah, from this angle, it definitely looks like Victorgorgia, but I mean, when we looked at it
When we-
first, it looked
okay,
like something
I'm
different.
glad I wasn't
no,
crazy,
I was right
'cause-
there with you.
Well, I mean, that's
Well,
At
that's.
least it was group psychosis.
Yeah. But we can take a look at this long one over here with
Okay.
the spiral. We'll just go
Shade over there.
take a pan of that, see what's associated with it.
Back to the emerging technologies question, thing I didn't think about was
Oh, this
better
looks like a-
underwater communications would go a long
yeah.
way in helping that too.
This looks like a dead Iridogorgia stalk, so no live coral on it, but there's a couple crinoids and brittle stars all using that
That's a
stalk.
percentage, isn't it?
Yeah, and a bracingid. Well, we're gonna have
Gonna have
a
an off jumper.
jumper. Whee! Yeah, what's that?
What is that?
Sorry. Maybe that's an anemone that is on the other side. Yep. Yep.
I always watch you guys play ping pong with the You're going ready.
I'm interested in this, like the center of that. Yeah.
I mean, it looks like the sea star's stomach to me, which that's not a sea star I would have expected.
It looks, yeah, very.
And it looks like it's trying to digest something.
Or it's.
Or is that a gastropod?
That's. I'm. Coming a
I
little
mean,
bit
there are species of gastropods that are known to be parasitical on crinoids.
It is a gastropod, right?
Yeah,
That's a gastropod.
That's a snail.
How do I feel about sampling the snail? It would be a flying operation.
Yeah, I don't.
Actually, I tried it.
We're out here.
Yeah.
So this was Baker, this was Helen Baker, and then we came out here. We stopped a little up here, and then this is Seamount, so we're gonna do a number of guys here, stop down here. Down here, not back to the
Do you want me to zoom out? 'Cause-
No.
Okay.
You can look where you hold down one of these.
Okay.
I'll show you where we're. So we did this one.
are moving, yeah.
And then
Yeah.
come down here, just as we dive on one of these three.
Oh, yeah.
And then one of these. Dives here. This one turns in here, the one across the
Dang it, Brisingen, move your arm out of the way.
Well,
Were you the crossing at the international dateline, though, you said?
no, that's over here. We're like.
Totally did that in September.
So we dive here, have some folks. Maybe dive here.
Uh-huh.
There's like 3 or 4 now.
See the wrong side now?
No, it's the right side. It's just this arm of the other one is in the way. So we're trying to look at this little guy that's right in the center here.
So the numbers were wrong. These were, yeah, so this one here. And then the numbers are all wrong because we've changed, like, this was supposed to be, yeah.
Well, I think we should just keep going. It's not the coral's dead, so it's not
Yeah, it's not
actually.
corallivory.
Yeah.
If anything, I'm more.
Now we're stationed. I do actually want to look at what this is down here.
Supposed to be 2 here, 1 somewhere along this transect. Winslow is, we mapped this ourselves in,
What I'm curious about is if it's parasitic on the Brisingen, like these are on the crinoids.
That's possible, right?
Yeah. Because I mean, the association with the crinoids is that the snail literally like sticks its mouth in and eats food out of the crinoid's mouth, and it looks like very
That-
similar
yeah,
to
it
me
does.
here with this.
And the snail looks much different than the other things we've been seeing on for
Yeah,
corallivorous
it
snails.
doesn't- Yeah, totally.
Yeah, we can keep going. It's interesting, but I don't think it answers our objectives.
No, I don't know.
and there's some other data where I have this, but they're right at the right on time. Too tough. 1 or 2, and then this is like, this is too tough here, this one here, here. No, this is and somebody else's data.
35 move a meter and a half.
Whoa. Slow her down.
Shush. Well, you guys, we were moving much more before.
Yeah.
We should be happy that we're barely moving. Means we're seeing things.
Might have running drone. Trying to catch the ship.
Yeah. Okay, we're like.
So this is the dive track rod, that was. So
Oh, this
Or
looks
this one
like,
too?
oh, it's another sea pen, yeah. We'll
Whoa.
look at both of these.
That's
like
right.
a-
That's
That's
the world's biggest sea pen.
Huge pen.
So they're called sea pens because they kind of look like old, and that's a Chrysogorgia, which I would suggest that we sample. They look like old feather pens, so the base of them is like a tipped little
So you
quill.
just
There you
take
go,
the top
like
off.
old feather pens.
When you sample this, you sample just a piece of it, or you sample it?
Um,
Sample at the top?
It depends on what
Try to
your
sample all of it.
objectives are, but I don't think we're gonna sample this one. We
It's too big.
sampled a previous sea pen, but you do, for sea pens especially, you do want the whole specimen. And this is a PRIMNM, but it's a Chrysogorgia, possibly Chrysogorgia clarki, because that is in this region.
Is that named after Malcolm Clark?
I do not know. I bet you Steve knows.
I'm sure Steve knows.
But this is the first one, so we'll just take a snip of this today. After we do some zoomies.
Sit down, 'cause I'm running both.
Oh yeah,
both
for
spots
sure.
at the moment.
Take your time.
Oh, okay. You thought you were gonna.
No, I didn't. I know that will never happen.
She wants to take a sample.
I'm actually really good with the eye.
I'm not doubting you.
I mean, I can make the box, but that's about as far as my eye.
What would like to sample?
Well, we're gonna take some images first, but then we are gonna sample this coral that's in front of us. Yeah, somebody, the unknown explorer mentioned that she likes to see pens that boop down when they're, pull down when they're touched, and that happened earlier to us. We tried to sample before Brian and I came on shift. There was an attempt to sample a sea pen, and it just completely disappeared into the sediment. There's a lot of ophoroids on this coral. Mostly astroschema, so not, there's no pheocanthid at the base. Let's take a look at the polyps.
Yeah.
Okay, so we'll want to get a section with a sea star, or with a brittle star.
I think that one you're just looking at is probably the best target.
I have a feeling Steve will also want branching, but I think you're right. I think that's the,
They said there's a branch in that arm, so yeah, that's what I would.
right, yeah, so just right below. Alright, Adam,
Yes.
I think we're gonna go for this branch, so just right below,
Just the one on the right?
yes, this branch.
Okay.
But you can just do it here, under the sea star,
We
or
want
brittle
tails.
star.
And then in equivalent?
Yes.
Quiver should be.
I think 13.
There's a big, is that a Brooksinder back there, you think?
Looks like it.
Yeah, jump up a little bit.
Oh, you got it. Okay.
Strad.
Right there, Alexis?
Yes, that looks great.
And yes, I did set you up at the very end after.
Yeah, it should easily snap off for you.
Whoever put the blades on these. Whoever put the blades on.
Two, Dave left?
Alright.
should just pat yourself on the back. You
Do you
don't
want me
need
to
to
pat
do
you
it.
on the back?
How was that fishing expedition?
I was gonna say these things, we all sat here when we, yeah, nope.
Sample 13, 14.
14. 314.
Sample 314 and quiver 13.
Yes. Nicely done.
You want the little pieces?
No, you're good.
Sure.
If you want to get them, you're
Yeah, I
welcome
think
to.
we should. Oh, never mind.
Just kidding, never kidding.
No, now we really want that.
They'll probably still be there.
Huh.
Do they control to see
Yeah, that was sample
how much
13.
the effects of not being in quiver has on?
It's called death. That's the effects.
Come on.
Nicely done.
Do 16 pop 16 everyone? So we stay with it, it's fine. Wanna take another one out real fast?
Yeah.
Set it up.
Let's go.
Was that one
That was
13?
13. Let's go 11.
11? Alright. Ooh, smell right.
Yeah. How could I not see it? Sticking up past the camera. Are you flying with a roll of like 6.8 or what?
It's actually not that heavy.
Really? Looks massive.
Yeah, I picked it up so that we could get some. Russ was complaining that we were too light. I didn't have one that's heavy. Alright, anything else in this view?
Let's take a look.
That was 11, right?
Oh, 13. I just crossed it out for you.
Oh.
You just
So we
opened 11.
Do you think we should fire a Niskin? I mean, there's some stuff here.
Some stuff.
I'm just worried that we're not gonna. We haven't fired any yet.
I know you were. Don't lie.
Earlier, we can do that.
Fake news.
Did they not? It's not on my.
No, I don't know.
We talked about it, I guess we forgot. We do it.
Okay, let's keep going. We'll do it.
Anyone this guy?
No, I guess not.
You're so illustrative.
I know.
Alright.
Just like flying towards
I should have done one at that heavy corallium.
Yep, we should
Really
have.
like that.
We both thought about it, but not, and then didn't say it.
Right.
Didn't do it.
Rescinded. Let's pull this up a little bit. There's another one of those sea pens that we sampled. They're all pretty small colonies. A couple primnoids, smaller Chrysogorgia, and some plexarids.
I think it deserves a closer look.
What is moving? Oh, I think it's an urchin. No, I think we can keep going.
What was that? It's what went on there now, it's correct.
Almost at the top.
Can I zoom on this a little bit? I want to just try and
Do
get.
whatever you want.
I want to get it before it pulls in its polyps when we pass it. So I'm just gonna zoom and you can fly.
Alright, happy?
Yes. That's good. I was surprised.
Pretty cool name for snaps.
You don't think Snap Zooming is a cool name?
Kaboom zoom.
What is
Zoom
it?
zoom.
Zoom zoom.
Alright, yeah. I mean, I won't call it that.
Zoom
We'll
zam.
write it.
Zoom cubed.
Adam's face is like, what is happening?
Zoom
I
square.
don't want to say anything.
I mean, I can see it.
So a question about using photogrammetry, yes, we are experimenting with photogrammetry on this cruise. Some
Did that
some of
sediment
the
just kinda start
high
coming
density
in?
communities we have been
Sediment
doing some
blanket?
photogrammetry work with as well.
It's a huge
Okay.
direction. Actually, no, let's go upstairs. I don't know where this is at.
We saw that it happened like
It's
one
Second
other
this
time.
has happened. It's really weird.
Yeah.
Fly right through it.
Bridge, ROV. That's an updated waypoint.
It's like a sediment cloud. Oh, there's another Chrysogorgia.
I feel like we're halfway between the United States and Australia.
I mean, like in the middle of the Pacific Ocean there?
But I mean, like, if you just kind of
I
spin
feel
the globe
like-
a little bit, it's like almost smack in the middle.
Where are we?
Right here. That's like too much work. I like just estimating.
No, we're closer to US.
Well, we're in US waters, but.
That's
Any
good.
little
Nobody.
jellyfishes?
Nope. Nobody's home. Nobody's eating.
Very purple Victoria.
Yep. I know, Deb. Where are we on the track?
Just show that.
Show me the track.
Ready? Started down here.
Where are you
See
at?
it?
No.
Stand by.
There we go.
Started down there.
Oh, okay.
We're almost at the top.
Ooh.
This is where we are in the world. In case you didn't know.
I heard that we're in the Pacific Ocean.
Halfway between the
United
US
States.
and Australia.
Another nice
Wow.
Chrysogorgia. And it looks like a little somebody behind there. So many astroschema on those.
And you watching right? Are you watching right now? Okay, let me know.
I'm gonna try and take a look at that pink coral.
I'm gonna spin around.
Cool.
Who's gonna watch?
Well, Pete, my dad was watching, and I was gonna get you to porch out with a Happy Father's Day, but now he's driving.
Tell him not to, no watching and driving.
Yeah, no watching.
Do that all the time. What are you talking about?
Deb.
Probably.
I still can't hear you, but
Where?
it's okay.
So no,
Oh,
right
no,
there, that's
oh.
straight, that's
Yeah.
straight out into the abyss. Same structures.
having trouble with the sippy cup and the cup over there.
You having trouble with your sippy cup over there? I'm not realizing, sorry. It's not zoomed. Okie doke.
Or it looked the same, but something. Deja vu. Wait, I think I got this
What do you
one.
think, Brian?
I'm not seeing any movement.
And then I'll tell you what I think.
I'm gonna vote Crowley.
Same. I would say hemicorallium.
The only way to know for certain.
Or you just move your arm next to it.
Just close the bulbs.
It'll, if the skeleton moves, if the coral waves, then it's a Paragorgia. If it doesn't, then it's a,
Bubblegum?
yeah, no, hemicorallium. Paragorgia is bubblegum.
Bubblegum moves, Corallium doesn't.
Did I say that backwards?
So just not
It looks
touching
like
it.
bubblegum.
just
Don't
wait.
touch it, just make motion around it.
Okay, do
Need a wave.
Gotcha. Dude, you just knocked me off the first doing that.
Intimidate.
That was pretty unstable, obviously.
Yeah, that looks like
That's
hemicorallium. Nice test.
Very sorry.
Oh, there's an A-plaque offering at the base of that.
Alright, so what did you look at?
Let's look at this one while we're here. Experiment concluded.
She was a scientist. Got it. You guys want an experiment?
It's sea log. I just cut the screen down and then, so if I leave that up, or I do figure out if it's worth it, that three, but like, it's yeah.
Let's take a look at that A-plaque offering.
Let's go check that out.
You see the house? Wow.
Yeah.
So it's
Call it
just
Chrysogorgia?
Yeah.
A lot of oats.
Yeah,
Lots of
basically
oaks.
it's just
We
screen.
sampled
So
one
it's the
just
same
a
thing
couple
I did
minutes
for
ago.
Excellent.
or-
Different
It's
colonies.
just put
Yeah.
it on the screen
Yeah.
and then I made it opaque, so it
Dan.
kind of looks like.
And the Coffin, is that for
Oh,
the
Dan
one?
Finari is the guy who made the GoPro. Or like,
We made the GoPro.
it's not what I meant.
GoPro.
He made the housing and the whatever.
He put the system together.
There you go. He's responsible for that technology.
That's such about me that that's the first time I noticed that there was a name on that.
Yeah.
Is that a bubblegum?
It is a hemicorallium. We did the arm test and everything.
Yeah, the base. Yeah.
All right. I think we're good to go.
So have we collected all these? I'm just trying to catch up.
We have collected. Steve collected a euplectellid sponge, a Norella. There, I thought he collected. Did he collect a plexaurid? Oh yeah, a plexaurid with an ophoroid associate and an aplacophorine and a cub coral, are in a slurp. And then they collected an ophoroid that was off of the seafloor, an ophiocanthid. We collected a sea pin, and we collected that Chrysogorgia. It has a astroschema on it too.
So here's the question.
Iridogorgia?
What other?
There's a lot of Iridogorgia. Oh, we do have a piece of an Iridogorgia because
Accidental collection of an Iridogorgia. Yeah.
because
Accidentally?
well, it,
Happy little accident.
Okay.
Bob.
Yeah.
Do you have any requests? Any?
No, I'm just
Demands.
I'm enjoying the diversity. That
There,
we
it is
see.
nice. Yeah.
Yeah, it
It's
doesn't
been-
look very abundant, but it looks like, is it?
No.
It's not
at all.
super
But it's,
abundant,
but it's diverse.
but it's
and
pretty
only-
consistent
Yeah.
ever since when, Brian?
I don't know what depth, but
Yeah.
it was just steep draped sediment for a long time, and then we kind of had one little small high density community, and then it's been just an even, consistent distribution. But like you said, high density and low abundance since then.
But Deb showed that we were just looking at where we were, and we're like close to the top of the,
Yeah, should be, right?
and then it would be going along the top.
Yeah, we're about to
Yeah.
transition.
Ever seen Chrysogorgia?
A couple.
And the Chrysos or not?
There was one bushy Chrysogorgia, like a little plainer one
Yeah.
that there was a chirostylid in, and then there was, I think, Europtychus looking something on bamboo. Oh, on the hemicorallium. On one of the hemicoralliums, we saw it.
Is Europtychus on the hemicorallium?
I think so.
Wow, it's usually an oaf.
There was something. Well, no.
As well?
No, there wasn't very much. There was crinoids. There was a lot of bigger colonies. Like they were pretty big colonies.
Yeah.
So there was crinoids. There was
Yeah.
other stuff that I can't remember.
This has been the crinoid diversity dive. Stock
Yeah, there's
crinoid
been a lot of
diversity dive.
different
It is.
types of crinoids.
Chrysocrinus and all that
Or
other.
like, yeah, a lot of them.
Bryocrinus.
I don't know. the
Steve
last
wrote
one.
some names in there.
Gillicrinus.
Gillicrinus, Proisocrinus.
A lot of dead skeletons.
No.
Not a ton, no.
Mainly still standing, the ones that are dead.
It's like fun.
Mm-hmm. I would. Victorgorgia.
Is that what we're saying? You sample that? I thought I saw.
We haven't sampled Victorgorgia.
Not this morning, no.
Oh, I thought.
Oh, I'm sorry. I moved the can.
You're fine.
Okay.
I'm gonna do my sample. I'm gonna find out.
We've been seeing a lot of these. Is it of interest to get the star with the coral?
It's one of our
Okay.
Let's see what it looks like.
Okay.
Yeah, I think if it's intactum, then we'll just. It's fine. No. I've been. Right. Okay. Yeah, I'm gonna get back on that line. Right. I have to hit autofocus.
Can I get manual focus?
I got no. You just do you. I'm fine.
You just relax over there.
Yeah.
I'm just-
Don't worry about a co-pilot. You just enjoy whatever you're doing. Easy like Sunday morning.
It is.
It's, you know what? You have a kid. It's Father's Day. I'll give you this one.
Look at that.
Happy Father's Day.
Yes. I have 2 daughters.
Yes, sir. How you feeling?
Yeah. All right. I don't know. Go. No. Let's keep going.
Keep going? Okay.
Yeah.
Carry on.
Are the Victorgorgia this morph? Or they're stalk like that? I mean, it's a pretty thick central stalk axis, and then
Yes, they're not like the ones that are real
on the ground.
brambling. Yeah.
I'm looking for the squatties too.
You looking for the other one?
Yeah.
Looks like we have something, yeah, up there. Caligorgia.
All right. You need anything?
Nope. Thank you.
All right.
Nice talk.
I won't do it now. I'll get you a series of like little paperwork that has all
New bumpers? Have you seen them? Oh, you haven't seen how they attach one bolt.
Okay, I just saw them on the vehicle when we were down there, so they're horizontal.
I hope to not rebuild.
We don't need to set down. We can just go by it.
What's it called?
What's the coral called?
Oh, no,
Oh,
we
sorry.
were side of it.
Oh, I don't know. There's snap zoom and then there's tight zoom. Flyby. There's another Chrysogorgia.
Fly by that one.
Fly by that one. I'll just do a little zoom. Can we get autofocus back on?
Yeah.
Thanks.
Oh, there you go. There's some more in the background. You can look at those.
Yeah, let's go look at one of those.
You know the problem with me having this thin constitution?
What's that?
I can't not eat bacon.
I know. Like for multiple meals.
Like,
Because you get to
come on.
save bacon for breakfast so you can have it at midnight. That's scrambled eggs today. I'm excited. Oh, maybe pancakes.
Ooh.
Ooh, the pancakes were
Ooh.
fantastic.
Ooh, pancakes.
Pancakes and scrambled eggs would be amazing.
The joys of the 4-day watch, getting to end on breakfast.
That is a joy. But it means somebody has to relieve us like 10 minutes early so we actually get breakfast.
It's true.
Who's supposed to relieve us? Tim?
We never set an actual official watch.
You can do what we
What?
used to do
We
back
never
in the
set
day.
an official who's supposed to relieve us.
You just
For dinner, but
back in the day, we used to spend the ROV on the seabed, dial down, thrusters really hard,
Yeah.
still eat.
really?
Well, I
We'd
mean,
be
you,
eating the same dinner every when we got home.
I mean, you see the shrimp boats go to sleep in the Gulf where they just throw all their deck lights on and
Yeah.
just go to sleep.
This one looks quite different. The polyps are.
I think it's something with sea log right now that's being really slow. Because earlier today it was flashing.
Polyps are pulled in a lot more than some of the other ones we've been seeing where they're more like barrel shaped even when they're retracted.
So would you call this a Chrysogorgia or a Flexor?
I would call it a plexaurid.
Okay.
Well, I mean, I can only really put in it what we have. But
We
if you
did
went
like we
on something
sampled like a
different,
very
you could
quintessential
see, like,
um
many websites.
Acanthogorgia. This looks like Acanthogorgia to me with how dense the
There's the,
polyps
is
are.
that the growth form?
So I can
That,
do like
I
spread like
don't think it is.
we don't have anything else to send them from what the overlay has all the people that are doing that. O2 and CT. Like
Yeah.
it like there isn't anything at the front of it graphic different.
Can I have manual?
So it's manual.
Oh, it is?
Lay it
Come on.
different.
I'm going the wrong way, I can tell.
But like
The other day I didn't have my glasses on and I think I did a whole shift where I was like, I'm pretty sure it's in focus for me and out of focus for everyone else. Yeah, there's a lot of polyps on there.
Yeah, Iris down there.
No, I think we're okay.
What were you calling those two? Sorry.
I would say this looks like a plexaurid to me, but this is so many polyps that
They look different.
it's more like, yeah, they do look different. But I would want to know. You want to do a test for me?
Yeah.
Can you-
I don't be reaching.
Oh, sorry. I want to poke this one and see if when the polyps are trapped, if it looks like
You
it.
got that, yeah. Get a much better
It won't.
job than I thought.
It's flexible, so you could just brush up against it.
Have you been seeing the one on the left?
What?
Have you been seeing the coral on the left?
Yeah. Yeah. you go. There we go. Yeah, so see how long the polyps still are?
Yeah.
So I would say that's Chrysogorgia, and then the one to the right that we were looking at before.
The left.
I'm sorry, why do I keep doing that? The left. When they're retracted, they're almost all the way into the tissue.
If you think it's sample worthy for coral, I am interested in the sample.
In this one?
Yeah.
Okay.
I don't know how big that is though. That might be pretty big to try to get that out.
That's probably a black box.
Ah. Okay. Quiver. It's probably it's bendy.
Okay, then yeah, quiver would be fine.
Yeah, it's bendy.
You can manipulate it.
That's that way right on the way from east to west.
There's a picture over there too.
It's right on the upper part.
Yeah, we'll do it.
So let's do this. We're gonna sample this one.
Okay.
We're gonna just sample pretty much the whole coral. So just down.
No, we're just shoving it.
It's bendy, so do you think you can get it in a quiver?
Sure. Quiver up.
Thanks guys.
You're welcome. Verify that, or
There's also a picture in the back.
Yeah. Is that what you're talking about?
Squatty, yeah, right?
But is that so? I think that's just a young colony.
Yeah. Okay.
You turn what off? Everything, turn it off.
I'm interested in what that is out there because that's a different morphology.
remember from the last time, it was my first time sampling corals. Base?
Yeah, that's good.
I would grab some, they shatter. A little bit disappointment on Eric's face. Where this is fixing that happened. Good. Sorry.
Really? He gave you a look?
Well, I don't know. I felt it.
Oh.
Maybe I was just projecting on it. Watching out.
So you want to just kind of jam this in straight up and down?
Yeah, it should go if you, or if you flip it upside down and kind of just like.
I might, try to grab it from the top and then drop it in.
Okay.
I think because then it bends, all the things bends up.
Don't
Yeah.
put another bit of coral in there though.
It's from the what? It's the piece of Chrysogorgia that's sticking out of.
Do we move that first, or is it?
It's okay. It's sticking out of 14.
Okay.
Or it was. I don't know how it's over there now.
Yeah. It's like down.
Probably.
You're just trying to make Alexis feel better about not knowing how to learn.
Nicely done, Adam. That was awesome.
3:15.
Breakfast, so if you guys
3:15.
want pilot change from Ben counts, just put the watch change.
Alright. Flex and 3:15.
Big
Flex with both.
All right. And next we're gonna go look at this coral over here.
Okay.
Not that I want to overload us, but just making sure. That looks like Swiftia.
Sample.
No, we already sampled one. Just take a look. Yep, that's great.
What kind of is 15?
Plexarda. Okie doke, we keep going. Got some big sea cucumbers over there. Alright, Adam, what's your suggestion for the new snap zoom term? Okay, okay.
It's gotta be something snappy.
It's gotta be, got to.
Zoom. This is really some stuff.
Okay. Another big sea pen, like what we saw.
Looks like the Texan spotted
These are definitely the biggest sea pens I've ever seen.
Yeah. We saw a couple like these, I think, in Pippa.
No, it doesn't matter. 8. 8
Versingid, lots of echinoderms, crinoid. Versingid. So another parid. I'm assuming now. It's always tricky. But yeah, I think that's a plexard. Everybody's talking about bacon. Now I'm hungry.
Can't smell the bacon wafting through the ship like we can on some others.
Which, thank goodness.
Yeah, which is probably a good thing.
Yeah.
Yeah, I try not to eat until breakfast, even though I want to shift it tonight.
Oh my gosh, it's a long time.
So fun to do whenever I'm like checking out my barbecues.
Oh my god. Do you guys smoke bacon? Is some of the bacon smoked?
Not
No?
by us.
Okay.
Pork belly the other day was smoked by us.
Oh, was it? I could smell, one of the bacons they have is really smoky. I was surprised at how much flavor there was. It's probably because it's not from the United States. Chrysogorgia. take a look at this coral, Chris. There's something at the base of those branches.
I guess those are percentages behind it, but they're huge.
Well, and they're only 5 armed, which is weird.
Yeah.
Oh, it's just a big gall.
You can fly through that?
No, I did. That's you.
I don't think we've seen any corallivory here.
No.
The only even like damage we've seen was that couple hemicralliums.
Yeah.
Those are crinoids. Recipients.
Oh, wrong way. Oh,
That light out of there, so it won't be.
yeah, it's the. This one appears pretty clear, actually. That's good enough. They are crinoids.
This is more crinoid diversity than we've seen any other time on this expedition so far.
I should, shoot, what's the guy's name that studies crinoids down in Florida?
Chris, Chuck Messing.
Yes, Chuck Messing. Okay, that's good. Thank you. Later.
Is that an anemone?
We're all still there.
I think it is.
Down the rock.
Oh yeah, a couple anemones, it looks like.
Should be out of there.
Yeah, I'm getting, yeah.
Nice shot.
Couple anemones.
Just hold that for 20
Yeah.
seconds. Just kidding.
You do see some abrasion around that one anemone, but it looks like
Yeah,
there's some tissue loss there.
or the hydroids are growing. The color is always so great. The purple is always so vibrant. 1, 2, 3 Victor Gorgias, 2. Primnoids. Okay, we can keep going.
Yeah.
Been on up.
Well, it's not your best.
What about the one we sang last week?
Let's buy a flash sale. Uh-oh.
Land vehicle. I should say it on my phone.
Bridge ROV. That's an updated waypoint for you.
Bump it up.
Thanks, Tony.
Is that Metallogorgia or is that a?
I think it's a crinoid. I could see it when we were sat down. I don't think we've. Oh, Steve said they saw Metallogorgia deeper, but we haven't seen any since we've been on.
It's about time you get it off. I just like being in the house.
Another flying urchin.
Whee! They're surprisingly,
Buoyant.
yeah.
Receptive to my thrust.
There's a
Little
nice.
sponge for once.
I think they sampled a sponge earlier,
You're
right?
right, they did, but we've seen like
We've seen like
there's
three.
been some small encrusting ones, but we've
Yeah.
only seen like two or three bigger ones.
Euplectellid. Has the screen over the top, which means there's probably some shrimp inside. Let's see if we can see inside. I always like find myself trying to see over and under things,
Yeah.
even though that's
Sitting
impossible.
up further
Yeah.
in the chair. Yeah.
Yep, there's a shrimp. I'm assuming that's what that is. Yep, does look like one. Yeah, maybe the other one's in the basement.
It's crazy. It's like a net over the top.
Yeah.
Looking way down, I can't tell.
There's another.
There is a net over the top. That's how the shrimp stays in there.
It's trapped.
Yes, there's normally two of them that are in there their whole lives. This is your first time hearing it? I feel like we say it every time we see them, so
Well, I always see them in. I didn't realize it was trapped.
yeah. They go in when they're plankton or like really small and then they grow up inside there and then
We died
what?
I died 427. Or you only been here for 413 of them.
It's another plexaurid, I think. Sea star on the sediment. Big crinoid. Ugh, yawn, sorry. Oh wait, is this your little, let's look at this.
Yeah, it's like
I distinctly remember trying to sample that same thing in 2017. It's right here.
Yes. There's, playing with the cameras and get blown away. Oops. Not quite yet. I can go around the other side.
Okay. I think it's worth seeing if it's the same one. I don't know if it's the same.
Try to grab it and see if it runs away from you.
The other one I think was more red.
It was definitely more red.
Okay. Mm.
Wanna try it?
Yeah, let's sample it. Do the same way you did it before. Where you just kind of pull it up. Yeah.
Jaws?
Yeah.
With the soft jaws?
Yeah. Did you get a picture? 1 second.
Yep, we're good.
Okay, we're good.
Hold on. See it?
Where is it?
See the shadow. Oh, it's right there. Pull that rock. Get right in your shadow.
You get a seat pin?
Mm-hmm.
You know what it is?
Do what?
What do you call this one?
A C-pen. Steve said we don't have samples of C-pens from 2017, other than the Antebellum.
Is that how you say it?
No, I don't know.
Little podium?
Is that it?
I don't know. They collected one already on this map.
No,
Collected.
we collected one, but the
They
one they
attempted.
tried to collect.
Was it like this one?
It escaped.
No, it wasn't the same.
It
The one they
escaped.
tried to collect sucked down into the sediment.
Yeah, the two you collected
sorry.
aren't as
Yeah.
special as the one that I tried to collect.
Way
It's like
gone.
just a regular
Way
sequence.
No way.
Oh yeah.
Like run in the mill.
They dug around for it, they tried to find
This happened,
way
and it
gone.
happened in 2017 too, where we tried to collect that same one, and it retracts itself.
Into this.
Yes,
Vanished.
like bats. Like,
Watch the not see
We'll play the video.
yes, it is a C-pen.
We'll watch the video. That's Nat Geo.
That's Netflix.
I mean, it was gone. Like it was just a watch chain. We walked.
Yeah,
in,
we
and they were
walked-
trying to get it, and it just vanished.
It happened to me in 2017. I remember.
I'll be back in a minute.
Okay,
316, right?
316, yeah. And quivered 10. Nicely done.
That's Eric playing Randy video.
Deb, can we have the chat back?
Oh yeah, I was gonna ask
Oh,
that.
yeah.
That means.
What? What, Eric? What's Chris? What?
Great, Great, Chinese
Yeah, apparently I
watch.
can't push buttons and talk at the same time.
Push all your buttons and then we'll get back to you.
Oh, yay. Push all my buttons. Push all everybody's buttons today.
You do what you gotta do.
What was your question, Chris? Now that I know your name?
So if Randy's over there watching videos,
Mm-hmm.
that means that the meeting's over.
Yes. Well, no, just the science part of it.
Okay.
They kick the scientists out. They're like science meetings and they kick MTs out.
Huh? I know.
Oh my God.
It's another big glass sponge. We don't need to stop at it. We'll just keep going. Did I write that down? Yeah, I did. Okay.
Wait, are you on this?
Well, kind of. I'm an association with the machine, but yeah, I'm in our shop. No, it's a VM Linux machine. I don't know.
Now let's, how about just PSP bucks?
Is that X? Okay.
I think it's just the being able to settle into it, you know?
Yeah. We're actually gonna go pattern.
You see only again across the steep.
Do you have to do like a
More
cap?
steep? I think we're hitting like where we're gonna go up and then be on top of a
Yeah,
thing.
because we're just narrowing it down. You can also process it by what's taking us.
Maybe there's not supposed to be a space
I
out
think
there.
you know you need to have space. I don't know about more, but for anything for search terms. But oh, you gotta do grab and G, right?
That's what I was like, cat or grab or whatever.
Oh, did we forget G rep
Yeah,
last time?
yeah.
I was doing
Like
my
this?
head,
Is
yeah.
that better?
Yeah.
There you go. So there's all your sea level stuff. And then if we look over to the left, we can see. Uh-oh.
Okay.
Oh, wrong machine.
I was like,
I think maybe changes something over there. And I don't know.
Okay.
Yeah.
Do this. Up arrow. Pushing button to talk it. Remember, we just had a conversation.
you gotta pick one.
Type sea log. Sea log lots of process.
I kind of want to look at one of these are, cause they're are those? Yeah. Well, next time we,
I already did.
they're kind of all over. So just pick one.
Okay. Right there.
Found the plan. What machine was I at? That one. So I was like, okay, we don't want windy.
So do the same command, but if you were to type on 3 and just type sea log. Oh, you're gonna get all those too, because all those are sea log.
Yeah. So it's really slow.
Push down a little bit.
Okay.
Or sea log, like it wasn't really flush rate wasn't happening. and that's
I'll do it.
like-
Yeah,
Either one or both.
You wanna go first or?
You can go.
Sure.
Yeah.
What's it?
No, I didn't get to today's sea log.
Sorry, are you still moving?
I'm just putting it out so you have a review.
Okay.
How's that?
Good.
Where's it gonna go?
Jeez, Pete.
Just relax.
Just relax.
God, just relax. Like a jumping spider. What are you doing?
That was loud. I want to see what. There's a bunch of these little patches that I'm just curious what
Oh,
they
hold
are.
fast or sponge? Or
No, they're like sediment all clumped together apparently.
That's what it
Real
is.
exciting.
They drops it in out here. Yeah. Look at that current coming out.
But there's like a lot of them. Look,
Yeah, I know.
so something's doing that, but I don't know what.
See MongoDB to from the database to this. It is a big this is, no, none of this is from
Okay, rest. We can keep going.
Probably goes
We're just
into
going
a
up this
stand base, but it's an influx.
rock face.
Okay, back to this.
It just got much more steep.
Wait, I was just.
Much more Steve's good.
You can do, no, you can do the same thing on top. So,
You want me to log?
and then go top.
That'd be great.
Okay.
I mean, really?
Yeah, yeah. We can switch, and then when Brian comes back, I'll go eat.
Okay. Well, hopefully someone else will show up.
And you can, all you have to do is
I'm gonna count on that.
All right, hello.
yeah, so now.
Hello, Russ.
Probably this way.
It's a nice warm seat here. Look at that. Settle right into that.
Nice and toasty for you.
So yeah, almost better to go the other way because it looks like it took off. Yeah.
Heavier, must have picked up more rocks.
Did you? Did you pick up some rocks?
Well, it must have done.
Looks like we did, yeah, a little bit.
Quite as light.
Yeah.
And then like with this, yeah. This is.
Okay, so let's get organized here.
You adjust the camera, you like to see less.
With me.
Stuck in there.
Oh, look.
What?
I already know it's
Ninja.
What happened?
Ninja mouse.
Call it out.
No.
I'm not gonna watch till 8, so.
Oh,
No,
okay.
I'm just kidding. Have you seen these?
This is
It's
a bamboo.
whip.
We have seen some earlier. We haven't
Uh-huh.
seen too many recently.
Right. Anything on them?
You can
What do you
pan
mean?
up.
Yeah, but then they'll lose, like,
Or-
in the middle of the log. I mean, it probably would go pretty.
Nah, looks like it's
I mean,
just
that's
a
the stage, I mean, I could, yeah.
just a bamboo.
I could log into the EEZ side of it too, but.
Yeah. We can keep going. Don't see anybody on that. Just
I'm just gonna read the
You checking out your social medias?
yeah, I gotta check on my status eye on everything.
Earlier today was working beautifully where this would just refresh. But even I
No, I'm just we have a hi, good morning everyone. Happy Father's Day. This is Tim Shank, a deep sea biologist at the Woods Hole Oceanographic Institution. I'm here with Alexis Weinig from the USGS, also a visiting investigator at the Woods Hole Oceanographic Institution. We're currently at 1419 meters and going up the side of this really nice seamount west of the Tokelau chain near the equator. 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 It sounded really different when I said it. Just said. Yep. And I think we've been seeing these corals before,
Yes.
so I'm not stopping when we see something we've already seen before. We've seen that one before.
I mean,
Tell me.
possibly, yeah.
Possibly.
Possibly.
Isn't that the one that we saw down low next to the other?
Yeah.
yellow?
It's hard, like those
The fleshy one?
flexars and acanthogorgids are hard, so you either have to decide to zoom on everything
You
or
do.
just kind
You
of
do. Right.
like
And I know we have a lot
make
of
some
track.
assumptions. Yeah.
We're going up top of this top of a ridge here, and then we're gonna turn left and follow up the ridge. Have you been? I know
We've
you
seen-
saw that.
yeah.
And have you seen?
Yes.
Oh, that's a
We've seen a
that's
lot
the
of different
price.
types of crinoids.
Chrysogorgia. That's what it was called.
We've seen those isocrinids, the big red stalked ones, and there's that other
So let's
sea
catch
pen.
it.
We don't need to
Is
sample.
that a rock
but-
pen?
Oh yeah, that's a rock pen.
It's a rock pin. And
Oh, I
yeah, I'm just looking for different things. I'm sorry, I'm catching up on notes from previous watches. And
too many notes in there. Everybody's sleeping.
what's a Gilocrinus?
One of the crinoids that we saw.
Proisocrinus. That's right. Rubrismus. I can never say that right.
Which one?
Rubrimmus. The big red stalked crinoid
Yes, yeah.
is, we're calling that Proisocrinus. So now that that's done, I can put that away. All right.
But you know, everybody's gonna wake up during your watch, and then there's gonna be all the chatter in the Slack that you gotta keep up with.
No, I know. I better keep the Slack open. Hold on.
His technical assistant over there? IT support.
IT support.
Absolutely. Gotta keep it
Alexis,
close by.
You've just gone from a coral expert to IT support. In one watch.
With no real IT background ever in my whole life, except just being
Social
a
media.
millennial instead of being what?
Instead of being
gener-
what? I keep going. Keep going. Keep going.
generation are you?
Neolithic? Is that what you were looking for? Neolithic?
She's an ancient coral.
Golly. All of a sudden, a Cro-Magnon man or whatever. Oh gosh.
Trying to tackle these things. Logging's hard. I haven't been in the
It is
logging
hard.
chair that much.
You want to slow down? You can
No,
slow the
no,
vehicle
no,
down.
no.
Just slow the whole cruise down, just so you can get. No, it's not easy.
It's not.
Not easy. All right. All right.
Almost at 1,400 meters.
You tell us. Let's look at the.
Yeah, we're kind of turned
Let's look at the
direction,
map.
so we should be at the top.
Oh, really?
Waypoint 4 is
Hold on, I'll put up,
15.
1, 2, 3, 4, 1354. So I'm
So-
thinking about 15 more meters, and we did see this Chrysogorgia morph
Yeah.
before, and imaged that well. We got that. Hope I'm on the right dive. Okay. How we doing? What's this? Have you looked at this? You see this?
You can take a look at that.
Can we take a look at that? Do you? I had to call in some sort of a sit down or not sit down fast.
Oh, yeah, we were talking about
We'll just.
the
I think
new.
we can just.
Adam suggests we need a snappier name for Snap Zoom, so that's on everybody's agenda to think about.
I just. Yeah, I just wait until the. How it's got it set up, and then.
What's that up there? That little thing. It's
So
just,
this is-
oh, it's a siphonophore maybe? It's so,
So this is like a Primnoid,
yes, it's
Yeah.
a Crinoid.
and I can already see it's got a spiny arm ophiacanthid type ophiuroid at the top, which is
Okay.
Yep, thank you. And lasers are on. They're 10 centimeters apart, so not a huge colony, but there's something else here. Can we come down with the irisoscope?
It's all the way down
Or-
there.
or-
It's not moving anymore. It's
Is that
just
right? Maybe it's other lights are on.
Yeah, is that a little better?
Yep, that is. It's great. All right, Emma. What do you think? So that's the Astroschema
Yeah.
ophiuroid in there.
And
Looks like intected, and these, yeah.
Have something sort of sticking
And you got downward-looking polyps, like Crinoids have. Right?
yeah. I think what we're seeing is a type of Morella, but I'm not great
Yeah.
at those, so.
Oh, that's great. That's great. I think that's two of those O's that are like the ones that got the black-legged. I don't know why I call
The
them
what?
black-legged.
A black-legged?
Yeah, it's just easier to say than. Okay, great, Russ. Thank you. So we took about 12 samples since we are on bottom.
Did you count them?
Well, yeah, we started at 304 and we're at 316. What is that?
Oh, yeah.
It's somewhere
No.
somewhere
yeah,
around 12.
13.
Yep. Okay. Have you seen any predation?
No, no. It's not,
No
we
corallivory
were-
today yet?
Yeah, we were saying, Brian and I were saying it's the only like kind of unhealthy branches we saw were on the bigger hemicoralliums we saw, but there wasn't any evidence of anything
Yeah.
eating them.
Interesting.
But no like snails, no. We saw an aplacophorine at the base of a Chrysogorgia, but
Yeah, I
it's
saw
that
that.
thing where
Right.
it just rocks around.
Yeah. No. Interesting. One of our main objectives out here is to look at predation on corals and the immune response of the corals, and the wound healing response of the coral if there is any. So we're on a careful watch for any corallivory events. Oh.
Yay, thank you.
Oh God!
Withering away.
All right. Thank you.
Yeah.
2 seconds. I'm just gonna do a quick,
Roger that.
Happy Father's Day to my dad, Mike.
Yeah.
In Hinton, Alberta, Canada.
Hinton. Happy Father's Day, everybody. Nicely done. It's well written, isn't it? All right. I can log, Brian.
No, it's not. I
Okay.
just ate.
Yeah, it's okay. Whatever you. There's a bamboo whip. Looks like it's just bare skeleton. And make sure there's no predators on there. Tilt up a little bit. Got kind of steep here, didn't it?
Yeah, just on the side.
Just over the top here, a little bit, right up, slow round.
1387. So I am just at the moment passing by corals. There's it's like a plexard with a astroschema and a anemone. It's a ring anemone that often lives inside these along in the branches of the coral. It grows over the polyps.
Morning, fella. I'm gonna start moving your waypoint to the southwest there a little bit. When you get to the line, you can start making your way down there, if that's alright.
That sounds good.
Great, thank you.
So the top we had at 1354. So we're up. Is this predation? What is this? What? Say, look at that, Russ. I'll stay where I am right here.
Oh, we're gonna stop and look at that.
Yeah. Well, there's
I mean, that's just not
astroschema
great.
oath on there too.
Yeah,
Look at that.
that's a lot.
It's a bad situation.
Yeah.
So we have a sea star here. We do have a situation. I think it's Seriaster. I don't know. It could be a Hipposteris. Looks like a Hipposteris sea star on a bubblegum coral, on which the stem of that coral, the central axis, has a astroschemated ophiuroid. We're going to have to get us for some tight shots here. We're going to want to look at the underside of that sea star to see if there's a stomach's everted. They often invert their stomachs and.
I'm gonna come down and you can move the camera.
Yeah.
Just-
Right. Okay. We should sample this.
I think it's probably likely.
I think we should. Yeah.
We probably should.
We'll get some really good shots of it, and then we can collect it.
Okay,
Yeah, go ahead,
I think
fella.
we'll stay over there. You
Good.
Take the camera.
All right.
I did. I just brought
Gotta
you
get the light down a little bit. I think.
Yeah, I can
That's
use
all.
that.
Thank you.
right now.
Alright.
So we'll get the lasers on it just quickly for some. We'll hold a minute. There you go. Great. Super.
There's Otto there too.
That's auto?
Or no, it might be manual. Try now.
Yeah. Manual. I think.
I don't see.
If the polyps are pulled in.
Yeah, the polyps are pulled in, but I don't see any signs of the sea star's stomach necessarily. The tube feet are
moving and
actively stuck on it.
Right.
And
Of course, I don't know if there's a polyp, inside, or.
True.
But you're right. I don't see the
Correct.
stomach outside.
But look at the coloration down. I don't see any really denuded tissue. Like the coloration's a little lighter, but I don't see any actual damage yet at all. Yet.
Look how it's perched on there. Let's just hold that for a little bit. I'll make some notes here. I know Randy's probably watching. I was waiting for her to come in the door too. Do you see
that
the
little crab looking down at the very bottom of the screen?
Yeah, there
another
is.
Little
There
crab
is.
Thing.
It could be a Umunida, I don't know, but yeah, it's monkey Umunida. Do you see the oaf arm going on there?
Yeah.
You wonder how long that oaf is going to stay on there. They're often very specific relationships. This oaf is not specific to Paragorgia, it's on other things. But they seem to spend a great deal of their lifetime on these corals. No, not Randy. All right, well, so.
Yeah, I'd probably just recommend reflect the whole thing.
Yeah, I agree. Anything else we should look at?
I don't think that's full zoom.
That's full zoom.
Yeah, I don't think
we're all the way in.
We're gonna get much. I don't think we're gonna get much better perspective. So.
No. Okay. We're gonna want to collect that dual quiver. The whole thing. It's pretty small, right?
Oh, okay, it's what you said, yeah.
Over here, watch. I mean,
No, I'm still working on it.
the lasers are. Thank you for hanging.
No worries.
Should I borrow your camera for that?
Yep, it's yours.
I have to move it back 'cause it's
Yeah.
kind of far away. It's
Right.
sweet.
You can see the expanse of the seafloor here, right? And it's amazing to me that these ophiuroids, for one, can find these corals that can be very small. You'll see how small this is in just a minute. We'll go to collect it, and then how a sea star ends up finding it. So there must be some sort of a chemical signal or signaling given off by these animals. That's mine.
Oh, that's yours. Okay.
Yeah. Thank you again, Brian. I'm just gonna. Yeah, I'm charging. Wait a second. It's on charge. It's good. No, I'm gonna put this back here. Thank you very much. Awesome. Yeah, this should be sample number 317. 317.
You should just pluck. Should do. Place them all.
So we may want to put the cutters on the other side. We want to go for the stalk of the
Yeah,
coral.
you don't want to cut it out. So yeah, see the slight impact is to knock out, place the bottom.
otherwise she's not gonna explode, you know. You gotta cut it.
Most likely. See, we don't want to damage the sea star, so you get a finger on tree. I'll zoom in.
You can put the cutters against the rock kind of thing. Then. Yep. No, miss. That's okay. That's okay. That's all right. You see? Yeah.
Do you know if that's a Paragorgia or Unicorn?
Yeah, Paragorgia. Yeah, sorry.
No, it's
And then sea star, Goliaster sea star, and Astroscala.
that's not right.
It's going into. That's fine. Yeah, that's good. That's not.
Wait,
Yeah.
because we're gonna go and get the sea star.
Yeah.
I think I can get that. Yeah. Okay. Let's see if I can do it.
Always almost.
Disrupt things, he didn't.
See it on that one, but it's hard to see this.
Yeah.
Hold on.
There's a little ledge here.
Hold on.
I don't get my hand up. Lots.
Okay, stabilize. All right.
Should be pretty robust.
Which is set as well. Back.
Nice.
Nice and gentle.
Think it will fit?
Sit right there.
Same quiver.
I would say so.
Yep.
Go check the arm. Okay.
Am I hitting the box?
Same quiver.
That's fine, then you can come down now. Set's got good view. Close.
Good. Great. Good job. So we're just saying we haven't seen any corallivory yet today. And there you go, as soon as you say it. It's like when a field goal kicker's never missed a field goal and you talk about it. He's how he never missed a field goal and that's when he misses it. Anyway, it's good to see that. It was amazing. Stomach was not everted on that sea star though. But it was interesting to me to see that when after it fell down, it didn't change its shape at all. It was like it was rigid in the feeding posture. Interesting.
Oaf was on there, or the astroschema was on
Oh,
there.
yeah.
Pretty good.
Wrapped around really well, getting up off the seafloor. We see that species of Ophiuroid on several corals, on this dive even, so it's not very specific to its host. So 1381 meters transiting upslope to the top of this this feature that we're on that's going to have a ridge at the top. We're heading west at the moment. We're going to turn and
That was good.
head south. Yeah. Do you want to see? Can we see this, Russ? Sorry. There's some action going on here. Looks like there's a couple different oafs in the anemone, but I didn't know if it was some damage.
Yeah, let's check
Here
it out.
we haven't seen a lot of.
I know.
That was our first corallivory.
I know. I was looking at it downstairs and I was like, "What?" So much for the. I was like, huh, the oaf. The oaf isn't protected. Did you see how hunkered down that oaf was?
How what?
How hunkered down the oaf.
Oh, I know. It didn't change shape even when we got it.
No.
Can we just bring the light down a little bit? Thank you. So I don't know if they saw that this morning already or not. We're 8 hours or so into the dive. And yeah, it's good. I'm going to come up and
Happy
that.
Father's Day, Tim.
Thank you very much.
Happy Father's Day, everybody who's a father.
So I don't know if we've seen these. We'll document these. I don't know if we need to collect them or not. Octocorals have 8 tentacles. These have many more, as you can count. That one in the middle there has, I can't count them from here, but much more. And that's maybe all I don't see any other damage per se. But I wanted to document that. That's great. Thank you.
Sure.
Yeah. Onward. Come up a little bit. So yeah, I'm just sort of passing by things I know we've documented already or sampled.
We should grab a good Crinoid though.
Like a big
Well, one of
experiment Primnoid?
We
Is that a Primnoid?
yeah.
Yeah.
I feel like we could use a couple of big branches. I don't know.
It has a lot of stuff on it.
Yeah.
Wow.
I
It's
can
not
just
gonna
do
be
it. I'll
here.
just do a half
This is a
zoom.
hard place to be, but
Just a half zoom here. Yeah, there's
Actually,
there's
with
bigger
the-
ones and better ones.
let's get a better one.
Tell
There's
you
plenty
what,
of better ones.
It's not too bad on the walls. I've got
Okay.
2 feet that I can put on.
Got it.
Some places easier.
There's been some larger ones. I was trying to see if they've collected one already or not. They've done a Plexard,
They did, but I don't know
Chrysogorgia,
how much they took.
Vittigorgia, Narella. Yeah, they do have several branches of Narella, it says.
Did you check the experiments this morning?
Yeah.
How did they look?
Good.
Same? Same.
It kinda. The graphs are the next one.
This is hard to tell. Yeah.
Yeah.
I don't know. I can't tell.
Yeah.
The other thing
I'll
is
break
that
it down this morning. What?
you may want to get your Primnoid, toward the end of the dive
Exactly.
more, I don't know where we stand on that exactly, but it's a thought.
Just speaking of which, I think we should, click.
You want to click?
Click.
Oh.
Great.
Oh,
Sorry.
sorry. Yeah. So we've got 21 meters to get to the top. Or we can turn and run along the ridge.
It is amazing how sparse this is when it is looks like the perfect slope, perfect rocky bottom.
Yeah.
I don't know.
I mean, we're seeing corals. It's not like we're not seeing corals. Diversity, the abundance is not high, but the diversity is
high.
pretty good. Yeah.
Did you guys fire any Niskins yet?
Negative.
Yeah.
No.
You should grab one, just as a.
Whoa.
Whoa.
You?
It's huge.
That's awesome.
We've
Alright.
seen a couple of really long bamboo whip coral.
Yeah. Branched or unbranched?
Unbranched.
Unbranched. Let's take a look at this one.
Singles
Russ.
like this, and there was one that had a sea star on it, but it was too long. It was the sea star was stored for bottom, so
I want to see.
Steve didn't want to take the whole thing.
Okay.
That makes sense.
Yeah.
But look at this baby.
It's got polyps.
It's grazed.
I'm gonna sit you down just in front.
Yep.
It's beautiful.
Sounds
Or
great. I won't
damaged.
touch anything.
Nice and simple, nice and slow.
Yeah, it's beautiful. It's got some bare skeleton on it.
What's at the note up
Looks
there?
like-
They seem to just say a pattern change. Second note.
It's got ophiuroids on the bare skeleton. It's got hydroids on part of the bare skeleton.
I will be in the other room, but if you need me to shout, I'll be on Slack and watching. I'll be listening to.
Okay.
Okay. I'm gonna
There's the aster.
Work together a proposal.
Roger that.
Okay.
Yep, ready?
Just your legs in there.
We'll go and look at these polyps while we're here.
What is that for like?
Yeah, it's pretty good.
Yeah.
It's maybe a SCO shot. I don't know.
Okay, it's gonna be I understand a little bit.
And there's the black node. You see there, the band of the coral, making this a bamboo coral. Polyps are pulled in. There are the hydroids. They have stinging cells, and they can do really a lot of damage to the coral. They can decimate a coral, frankly, if they really get well established. Some of these bamboos have crabs that live on them, very specific species that live on them. In fact, a new genus was recently erected for a crab living on a bamboo that we have here in the Phoenix Islands Archipelago. Let's see. Just trying to see where this branch goes off. The diagnostic characters for these bamboo corals, I think I'm all the way up, or where they branch, how they branch. These long whips especially. But they get really confusing sometimes because they'll branch or do something different if they get injured. So you have to be careful. But yeah, it's a long branch. All right, we've got that. There's a lot of work being done on bamboo corals right now. There's a big paper going to come out soon. Trying to have them all organized, so I don't know exactly who this is, but we've got an image, so thank you.
Do you want to fire Niskin?
Yeah, we can do a Niskin here. Why not? We're close to the top. Depth is 1365. 19, 17. So this would be 318.
Do you want a 5 liter or 2.5?
I think a 5, please. and see what he ends up with. So we're collecting a water sample right now using a Niskin bottle. That, we push that little button there and it closes the end caps on the bottle. We're just checking out to see if it can confirm that it actually fired and collected a sample. And we'll use that for looking at environmental DNA. So yes, so Niskin number 1 sample. That is our 318th sample of the cruise. Great.
It's actually got a foot branch there as well, man.
Yep. It's, and that's all the live material really is on that end down there.
Yeah.
So it's
Just saying, like,
it's out, yeah, it's outpacing the Grim Reaper. Right. Awesome. So are we coming back down now? It's just a momentary thing here.
yeah.
So the question is when to turn it and run. Is this the ridge we should run along?
It's
So it's gonna come down and come back up again.
60 meters out of chart position.
Yeah.
Nothing to say to that, isn't it?
I'm just looking at the sonar too,
Yeah,
if there's something up,
sonar.
coming up forward more soon.
It's a nice spot. Just come down ahead, you see?
Yeah, might as well go out there and explore out and then turn back if we have to. I don't think the ridge would be that sort of localized, right? So it's small.
It's only like a spot.
Seen a few of these euplectellid sponges, one off in the distant center screen there. I think we sampled one of them. They often have crinoids on them or a shrimp inside. Here comes what yellow plexard is what I call it. It's got anemone on it, ophiuroid, which we've seen today. You can see those little white, just beyond that sponge, you see two little white things in the coral. Those also will be anemones. And that yellow plexard. We sampled something like that today already. Is it me? It looks like it's sloping away from us, but our depth is.
We're increasing. Yeah, I think the ledge runs like that,
Yeah.
and then we're running up the center, so it's
Okay.
definitely going higher at the moment.
Yeah.
Just looking at the sonar,
It's
you see the hits ahead.
Yeah,
So they're
yeah.
high hits. That's high hits.
Yep.
Stocked
So we're good.
crinoid, plexaurid,
Okay.
Iridogorgia. Are those Crinoids in the background?
They may be primnoids. You want to stop and look at them?
Only if
It's-
you
I
think
would-
it's big enough to collect.
Yeah,
so,
I would-
so your call.
I think we're going to be maybe 4
Get
more
another
hours
one.
into the bottom.
Okay, yeah. Then
It's
no.
better for respiration. So Iridogorgia is here, the smaller one. Don't see anything living on it or in it. This is a really heavy coral ophiuroid
Oh,
place.
Metallogorgia.
Each one of it where? Yes. Can we go look at this? No, that's a crinoid.
You think? Yeah. Yeah, you're right.
It's a isocrinus.
It's a big
That's it.
stocked crinoid.
Got me
Sorry,
all excited.
I
Oh my
know,
god.
I'm like, oh,
I don't think
shoot.
they've seen a Metallogorgia on this dive yet.
The one where
where it's like coral at the end of the stalk like that? We did it.
No, it's a long central axis and then a poodle.
Not
poofy
a
little-
round, but like a flat thing of coral. So
Yeah.
where
I mean-
it was coral at the end of a long stalk. Like
Yeah.
that, like flat
Was there
thing.
something inside it?
I don't know. I'd have to go back and look. But I know we
No.
saw.
Yeah. Yeah.
There's quite a few ahead of us.
A lot of bamboos.
Oh, yeah.
A lot of whips out here.
The biggest city there.
Treasures for ROV thrusters.
No, it's like pressure's on now.
Gotta run the gauntlet.
It's like, oh, Dow Jones.
Another crinoid. Yeah, we've seen Paragorgia and Helmering coralliums, which is on the left here, bottom left, the red.
I think that's the
Coral.
array.
Yeah.
Hard to say.
It's hard to say. You can tell.
Without blowing it.
I think it's a Paragorgia.
I'm calling
Yeah.
it a paragorgia.
Yeah.
I'm just taking this video.
Yeah. We can sit down here for a second. What's that? Well, she's doing something over there. Yeah, we'll just do this. I've actually got my eyeball on something that's in that Iridogorgia up there. Yes, Deb, that's it. That's Metallogorgia. So they did see it earlier. Wow.
There you go.
Wow, that's pretty impressive, Deb. So we're gonna zoom in here to this, what I think is a bubblegum coral.
It's got a lot of like free oaks or two. There. It's just
Yeah.
Oh, we also got a different kind of.
So, what's that?
This is gonna be just a big version of the sea star in the ice.
I think that's a corallium. That's really interesting, though.
Are they all over today, or some of those astroschema? I can't really. They all look like this.
Yeah, they're all. No, they're astroschemids.
They are. Okay.
Wow, I don't know this coral. I mean, that's not a first or anything, but I have to think about this for a second. Let's get images. So normally we tell the hemicoralliums from Paragorgia bubblegum corals by looking at how stiff the skeleton is. You can poke it or you know, just put some current on it, and if it waves bendy, it's a Paragorgia. If it's the hemicorallium, it's really solid and brittle.
I can give it a little.
And this,
something on it.
yeah, sure, but this I just can't tell.
RV doesn't do this.
Nope.
And is that sea star something? We collected one of those earlier today.
Yeah, I saw that. That was great. You got it.
I can fan it.
Yeah, I don't know.
How shifted.
Let me ask you a question. Let me get the lasers on the top of this for a second to see how big it is. If it's quiver sized.
I think it may be a little bit.
I thought so too. That was my.
You can take a piece.
Yeah, but you wouldn't get the ophiuroid, right? You wouldn't get the
You'd have
poster.
to
You'd
go
have to
right
cut
in.
here. That's what I'm saying. You would.
Yeah,
right?
right.
All right, let's get a piece off the top of this.
Okay.
And go for quiver. Don't worry about the ophiuroids. You got the camera? Yeah. All right, we're gonna sample this.
First, Lisa.
Those polyps look
I
different to
agree.
me. I don't. So
They're like,
if you
yeah,
really don't know, let's go for it. I don't want to.
coloration too.
I don't want to pass it up. Deb Smith, our marine tech extraordinaire, is unbelievable. She went back and found images of things during this dive. Was that during this dive?
Yeah.
That's fantastic. See,
I don't think
we're
we sampled it though. It's listed as a pre-sample photo, but there was a confusion about that button being used.
that's Metallogorgia, and after the sampling, I'll talk about it. But that really is extraordinary to be able to call up images from the dive and look at them and on our screen in front of us. Really phenomenal.
That's the PRIMNM edition.
It's really fantastic. We have a pilot and a co-pilot and a marine tech sitting here all to our right, doing amazing work here. Watch them do it here.
Do you think we're too far away, or you think we're
319.
Okay, did you hit pre? So,
Do it one more time, actually.
yeah.
It's a good one.
Right, I'm gonna bring it out, man. I'll scoot up.
It could be a bubblegum coral with a really sort of translucent zoanthids on it, but I don't. My mind's wide open and spinning about what it could be here. I think it's a bubblegum. But it's a different growth morphology than I've seen before here.
The branches are really bumpy. Like even where the corals are.
Mm-hmm. The polyps.
The polyps
yeah.
are not. That's what makes me think it's a bubblegum.
Just a piece of it, whatever's easy to get here. Something like that. Yeah. Across the cutter would be great. Just little further deep, a little deeper in. A little deeper in. Yeah. See the polyps retract in?
Yeah.
So that's, I'm happy with that. Yeah. It's most certainly a bubblegum. It's great. Perfect.
Beautiful.
We got close to the oak, didn't we?
Summer haircut.
Yeah, just a trim. Okay, so that's 319.
I know what you do.
So it's going to 7.
Once I go into quiver 8.
Yeah. Alright.
And confirm that's 319.
Yes. Roger. Beautiful.
Wonderful. Awesome. Okay, the top of this ridge is supposed to be 1354 meters, and we're at 1354 meters.
Right.
So, before we leave, can we look at that Chrysogorgia, the top of that granite? Thank you. Just do it from here. We can just. You're gonna go get it. Go get it.
Oh, it's got some done.
Yeah, that's what I wanted to see. 32.
Yeah.
Yeah?
Just take a few shots.
Sure. Do you want,
How's yours?
you
What
want
do
the
you
camera? Do you
No,
want to?
I'm fine off the side.
All right, good. Can we come down a little bit with the light,
Yes.
please?
So it's-
You okay with that, Russ, if I go in?
No problem. You keep going. I'm just steady.
No, that's not good. Hold on. So this is a Chrysogorgia, Chrysogorgia coral with the pyrostylid crab, it looks like. Just going to try to get some, very hard to identify in video. Just want to see some of the characters. The diagnostic characters are actually on the underside often. There's a notch on the breastplate, if you will, and seeing the head here will really help a lot with the rostrum they have. So Russ is trying to fly the ROV. Well, I'm really tight and zoomed in here. This thing could maybe, 6 inches long, 8 inches long. Now let's take a look at this. I thought it might be one of those jellies. That's an amphipod above it. We often see shrimp or a different kind of crab in these Chrysogorgia corals. This is a different crab than we normally see. Yeah. Interesting. All right. Some anemones there too. Did you get it? All right, that's great, Russ. Thank you. That's good. $13.53. Making some notes here. So are we off the edge a little bit, you think? Or are we gonna?
Looks like it's dropping off now.
Yeah, I mean, I think should we turn left and come?
So we're at the top of this ridge.
Yeah.
So it would be going towards the ship.
Yes. That's our dogleg left.
Yeah.
Another Crinoid art.
More stock crinoids and bamboos.
Okay.
These were the images earlier, Tim, of that.
Yeah, so this is, Deb is showing me a picture of the Metallogorgia coral, which is a long central axis, pretty tall, with a little, what we call a poodle, it's very small polyps in a cluster. And it's got a single ophiuroid living on it, brittle star. And that brittle star, throughout most of the world, is a single species of Ophiuchreas. But here, in this equatorial Pacific, we seem to find this other species inside it. And so one of the things I'm interested in here on this cruise is finding that other species and seeing if the corals are actually different, their hosts are actually different or not, and trying to understand why it may be different here. Here's another bamboo whip. Just gonna do a small zoom here to look at the branch, and then we can keep going. That's great. Some bare skeleton there with not much on it. Normally there's barnacles and crinoids and other things on it, but not on that bare branch. Okay. So can we back out on the nav screen and see the waypoints? Get some estimate of distance or.
You did a new one.
I still have 640 from where we are to the last one.
640. So you can see there's some little depressions and dips there too, right? So we may come down shortly for a while and then go back up again. Yeah. Okay. That's great. So I think our next elevated point is 1328. So this was an unmapped seamount yesterday. So we mapped it earlier today, and now we're diving on it. We pick features that have high slopes. We've been going up, I think, 40-degree slopes today, 45-degree slopes, and we've come to the top of the ridge, and now we're turning more to the north, and coming down going along that ridge. We know that corals and sponges really like to have lots of current, and so they're often attach themselves to places that are on ridges and high angle slopes. There's some primnoids. There's a Brisingid sea star, the red thing on the right. Seeing other comatulids, crinoids in front of us. These are all things that we've seen already on this dive. So been a lot of crinoids on this dive. And when we came down through the water column, it was really soupy, really lots of particles and lots of siphonophores and squid, and it looked like it was really a highly productive, active place. And we thought there'd be a lot of things on the seafloor. And I feel like we've seen corals, there's always one in sight, I feel like, for the last couple of hours.
Yeah.
You know?
Yeah, when I came down to breakfast, I was, told that there wasn't as rich in abundance, I don't know about diversity, as one might expect from if your descent is a predictor of your site.
So this track line should have us go through very variable habitats, like you see more sediment now, and small rocks and some pavement here versus the large boulders and slabs we saw earlier, and the walls we saw really early on in the dive at 2,000 meters.
You move the ship?
We've made about 10 collections so far. Let me just check this. No, 12 collections so far. Come on. Nothing.
So you get the ship, take the couch, get away, so it's the same.
Oh, you're on the, yeah. You want to, on the sediment?
Yeah.
You can go down if you want.
Just use the sediment.
So we're just waiting for the ship to move. We are a single-bodied ROV, so the ROV has a tether on it that's connected to the ship to a winch, and we have to move in a dance. Ooh, there's a Chrysogorgia.
Yeah, with an associate.
I didn't see that with a chirosalic crab. Yeah, that'd be great. Yeah, let's look at that. Seeing these little floating siphonophore things. That might be it. Just went off to the right. I have not looked at this Chrysogorgia jet today. This is going to be an octocoral that has a shrimp in it. And you ready? Sorry, here we go. Sorry, this zoom is fast. Come on. Come on. All right. Do I have focus or you guys have it?
You should have it.
We're both twisting. There it goes. There we go.
Goes and turns that way. There you go.
This is fabulous. This is a sparsely branched Chrysogorgia coral that has a shrimp in it called Bathypulmonella. You can see the long arm right in front of it. You see the eye of the shrimp, and then go just to the right of the eye. There's this long arm. It's massively inflated.
Roger that.
Sometimes we can sample these shrimp if they are with the coral, if there's lots of branches and they stay huddled within those branches. I'm not sure how this would be. There's been some new species described recently, and I don't know how we can sample this. Maybe with a suction sample.
Suction sample is possible. They're usually
Probably the best
fairly
way.
quick.
What's that? They're quick.
They're quick.
They're very quick, and so you got to be really.
Very good, yeah.
But this is some great imagery. Hold on a second. We rarely get this kind of imagery because the branches are so sparse. That's amazing. It's going to hold this for a minute.
Taken by surprise.
That's fantastic. Ceratopalma is a common species. I'm just going to try to stay with it here. It's
He said I cannot. I said, push that.
Come on, there we go. There we go. This shrimp too is really relegated to living in coral species, in the branches, among the branches of these corals. What attracts them to these corals? What keeps them inside these corals? I have no idea. We will, if we can get a sample of this, we'll determine whether or not it's feeding on the coral in some way. So maybe they pick out of the polyps. The polyps collect organic material, and maybe they pick the polyps, or they picked it out of the mucus that comes on the coral. Wow. Hold on, this is a great view. Hold on, this is great.
That
I
arm
just don't get
imagery.
this. You got what?
You just see the big arm really well
You
from
do.
this angle.
You do. This is, and I think it may be gravid. It may be with eggs. Those purple things in the back,
Oh yeah.
in the abdomen, maybe eggs. Fantastic. Nobody move. Nobody move. That's just great. It's just such a rarity to see that. Oh, look at that. Oh, look at that. Yeah. Wait. That's okay. Sorry.
No, this is.
Yeah,
Like
sorry.
you said, you don't get to see this.
You don't. I'm sorry. This is just too good to see a profile like that. All right, great. Thank you. You got the camera. Holding my breath the whole time. All right, here we go. That was 1940, 1947.
There's actually one at the bottom as well. No, it's not what I want.
They are fast, though. Collecting them is really not easy.
Well, if I missed that one, there's one below it.
Is there?
Yeah.
Different shrimp?
These are different guys.
Yeah.
Okay, not that one.
Yeah, you can see. That's an, yeah, I won't talk now. They're going to sample.
Okay, man.
You want the one by the coral, right?
On the coral, yeah.
Just that shrimp.
It's that shrimp. Yeah.
Sure. Now. And. Wrong side. Now. Turn it off.
Alright, let's watch the jar.
The shrimp goes in the jar. Yeah,
Ah!
shrimp be man.
Outstanding, man. Both of you, great. That's unbelievable.
What
So
jar
you-
was that, Russ?
Yeah.
Not 5. Yeah, it's 5 actually.
It's 5,
Yeah,
alright.
just double checking.
Thank you. It's 320.
So you see that shrimp, that shows the difference. That shrimp only lives in that kind of Chrysogorgia coral versus
Right.
this is just a deep sea shrimp that's
Right.
found around the world, right? It's Nematochrysinus
So, keep it
down there.
shrimp.
Fantastic.
We have fast shrimp.
Do you know what else? Can we get us just a snippet of the coral and shrimp, please? Jar number what? That was
5. And it was sample of 320.
that was
Do you
F-
wanna?
that was FK 21
Nudge your head a little bit more.
320.
Yeah.
320.
Okay, there you go, thanks.
Do you want to associate this quiver with the same number, or do you want to give it a new
Oh,
number?
right.
You can just give it the next number.
That's fine. Yeah, I'll link them. Shrimp.
We'll link them in the notes too.
Yeah. So we sample this coral. We'll use that also with isotopes to look at if that shrimp is feeding on the coral. So we will look at them. It's one of the primary questions we have for this cruise.
Well, I may as well do it.
We have a lot of primary questions.
Doesn't preclude that as one of them.
I call all of our questions are primary.
Yeah,
That's
that's
what I
right.
just say. So when no one gets offended, well, they're all
And a
equally
broad
important.
range of interests.
They're all equal.
Alrighty, and so let's see what we got.
Great. Just make sure no one's slacking us.
Would you like an in situ picture before we take a snip?
I think we got it.
Okay, good.
I mean, we got, yeah.
How much we have?
Yeah. So, I'm not sure the angles here, but you know, something across like this.
Okay.
I mean, even here, if that's the narrow part, right? Maybe that's the best place to take it.
Alright. Okay, zoom it even back so you can see.
Yeah, I was thinking.
He's there. Yeah, you get that?
Sorry about the focus.
Sorry, you
Fully
said time
extended.
before that.
There it is.
Fully extended.
Really? Alright, off the shifts.
Yeah.
2 seconds.
I can zoom out if you want.
It's not easy to sit there.
All right, we'll try to make some tracks.
So.
Yep, that's great. Yeah, that's great. Come on, it'll come. I think you got it. Yeah, look at that.
Just takes a couple. It's quick.
Nice.
It's a little bit tougher though.
Yeah.
From last time.
Beautiful.
Fortunately, the camera right now.
That's a nice sample. It may try to stick to the. These can be sticky. Yeah. There you go. Nicely done.
This is how I'm labeling
That's
it.
great. Yeah. What are you saying? What are you calling that? 3?
21, and then I'm saying Chrysogorgia, the shrimp, and I'm listing the shrimp ID, the jar it's located in.
Beautiful. I got the same thing here. I'll do the same thing.
And it's quiver 7. Okay.
Farther left.
Great. And that'll show up on the printout, right?
Mm-hmm.
Like that.
Exactly.
Beautiful. Is somebody calling? I keep hearing that thing.
It sounds like an alarm.
Yeah.
Like,
Oh, nice job. Yes. That was really spectacular. Ben's good with that thing.
It's getting there, isn't it?
Huh?
It's learning.
Huh.
See,
Oh.
all the things we're teaching, you know.
It's the teaching.
I know, it's all teaching.
gravid shrimp on Father's Day.
Oh,
I
look
mean,
at that.
Circle of life here.
Circle of life.
Speaking of that, I doubt they're listening at the moment, but maybe one day in history. Happy Father's Day, Dad. Chris Gauthier, Henniker, New Hampshire.
Why don't you introduce who you are? Since how proud he is of you. So why don't you say who you are and what you're like, what you're doing?
My name's
You know?
Anna Gauthier. I'm finishing my PhD at Harvard and Boston University. And I'm interested in innate immunity and marine microbiology, especially that of the deep sea. So I'm just happy to be here for this journey along with everyone else on my team.
And this is your second ROV cruise, right?
This
Is that
is,
true?
yes. I was on the inaugural HIPAA cruise in 2017, and now we're back in 2021.
You called? I did. Hi. Go. Hey, Tim.
Hey, Randy.
How's it going?
Fantastic.
Okay.
We're moving now.
We are moving.
We're doing great.
Look
Things.
at that beautiful
Yeah, we
Crinoid.
need some
Should I
focus
just hear
here.
there were Father's Day messages going out? Can I give a couple too?
Oh, it's all yours. Absolutely.
Hi, everyone. I feel like we are so lucky to be exploring the oceans, and I am with Father Extraordinaire Tim Shanks, so happy Father's Day.
Thank you, man.
Who else is dad in this room? All right, well, my... What? What? No.
You want to see
I know.
that picnic on it?
Yeah, I do want to see the picnigonid.
Zoom in on the spider.
A huge shout out to my husband, Jeff, who's with our 2 kids at home. So happy Father's Day, Jeff. And to my dad, who is also helping to take care of the kids while I'm spending a month at sea. So the dads are stepping up. Happy Father's Day to both of them. To allow Mama to be at sea.
Whoa, look at it.
Whoa. That's a
It's
fast
rare,
moving
that is a
picnigonid.
rare thing to see, but we're also
Moving? You
helping
mean?
them maybe. I don't know.
I just want to watch it move for a while.
Yeah. This is a picnigonid spider. It's actually not a spider.
No.
That's what we
But
call
it's
it.
in
It
its
looks
own
like a spider.
family. It's Picnigonidae.
Yeah.
But we call it a sea spider, and you can certainly see why. It's very spider-esque, but it is, importantly, it's a corallivore but it seems to be more of a generalist. We've seen them eating anemones,
Yeah.
eating holothurians, so sea cucumbers. We've seen them eating corals, and a variety of different kinds of corals. I actually don't know if we only have one major species of Picnigonid out here. I have no idea of their
Don't know.
taxonomic
Yeah.
Diversity.
We've collected one now.
They
I
all,
don't know.
yeah, they all kind of look
Yeah.
similar to me in sort of size and coloration, but I don't know if anyone out there is a Picnigonid taxonomist and you want to tell me how many different kinds of Picnigonids there are.
Our depth is 1350 meters. That often helps taxonomists.
That does
know
sort
just-
of help.
it
Yeah,
does,
exactly.
it's something
We've seen a
to
couple
go on.
smaller ones actually, now that I say this, that are probably a different species, just because, I don't know, they could either be small ones of this species or maybe a different one, but
Yeah.
of this size class, they seem to look very similar with a sort of orangey pinkish coloration in the center, with the whitish body and the yellowish arms, or legs.
They have a really long proboscis they use to feed with too. It can be at half the size or half the length of one of those legs. They sort of inject that into the anemone or coral and feed from it.
Where does it all go though? I mean, look at them. There's nothing here.
I know.
Like in terms of, this must be. There's no opportunity really for stored energy in this creature.
No, I think the male carries the eggs and it's right underneath the central.
Oh, wait, is this about to start feeding for us? Wait. Can we just watch this thing for a minute? I thought it was about to sit down on that sponge.
See that long proboscis there.
What? Oh no, it's just gonna sit down. But, oh my god, wouldn't it be amazing if it just sat down and started to eat that sponge? We
Yeah.
could watch the behavior. Observing behaviors in the deep sea is really hard. Because often we don't see these animals moving so much, and we're only there for a few seconds. So this is pretty interesting footage here. Get the pun? Footage. But anyway, never
Foot?
mind.
Foot?
It's all feet.
It's all
legs.
feet.
Stop.
You mean that we're getting leggage?
I do mean luggage.
Yes, we're
Exactly.
getting lots of luggage.
All right.
Oh, I got you.
Just as long as you're with me.
Always with you, Randy. Oh goodness.
Luggage.
Yeah, we don't often see them walking a lot across the floor, or if we come across them, they're feeding, already. Interesting.
I bet those long legs are really adaptive for two reasons. This is just hypothesizing conjecture here, but one is that, just like a lot of these highly stocked organisms, it allows them to move through sediment drapes pretty efficiently without getting kind of quagmired in it. And two is it gives them the elevation they need so they can get height to basically descend their proboscis and feed efficiently from, you know, from high up so that they can target their polyp or their section. So, but still, I'm just looking. I've been thinking a lot about food resources and energy in the deep sea and, you know, energy storage, and this creature just has very little tissue, very low biomass.
Yeah, we see them on bamboo corals too.
Yeah.
Stalk bamboo corals.
We've seen them on black corals too.
Black corals. Yeah.
Anemones.
Yeah. Indiscriminate feeder.
Totally.
So.
I see. I suck. It's terrible.
All right, we good?
Yeah, we're good.
All right. Good rest, thanks. That's great.
Looks like it's picking up here. You're getting a couple more corals. It's been watching you guys.
Yeah,
You've
I'm
been-
just we have about a 600-meter line here
Yeah.
to go along.
I think it's worth doing.
No, absolutely. Let's go see.
It's hard bottom. It's
Let's go see.
we're along the top of this strait. Have you guys picked up any rocks?
Yes.
We did. Yep.
Oh, good.
No big rock and no rocks.
Oh, good.
I'm trying to read. It says rock
See the
there.
rock anemones on it, and there's a
Perfect.
Two rocks.
2 rocks.
They did. Yeah, they did quite a bit of searching at the beginning of that,
Ah,
so they got some stuff. But yeah.
just these look.
Go.
Yeah, when was the last time we picked up a rock? 'Cause these are looking like good rocks.
4 hours there. 3, 4 hours.
Yeah.
Well, should we happen to see a rounded, loose, accessible rock, feel free
I
to.
believe there's a few.
It could be.
We have to take a pick.
Right. These all look pretty consolidated. If we see a loose one, yeah. If one comes into view, just say, "Hey, that looks like a good rock," and let's get it. We've got Russ and Ben on the ROV pilot controls, and we have our fabulous MT Jimbo in the room. I always say this, and I never get tired of saying it because it's really true, and it's really important to acknowledge all 39 people on this ship who work to make this possible, as well as actually, frankly, a lot of people on shore who also work to make this possible. So there's a whole lot of people working to bring you this imagery of this beautiful stalked crinoid and a shrimp in the background, and of these incredibly cool volcanic rocks on this previously unmapped, uncharacterized seamount in the US EEZ, which we're calling the Far West, which
Far west, right.
it's the farthest west. I don't know if it's actually before I say that, I'm not sure if it's the farthest, but it is a far west seamount in the US EEZ, in the Howland and Baker area, where we were yesterday.
We were there just yesterday.
We were in Baker yesterday. No?
I don't. That's what.
We were.
You know.
I was
I think
there.
we dove.
It
We had
happened.
3 dives yesterday. I don't know where they.
Russ, were you there?
I'm just.
I think I was, yes.
You sure? I'm kind of having like a Wizard of Oz moment. Did this really happen?
No, it did happen, yeah.
It
Yeah,
happened?
we did.
Is there
2
YouTube
dives.
footage of it? Can we prove it? Is there data?
It's definitely in
There's
here
data.
somewhere.
I know it's. We run these expeditions and we're all so tired. We're running on very little sleep and we're running on caffeine.
Cat cats.
Yeah, caffeine, Kit Kats, but also genuine exploration excitement. I mean, this is where it's at.
I wonder if some of those are separated. So see them looking at the set camera there?
Yeah, yeah. I just love this landscape. These big bamboo whips. Yeah, let's take a look at one of those.
Dig in, yeah, see if something's loose.
We are rock hunting.
That's
Seems like a good place to go rock hunting, but it's to me deceptive. Even though there's rocks everywhere, they're not all easy to pull up.
So why do we want to rock?
Why do we want to rock?
Why do we want to rock?
Why don't we want to rock? No,
Well-
come on, we want to rock because we are collaborating with the U.S. Geological Survey in part to help characterize the mineral composition of these seamounts, which are about 80 to 90 million years old. And they're all volcanic in origin, but the basalts gets covered with a mineralized crust over time. Various seawater precipitate.
No, it's
Um,
joint.
No, it's totally.
I'm sorry, I'm gonna come back up.
Are you
And
sure?
yeah, and There's a cute little urchin, and looking at the mineralogy of the crust tells us a little bit about the volcanic history, seamount age,
Yeah.
water chemistry over time. I'm not a geologist, so that probably
Yeah.
tells us other things as well. Tim, help me
No,
out.
thank you. Hit the hot spots. Just perfect.
But
That explains why
but
we're
that's
looking
why
for
we
a rock.
will, we will get a rock and hand it over to our colleagues Kira and Amy at the U.S. Geological Survey, who undoubtedly know way more about what to do with a rock
Yeah.
than I do. I
That's
would,
it.
if you gave me a rock, all I would do is look at it under a microscope and scour it for interesting biological
Same.
reports.
Yeah.
Mhm.
No. It's a prestigious sunstar. With its arms up in the water to capture particles.
It's making a little net.
Lots of nice whips here on the edge of something.
The edge of the world.
This is.
No. Look at that. Wow. Look at a bunch of stuff in the background too. It looks like another metallic.
Yeah.
No, it looks.
Yeah.
That's a Chrysogorgia. And maybe another bamboo. And that looks like
Yeah.
a de-stalked stalked crinoid.
Which one?
The yellow stalk right in the middle.
Yes.
Or I guess a decapitated stalked. I don't know. I'm tired too. I can't.
Well, those.
I guess we are. Our rock hunting exploits are.
just
We're
give
still
you-
going.
Give you an overview of the area here. What we can. Let's see. Still looking. It's only seen one corallivory.
I know, and it was
Haven't seen any
and it was a
gastropods.
pretty phenomenal corallivory too, wasn't it?
It was.
It was more
No.
corallivore than coral.
Yeah.
That Paragorgia didn't stand a chance.
No. There's there's another one of those crabs in the Chrysogorgia. You can see in the background.
Yeah, I see it. Did you find my little jelly yet?
God, I thought I found jelly on a Chrysogorgia, but it wasn't.
They love a
So,
Chrysogorgia.
I know.
Is
Thought
that
I-
a
thought I had
halosaur?
it. No, it
Fish?
looks like a halosaur.
Fish,
See those,
fish.
rocks and sand? That looks like maybe.
You think these are loose here?
Yeah.
Yeah, either that or it looks like there's a whole field of loose ones to the right.
Yeah, I don't
Looks like a
know.
little bucket full.
This looks loose.
That's more accessible.
Ish, yeah. Or even those.
Yeah.
Mm-hmm.
You just didn't want to damage that coral, so.
Yeah.
These ones
Or
are-
over there. The whole.
Yeah.
Yeah, we can go for these ones. Yeah, no worries.
I mean, it's up to you. Grab the rock. You can.
Oh, wow. Yeah. I'll take, which one would you like?
What?
And which one?
I don't know.
I mean, these look like they're free.
We did.
I think okay.
So
Our
I'll go-
rock characteristics are round,
Yes. Which they are.
black.
Yeah.
think that's
Available.
Available.
This is
Available.
covering all the
Yeah.
Exactly.
The
So
manganese crust
it might be our ROV pilot's choice here.
But
But
it-
black rock.
I don't know. What do you think about? How do you feel about that?
yeah. There's a couple here.
Could be tricky. The round is better. Maximum
At least they're
blackness.
not mobile.
I mean, if you start them rolling, they'll avalanche.
I like the one in the middle. See that one that's slightly square but not square? You know what I mean? Slightly square.
I think
Yeah,
those
the one
over
with
there.
the ladies.
Yeah, those look really good.
Go for these.
Or the two in the bottom over there. We
Still
said
think-
pilot's
still think
choice.
they could be attached.
I agree. Oh no! Look at that.
Nice. See.
Beautiful.
Perfect size. It's black and round.
It's perfect.
Mm-hmm.
Russell's Rock. Where's it bends?
On Russell's Ridge.
Russell's Ridge, Russell's Rock.
I know, I would like to have a ridge. I'd bring it up to me.
I know. Yeah, I think you should.
I agree.
No, we could make it this way.
Russell's Ridge and Ben's Bridge. Look at that rock.
Wow. Here we go. It's got some iron oxide on it too. Some staining. Yeah, we'll get a nice image of it. It's in the car. There we go. Can we just image it here and spin it a little bit?
Yeah, we're going to swim around.
Excellent. Cool. All right, we'll get the dust off.
You good?
Yeah.
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 Is today Sunday? Yeah.
Today is Sunday.
Today's Father's Day. Right. So before we left, I sent cards from Hawaii to my
Nice.
husband. I don't know. He's gonna feel like my wife's at sea and I got a card. But honey, you're with the kids today. It's
Oh.
on Father's Day.
Wow.
Ooh, that's all crust.
That is all crust. That's interesting.
Well, so
There might be some rock inside that.
yeah, maybe.
Let's go for this one.
That one looks a little bigger, so a little more promising.
There must be. There must be a rock inside to nucleate the
Exactly.
reaction. Nice.
We could get the other arm out and play rock, paper, scissors.
Oh, Randy.
I'm in a weird
Too
mood
much
today.
sleep.
I'm sorry.
Too much sleep.
Just enough to get a terrible sense of humor. A little more sleep, and I'll have a good sense of humor again.
Alright, ready? So you see where it's been oxidized. And the sediment, really interesting. You can see the sediment line there. It's could be mostly.
Oh, they're on it.
Yeah, he's got it.
Absolutely.
Maybe the auto. Bizarre, sorry. I can try.
You want to shut it off? Turn it back on? That's the wrist one. You want to shut it off? Turn it on.
Yeah. There you go.
Oh, beautiful.
Nice. Okay, so this will be 322.
Yeah. 322. Yeah.
322.
Do you want me to know anything about the area it's taken from?
That's great.
No.
I got it. Thank you.
Oh, that's a very little rock compared to our other rocks. That's okay. That's a good rock.
That's a big rock.
It didn't
That's
look that
big.
big when we were picking it up initially, and then it grew from the sand.
I'm glad we don't have two big
They
rocks. We
came
gotta
out of
ship these
sand.
rocks when we're done. I like the little ones, but they like the big ones. I don't know. It's good to have a diversity of rock sizes.
Absolutely. That's right.
You can't have enough rocks.
I mean, you can actually have enough.
I like rocks.
We had too many rocks once. We were watching a volcano erupt on the seafloor live. Sounds strange to say that. And all the rock, it was
That's crazy.
it's going up into the water column as lava, if you will, and then solidifying it in mid-water column, and then dropping it back down to the earth as,
See that
as
in
rock.
Oregon? Tim?
What's that?
You see that in Oregon?
No, this was West Mata volcano
It's not.
near Fiji,
It's not that in Oregon.
and we had over 200 pounds of rock landed in the basket. We had to pull it out. So that was too much rock.
That was too much
That was
rock.
too
That
much
makes
rock.
sense.
But that was a great rock. Nice sample.
You know what? I used to give my kids when they were little to play with as bathtub toys. Is pumice. It was so
Oh,
great
yeah, right.
because you'd you know, it floats, and so it's the counterintuitiveness of, like, here you are, you give them a bunch of rocks to play with, and they think they're going to build blocks with them, and instead they all float. It's just
Yeah.
when they were like 2 and 3 years old, there was nothing more fun than pumice
great.
as a bath toy.
I got a bunch of pumice, but never took it in the bathtub.
Oh, it's so fun.
Mm.
So we used to. It's volcanic foam, right? It looks like a rock,
Mhm.
feels like a rock, but it's very light, and it's full of air, and so it floats
Yeah,
up.
it does. It's great.
It's great. So we're in the shores of Belize. There's tons of it, and we're out there all the time. And so I sometimes give them to parents as gifts for their 2-year-olds, and they're like, "She gave me a bunch of rocks," but then the magic of floating rocks in the bathtub kicks in.
Yeah. You want to zoom into some sand? I got it. This one. It's going in right here. This is fast, easy.
Whoa.
Sorry. I know. Hold on, everybody.
Yep. We get a good close up of the sediment too.
There we go.
It looks like they're 4M
Look at
tests
that.
again, or
Really?
maybe oolitic. It's good. What a difference.
Sort of uniform particles there.
So cool.
All right, coming up. All right, let's keep going on this ridge. Good stuff.
Rock and roll.
I feel like the
Go ahead.
the manganese coating has really gotten much more extensive.
How do you know?
You know, it's actually the patina. I feel like a thin veneer has a different patina than this. I feel like this is really thick, and you know, we just crushed that rock in a second
Yeah,
ago,
yeah.
or whatever it was, crushed,
at the
and you could see how thick it
Yeah.
was. So I think this is thicker here.
Interesting.
Yeah.
It's a pretty little sea pen. Peninsula.
They said they went to sample a sea pen early last night, and it sucked into the sand.
Oh, well then, let's.
Is that a sea pen? I don't know if sea pens can really suck down into the sand. Like worms do that, you know, other things do that, but
I don't know, but it
it's
looked like a sea pen.
fascinating. Did you see it?
No.
No. So yeah.
Last night,
Look
for the
forward to
first
go back.
time since this expedition began, for the most part, I passed out pretty hard. I just,
Yeah,
slept
good.
for 6 hours. It was amazing. I mean, don't get me
Yeah.
wrong. Like, I'm still sleep deprived, but
Yeah.
6 solid hours is life changing.
Yeah.
Mm-hmm. Like you said, just enough to make more bad jokes, like terrible ones. What is that?
Oh, that's a Persinket sea star. There's
No,
two of them here,
that's
right?
a crinoid on the bottom, I
That's
think.
probably
Just sort of a
or
strange
another-
imagery.
Yeah.
Every time we see these
Stalks here.
bunch of dead stalks, I'm left wondering cause of death.
I hear you. These are crinoids with little
You can
oaks
see
on
all
them.
the bases too, right? There's a lot of bases on these rocks. I've been noticing them.
Yeah.
You see that? Which sort of indicates that there used to be a much higher abundance of life here at some point. Falling star.
Someone had asked earlier about brittle stars, and they see them falling off the corals or something like that. So yeah, and they said that they thought that the lights were causing the ophiuroids to jump off the corals, and actually there's really seen one species, mostly a brittle star, that's not specific to any particular coral, but they appear to jump off when we approach, most likely due to the pressure wave of the vehicle.
That makes much
They
more
sense
sense.
they sense
Yeah.
pressure rather than light, and so we often see that, and we note it. You know, we know how far away the vehicle is from the ophiuroid, so we get a sense of the pressure wave. So absolutely, this vehicle is pushing water out in front of it and having an impact. But it's funny that many fish and things like that don't seem to be bothered at all by the pressure.
Not even a
by the
little bit.
pressure
It's
wave,
so
right?
funny.
So it makes you wonder what sensory adaptations they have. There's another Chrysogorgia. The last couple have had a crab in them.
That one has a crab too.
You see it?
Yeah, I do. Right there. Here, zoom
Yeah.
in.
You want to zoom in?
Well, if you want to see it, it's right in there.
Yeah, I do.
And then there's something else.
Help me out. Yeah,
That's something else. Can we go
there
take a
were
look?
anemones on there before.
I bet you that's what it is. It looks like it.
Looks like it. All that rust.
But you've got a crab in the top underneath the second whorl. It's just hard to see right now.
It's funny, depending on where you are, you get shrimp, you get crabs.
See?
You often see barnacles on available skeleton. So nothing at the base. You can see from here.
One of the great things about being out here is that when we take samples, we can bring them up and actually look at them in the lab,
that's
hold
right.
them in our
You got
hands,
Fly better than
look
I
at them
said.
under the microscope, really get a sense for it. Some of the things, this camera is amazing, and, I essentially use it like a microscope, but then there's a whole other level when you can actually dissect parts of these organisms and look at them carefully and really get a sense for how they work. It's awesome.
It's like flying a bus to find a star.
It's like what?
Flying a bus to find an M&M or something.
Certainly an anemone there, a ring anemone. Anemone? God.
Anemone.
Where's that crab?
It's right up there. You gotta go up to the second whorl.
Oh.
Second from the top.
Andy has Andy. Look at me, I'm
You need more sleep,
putting
buddy.
a Band-Aid over my mouth.
Randy.
Thank you.
Nice to meet you.
It's really
Randy
spotting things.
Broden. How you doing, Tim Chang?
Hey, Andy.
I'm sorry. Randy, what? What are you gonna make fun of me for now?
Andy Ocean. All right.
You gotta go up.
We gotta go up the second world. So we got the
Wait, so what
anemone
did you want
there.
to? What did you want to say? It was just I don't know. I just
can't
made fun.
remember anything.
Me neither.
All right, second world.
Second whorl.
Fantastic Magnospiralis. Right, maybe the species name of this. So it's
Yeah.
Chrysogorgia Magnospiralis.
It's a
which
little
makes
shrimpy.
sense, doesn't it?
That's not what I'm talking about. There's
Whoa,
your crab.
there it is. See?
Oh no, that's another anemone. I was wrong.
Yeah.
Andy says there's another crab on there.
Andy says there's another crab.
Just one.
All right.
Oh, that's
So
a
it
great
was
shot.
It's a great shot. It was a Europtychus crab before. Looks like it's the same morph here. This is a coral of the family Chrysogorgidae.
They're so pretty. If there's any lighting designers listening to this chat, I
Take it down a
want a
little bit.
light that looks like this, like a chandelier that looks like this.
A little bit
I don't know
more.
where I'd put it, but I just want one to be made in the world.
There we go. Nice, Ben.
It's just such a beautiful form.
Yep, please.
Gorgeous.
You're good. You're good.
There's something on the top of that stalk. Bamboo stalk.
They've been crinoids at the top.
Ah, makes sense. But that's looking a little more bally, like balled up. But. Alright, that's an anemone for sure, not my little jelly. Gorgeous.
Very similar to the last one, right?
Okay, but you think it's the same?
Crabs and anemones.
You think it's the same crab?
I think it's the same crab.
Okay, then we
Which is
know.
fabulous.
Good.
Now we know.
Now we know. Shall we motor?
2, 1. Yeah.
Go.
For Eric.
All right. Good.
That's just
good call.
he's got it. He's dialed in now. We should. I know. We have to.
Huh? Yeah, exactly.
This is perfect.
We have an incredible multimedia correspondent on board, Eric Holtzen. We are so lucky to have him. He is absolutely top-notch and astounding, and has been putting together some beautiful videos. So if you haven't checked out our videos yet on this cruise, you can go to our cruise webpage and check them out. I think they're on YouTube, but I think they're also just on the Schmidt Ocean Institute webpage associated with this cruise. If you look for updates, you'll see everybody's blogs, you'll see our videos. There's a new one coming out later on today. I'm so excited.
A lot of
Father's Day.
background write-ups about what we're doing out here.
Absolutely, but some of these shots are for him. I think we can go.
know.
But some of these
I just
shots
don't
we
want
hold
to.
for him so that he can include them in our videos because they are beautiful and also sort of tell the story of what we're doing out here. So check out the video, week 1 video. You can see Tim and I not joking around for once. We look serious in the video. And then there's a really beautiful one coming out for Father's Day about father's life at sea. So that'll be coming out, I think, later on today. Check it, or maybe tomorrow. So check for it. It's beautiful. It made me like tear up. Eric is really an amazing,
Yeah, I just saw a
amazing
minute of it.
artist.
That's really
Stunning.
outstanding. All right, Russ. It's stunning, right?
Yeah, I know.
I know.
But
All right.
we got exploring to do.
There they go.
Is that a crop?
He really- all right.
Let's go.
That takes some really some awesome flying right there.
Really
You
fancy
know,
flying.
Space shuttle, man, I tell you that.
That's amazing. He had it dialed in. That's really not
Soyuz.
easy to do. Soyuz. All right. Thank you.
All right.
Okay, so
So.
we can if we've seen it before, we're gonna keep going,
That's a good idea.
right? So we'll stop for things that are new. Ish.
Okay. Ish.
Okay.
Ish is my favorite word on this.
Oh, Andy.
Oh,
Yeah. Oh, God. I'm not even a hump day yet, am I?
No.
Right? This doesn't bode well.
Look. There's two ways to handle a cruise this long. This is a 34-day cruise. You can either get bitter and angry and upset, or you can just get a little silly and laugh, and I much prefer the latter.
Absolutely.
So
Absolutely.
I say
We've worked very hard to be here, and
we've worked really
it's
hard
a
to be
privilege,
here.
you know, to be here. And
Absolutely.
really grateful, and so we're making the most of it, and
But I have to say, you know, like, it's so, after a year of COVID, right, and more, it's just been so hard on everyone. It is so incredibly freeing to be on a ship and to be exploring and to be doing science in a really focused way. Even though we're staring at a screen, which we've all been doing for like a year and a half, you know, we're here physically in person with the pilots, with the ship, with the team on the ocean, and to be joking around with you. I can like kind of elbow you when I say
Well-
something stupid. You know, it's what a privilege to be able to do that in person instead of on Zoom.
we worked so hard to deal with COVID and whatnot. We even had a COVID test yesterday,
Yeah.
where we all were negative, and you know, we quarantined. We
We had to
did
change our
everything,
itinerary
and
because
we
of
did-
COVID. We had to do
you
a
know,
lot of things for COVID.
It was a lot of work.
But you know, I have to say that despite it all, with every challenge comes a new opportunity and a new, you know, if you take it with humor, even bad humor,
Yeah.
in stride, great things happen. And look, here we are, looking at this incredible bamboo whip on this very cool landscape. A halosaur swimming by, I think, on the seafloor.
Just looking at the terrain here, really is amazing to see this. This is below this carapace of black manganese and iron manganese are rocks that erupted. I don't know if it was 80 or 120 million years ago, or in that time
80
frame.
to 90 million years
you know,
ago, yeah.
something like that. Just astounding, and this subsidence must have happened, and then all the sediment you see has been raining down over thousands of years.
That, and I keep looking at all the coral bases. I think a student of mine, if you're listening, I'm not sure who it'll be, but is going to end up studying, just counting coral bases, just to figure out what the abundance that used to be here. You know, aging them. What?
Yeah, Russ said count the rocks.
No,
He's the other student to count the rocks.
not the rocks. The
One,
coral bases.
Two.
You have to know where the you have to get an idea for.
That's right. Well, that's right. How much area there is. So the density of the holdfasts of the coral.
Agree.
Right.
There you go.
We're not. But
That's it.
okay,
It's a
Russ, you're
PhD.
hired.
We
You can count the rocks.
We just came up with a PhD project.
No, but I do think that there's something really interesting about places which obviously used to support really high abundance of life, which now have much more sparse abundance. Or maybe it's not sparse. Maybe it was just this sparseness, but over time you accumulate pieces, right? And so those kinds of ideas are things to think about.
I mean, you can look at substrate like that, and here it is. You know, you say, why aren't there corals here? Well, here and here and here, because the rocks are there.
Yeah.
But
Let's
to
look
me,
at the
you know,
sponge.
what we don't look enough at is, you want to see the sponge?
I do.
So you
I want to
tell
look
it-
for.
sponge.
I want to look for spongivory. But go ahead.
But we don't really have much of local current information, and there are new current meters out that are small, you know, size of a canister, you know, 4 or 5 inches across, a foot and a half long, that you can deploy and leave, you know, for a year, and come back and pick up. And so it's the kind of data, we don't have physical oceanographic data on the scale of the species that we're seeing.
Why is
they
that?
disperse
Is it
their
just the
larvae.
technology really hard to be able to measure currents?
Yeah, I
out
mean,
here?
current meters can be major moorings. I mean, they're small, but you got to put down a major mooring to
It's a hexagonal.
and batteries, right? I mean, there's just
It's a pretty one. I wonder if there's shrimp inside. There probably are, but I don't see.
Right.
I don't see spongivory going on.
No.
Just wanted
There's
to
a little hole
look.
30 in there.
Nope.
No top on that one either. There's probably
Nope.
something living inside. What a matrix. Fantastic. It's a glass sponge.
Beautiful glass prison.
Yep.
That's what I always think about. Okay.
Okay.
Thank you, Russ. Thanks, Ben.
Yep.
Um,
I'm gonna come straighten up.
what's on those coin? Oh, well, it doesn't matter. Let's go.
I'm full out.
But you're right. Trying to think about why. I mean, this to
Thanks,
me,
Ben.
if I were a coral recruit, would be perfectly acceptable.
Well, you know, even with like the Gulf oil spill that did damage a lot of corals, deep water corals. You know, so how can you restore them? So we put out rocks
Uh-huh.
for them to land on. But there's already rocks out there.
There's plenty of rocks.
I don't know how you could put down slabs of carbonate. They're sort of their native rock.
You know,
or substrate, but
well, so then it becomes
What do you
questions
do?
of, is it a larval supply issue? There's just not enough larvae here.
right.
But, you know, there's enough stuff that settles here that's successful that it's
Yeah.
hard to make the argument that this is not a suitable habitat.
Right.
There's plenty of stuff that's doing just
Well,
fine.
But we just for substrate, but we don't know about the currents.
No, but
What
even
to
the
do over
things that
time.
are here, we've seen large corals, right? Some
Yeah,
really tall
yeah,
bamboo
yeah,
lips, and we've
yeah,
seen
yeah.
a high diversity of things, which suggests that things that are here are happy and they can support a really high diversity of fauna, and
Yeah,
yet
yeah.
it's just sparse. So
Well, look at what we had the other day when we were much shallower, and we saw just bands of abundant coral.
Right.
We go through Metallogorgia bands. We go through the Crinoid field.
right,
Right.
yeah,
So maybe those questions are more easily answered in the not mesophotic.
but in
just
the in the shallow deep,
yeah.
for lack
That's
of
it,
a better
right.
word.
And I know what you mean. That's what's so strange about this language.
I know.
We use. I know what you mean by shallow.
I'm glad you do. You don't know my
It's deep.
name,
It's-
but you
it's
know.
just- oh, Andy.
I'm just kidding.
Oh,
Oh, Ann.
God.
But you're exactly right. Right, the shallow deep it was really different. I'm so glad we had the chance to really look at that.
You know.
The insights from that are.
Well-
I keep replaying it in my head. I can't quite let it go. It was so spectacular.
The perspective it gives for then interpreting what you see deeper.
Yes.
You know, that's what I think really
Yes.
makes a difference. So we've seen this guy quite a bit today.
Yeah.
Another large Resinget sea star,
And there's
more
something else
crinoids.
big in the back here.
Feels like it's leveled out here, but
You know, I'm a shallow water biologist by training, and I still play in both the shallow coral reef ecosystems, in the open ocean, and in the deep sea. I'm one of those, I don't know what you want to hear. We'll call it an integrative biologists. But I work across multiple ecosystems and across multiple systems to try to think about this archipelago, the Phoenix Archipelago, and how it all works together. But one of the things about that I've realized is that when I'm hanging out shallow, I'm always curious about what's below me. And when I'm in the deep, I always want to know what's above me. And so I'm always trying to think about,
I'm good with this.
yeah, me too.
Yeah.
I'm always trying to think about the
Yeah.
whole system.
Sure.
Oh, swimming crinoid, or floating, sailing. I don't know what do you want to call that. He's
Say
just floating.
soaring, sailing.
Look at him.
Never.
He's just kind of like
I think he's just. I
gliding.
would say
Gliding. I'm going with gliding.
floating. I use a buoyancy reference. I don't know. But you're right. He's just. There's no current here, is there? Look,
He's just.
it may never come down.
Well,
It may never come down.
I mean, the crinoids
So-
can move and they're very cool when they swim, but this is just a gliding crinoid. You're right. The patina on those rocks is very different. I agree with you.
Right? You can. Yeah.
So there's a question. Is
What?
there an approximate amount of time you would expect coral bases to disappear? We don't know. Hard to study. Probably not. So for me, it's just an indication of the fact that.
Oh.
But you can look at. We don't really have lasers on the bases, which it would take a lot of time to do that. But if you did, you could at least get a sense of how big the bases are, which might give you a sense of how old the corals got to be before they died.
Surely that depends on the acidity of the waters.
Yeah.
Yeah, for
No,
sure.
absolutely. It depends on what water mass you're in, what
Yeah.
water properties there are for dissolution.
Actually, Steve's blog is all about that, because his paper is all about that.
Yeah.
It's great. So check out Steve's blog.
Is
Steve.
Chrysogorgia coming again? Okay.
Yep. Alright, I'm gonna let you and Anna get back to this, I think. Want me to stay?
Well,
Oh, I am.
come on. Oh, God.
I'll stay for a few more minutes.
All right. Okay.
I'll come back.
Feel free.
No.
Go.
If you don't mind, I'll say
No,
it was for
I don't-
your sake.
I don't. Can't tell the difference.
Fine.
So no crab on that one.
And no anemones.
You see one?
Yeah,
Yes, see one. Yeah. So.
I do. There's a another one in the back.
So I can take a moment just to say that hi to my two daughters,
Aww.
Emma and Callie, and they've certainly put up with me being out at sea for almost every holiday there is. So miss them greatly. I know they're out there. I know they're not watching. They're both working right now, probably in their jobs.
But you're such a good dad.
Well,
Every time
you
I
know-
talk to you about science, you're always like, "Well, I have to do this thing for my daughter too." I love the balance that you've been able to
Well,
strike. It's
I
amazing.
I don't know how. I think I've spent over 5 years at sea, if you add up all the time, and so I feel like I was really a remote father for a lot of this. And then, the RV pilots out here and the crew out here must experience the same thing, that they just don't get to spend as much time as they'd like. So it makes it really, really tough. So.
Yeah, but I've thought about that a lot because obviously in the same boat. But it's, kind of literally. But sorry. But the thing is that I think in some ways it makes us much more present parents when we're home with our kids. Because I just when you're away, you really appreciate the time that you have when you have it, and so I try not to leave too often, but sometimes we have to in order to do this incredible work. What a privilege to do it. And
Your kids are young, right?
They're
Your kids
little.
Yeah.
It's hard. It's actually hard. They're at that age now where they're old enough to understand that I'm leaving and really miss each other, right?
Yeah.
And they're young enough that it's really they're 6 and 9. Those are tough ages to leave.
Wow.
They're the
Yeah, I
cutest
remember doing
ages.
that.
Oh,
It was, I'm not going to pull out my better dad moments, but I was at sea 10 days after Callie was born.
oh my God.
The Galapagos Rift, and just have to. Yeah, you're right. You do value them so much more. They're the
Yeah.
loves of your life.
I had to skip 2 expeditions because I was one for each kid as I was nursing them. I just couldn't leave to go to sea.
Yeah.
Because
it's a tough.
just couldn't
and
leave.
I don't know how mothers do it. I don't know mothers really do like you've done, Randy, amazingly.
No, it's because
you
of the
give them
dads
so
who
much
help.
attention, and then you're also amazing in what you do in your career. It's
No,
just
you
astounding.
too. It's because of incredible husbands and incredible fathers, right, who really help out. It's Father's Day. I really, really want to. Here, I'll tell you a story, and I'll tell it publicly. This is crazy. I was working in Belize pretty soon after Zoe was born, and it was on a shallow water expedition where I was scuba diving, and I was still nursing Zoe, and Jeff came with me. My husband, Zoe's dad, came out with me, and I would go diving, and he would meet me at the dock with a bottle of water and the baby, and I would nurse Zoe, drink water. He would swap out my gear. He would take the baby, and I would go back underwater, and we just kind of flipped around like that, and it takes a lot for him he used his vacation time, right, to come out and enable what I needed to do and be with Zoe on shore while I was underwater. So amazing fathers out there who enable this to happen, and amazing fathers who go to sea who have kids who really appreciate the amazing work they're doing. It's really, parenting in this business is tricky, but it's possible because of incredible people who give support. Really. Yeah.
Well, my kids have been amazing. They've had to do a lot without their father around too, so it's pretty.
They turned
really
out
amazing.
pretty well, so you did something right,
yeah.
Tim.
I'm proud of them.
Really
They're
just-
great.
I bet they're listening. You could
No.
pull this clip and send
No.
it to them.
They just think Dad does sciencey stuff, and they go on and do their stuff.
Surely they know
Once-
that their dad is one of the premier deep sea
No,
biologists
no,
in the
no,
world,
no,
who's
no,
basically
doesn't
spent
matter.
had one of the most
No.
incredible
I'm
careers
just
out
the
there.
guy that when the car needs an oil change kind of thing. I'm not,
What?
Dad, can you help?
But that's actually so normal. It's so nice.
Anyway.
Here you're really. I don't mean to make you blush, but you're a giant in the field. But the fact that you're still dad, can you?
Oh,
You know, I
I'm
love
just
that.
so proud of them. One's going to graduate from Villanova this coming year with a degree in applied statistics, math.
Oh, wow.
Yeah, and the other is really interested in physical therapy and psychology at University of Delaware. And so doing really well. Freshman, just finished her freshman year. So they will
Said
do
like
well.
a very proud father.
I know,
love
I
it.
know.
It's so good.
So we're maneuvering the ROV with regard to the ship right now. We've come along the same track line here, and we have to keep a safe distance from the. We have a watch circle around the ship, given our tether. We don't want to run it into the.
Catnir?
No, we don't want to run them into the props. Thank you very
Okay.
much.
I'm like, what are you
God.
talking about?
I mean, I just dropped out completely. You're right. I was looking for another name for them.
Yeah.
But props. Okay, so we have to keep a safe distance. So that's what Russ is doing right now. He's keeping us safe, going slowly here, and watching the ship move further to the south.
Is that your daughter saying, I heard,
No,
I heard
and
you,
then my
Dad?
phone just went off. Yeah, let's see.
Rich, RV.
Eric goes.
I was convinced it was one of your daughters being like, I love you, Daddy.
No,
Happy
no, they're not
birthday."
watching.
Just to get ahead.
We are doing
I'll say that for them.
Okay.
Happy
Can you do
Father's
0.7?
Day.
What's the point?
Oh, it is Eric.
Yeah, no.
I can try.
Yeah, just to try and get ahead of you before the last one.
Yeah.
Thanks, fellas. Appreciate it.
So we've been seeing a lot of the same taxa here that we've seen for the last. Of course, we did come on top of the ridge, so we're at the same depth. We're not really seeing depth differences now,
No.
and we likely won't see much. I mean, our top depth is 1260.
Yeah, it's something
So
we should get there.
We're only
'cause-
60 meters from the top, and we're going to come down before that.
Right.
So I don't expect to see much in the way of different fauna here, as we saw on the trek upslope.
But I think that's part of the game. We gotta just check it out and see, because maybe not, but I feel like sometimes things change in a dime and I'm surprised, and sometimes things stay the same, and I'm
Yeah.
still trying to dial in. But I feel like we're, doing this day in and day out, some of the same depths. We're kind of getting a feel.
Yep.
The Phoenix Archipelago in 2017, so I've been working in this part of the world for like 15 years now. And if you had asked me in 2016 what was in the deep sea in this part of the world, I would have had to say, point blank, I have no clue, because nobody had ever been down
no
here.
surveys done, that's
Nothing,
true.
zero. And then in 2017, there was this like launch of exploration with the Okeanos Explorer and with the Falkor, and we had 25 dives south of the equator in the Phoenix Islands, and then a handful, I don't know, around 10, I think, between Okeanos Explorer and Nautilus up in the U.S. Phoenix Islands. And now here we are doing, and then Nautilus has come back a couple times, and now we're here doing another, whatever it is, 15 to 20 dives. And so by the time we're done, we'll have about 50 dives in this archipelago. And to go from we don't know anything to we've got 50 dives in a 10-island archipelago to try to dial in, that's a lot of information, especially for a very remote part of the world. I mean, we are in the equatorial Pacific. We are 0.3 degrees away from the dateline. We are a degree away from the equator. We haven't seen another ship for days and days and days. We're in the equatorial remote Pacific.
Yeah.
Yeah.
And we're close to the international date line too, right?
Yeah. 0.3 degrees away, I think. We were gonna get the whatever it is, purple porpoise, golden dragon.
Golden dragon is if you cross the equator at the international date line.
We can't do it.
We bestowed that now. It's
We got too
about
much work
60
to do.
miles away, but it's still
You could-
as close
you
as you
could-
get.
Well,
On most
right.
cruises, I mean, you don't
There
really.
just is not a Seamount conveniently located right there. You know what we should do? We should lobby the UN to change the dateline to where we are right
It's
now.
been done before.
I know.
Did you tell that story?
I did. Do you want me to tell it again?
Yeah, what was that story again? they changed the deadline.
Yeah. The former president of Kiribati, Ambassador Sito, current sitting UN ambassador, was the president who tried to, and successfully did, change the dateline because his country, the Republic of Kiribati, who I work closely with, was spread out over 3 archipelagos, but the dateline ran right through the country, and it was really a hard working situation. because there were only 3 business days
yeah.
where the whole country
That
was in
skeleton.
business, and somebody always had a weekend. So he lobbied to change the dateline, and did, and that's partly why you have this jog now through Western Samoa and American Samoa, different by 20 miles in 24 hours. But the whole of the Republic of Kiribati is now united into a single date, which makes a huge difference, because now the whole country can unifyingly work and share a weekend and share the same working days, which is really important. So we could move it again, just so that we get the Order of the Phoenix Dragon, or whatever it is.
No, the golden dragon is
Sorry.
Phoenix dragon. Yeah, another one's got a crab on it too.
Well, I'm just saying, if we're changing the dateline, we get to make up a new thing.
Less like a magnet to these things. It's beautiful. No,
It
it's
is beautiful.
Yeah. These are just, well, it would choose that form, right? This browse form to, all these corals want to extend their polyps out into the water to trap food particles, organic material. And so they have different forms of doing it. Some of them stand in a plane view, in a plane form, and some spiral like this. It's truly amazing.
Spiraling, I think, makes a really good web, right? A really good net. But what I also love about it is, if you think about tropical rainforest plants, a lot of them have the same spiraling formation, and they do it to avoid self-shading, so that they can actually each capture enough light as they spin around, and so that they can all get enough access to the sun. And I think this is kind of the same thing. Like, they kind of create, I assume, like a better net this way, but they also avoid being so stacked on top of each other, so that this way, that if they catch something.
Well, you know, there's one species that is similar to this from Chrysogorgia that actually has tighter whorls, and so I've often wondered if the different levels here indicate the spread you want to have to get more food particles.
Probably.
Do you want to be really close together in your spiral levels, or do you want to be further apart? And I don't know if they just evolved a way that.
I mean, it could just be a form of phenotypic plasticity, right?
Yeah, no, could be.
And so we see that in tropical shallow corals all the time. Where there can be plastic growth forms to accommodate for changes in current. We just saw it 2 days ago on Baker when we were in the shallows. You saw some acroporids and some favids and some other corals that had a really low profile to the current. And they're the same species as ones that can grow much larger, but they were just sort of plastically adapting to the really high current, really high wave environment of the windward side of the island.
Yeah. Is there a wait there, wait a second. You were porched out?
No, I'm just, the camera was right then,
I
so I
see.
can see out the bed.
I mean,
Yeah.
we won't miss stuff, but
Yeah. Yeah, let's do grab one from Noah before we leave, though. So remember, one
Roger
towards the end
that,
of the
we'll
dive.
get one, yeah.
Okay.
Bridge RV.
Alrighty, go ahead.
Yeah, Bella, we're gonna keep punching ahead.
Yep. So
Excellent. Do you like a 5 or a 0.7 speed ahead?
0.5 is good.
5 is.
46, 2
you know, we're not leaving the seafloor yet, but we've decided to make some tracks to try to get to a feature on the top of the ridge to see if it looks really different from this. So that we really want to continue to see this landscape, just so that we can look at it afterwards. But I don't think we need to zoom on every coral. We pretty much have a feel for what's here.
Right.
But if we see something interesting or different, we will certainly stop. And
We
sure
don't-
enough, here we have a change in landscape.
Right. But we knew there was going to be a depression here.
Right.
Based on the mapping
Sure.
we just did last night, and so we'll see how this transition of seafloor type changes the composition of the fauna.
Yep.
So we've just been in that Chrysogorgia area with those bamboo stalks, whips. almost like lobate lavas here, but when that crust comes on them, it's harder to.
It's very weird.
The other
Here's a
side.
big change in feature.
Yeah.
One thing we should mention while we've got a little time as we transition is that we are very, very proud and honored to be the very first expedition associated with Challenger 150, which is a newly adopted UN ocean action to help standardize data protocols and increase diversity. And we have a very diverse group of scientists here. We have a very international and diverse crew. We are doing our best to make all of our data public and to standardize our data types. And so we are, and you know, diversity takes on a lot of different forms, but we have really good gender balance. We have a diversity of religion and of sexual orientation, and we have a Pacific Islander, we have somebody from the University of Guam, we have people at all different stages of career. We're the old ones. But
Wouldn't mean we.
we.
Right.
Young face, young voice, but
Yeah.
yeah, I'm right up there with you. And we have everybody from a college freshman up to senior PIs, and I'm really proud of the fact that we have such a diverse group working here in the Pacific Remote Islands Marine National Monument, or I guess that's where we were. Now we're in the US EEZ in the Central Pacific. And we're really happy to be part of Challenger 150. We are the inaugural cruise for it, and it was just announced actually while we were at sea, and so we are really thrilled to be part of this initiative to help diversify and democratize the oceans.
It's also an effort really to standardize, coordinate crews and standardize our observations and data. I think we've been too much in the past. We'd have an expedition that would be known by those who went out and did it, and then not by others. And so it's trying to, I guess, inform ourselves, the scientific community, and others around the world about what we're doing scientifically on expeditions around the world.
And it makes total sense, you know. It's one of these things which I wish I had the time to engage in every expedition. They're all live streamed, right? Like I have the opportunity to do that, but the demands on our time are such that that's just an impossible thing to do. To really watch, especially not just one ship, right, but all the different ships, and integrate everybody else's science. And so finding a way to make the data accessible. Huh? Okay, is make it ingestible for each other is a really important enterprise.
Yeah. No, I'm glad you mentioned that, Randy. While we were talking, we've had a watch change up on the front row. We were the pilots. Russ will take off for a while, and we've gained Adam.
It looks like a face right now.
Hello, sir. How are you?
What?
Good morning.
Good morning. Oh, I see it.
We're looking to go down that line. I think the ship was doing 0.5 or something like that. We had just stepped up their speed. We're seeing the same things. We're trying to get to a higher.
You want to go along the line though?
Yeah.
Correct. This should be along the ridge, top of the ridge.
Yeah. And yeah, I think that's a good idea.
Right?
Yeah. Perfect. Happy Father's Day, Adam.
Thank you.
You're welcome.
Yeah.
Nope,
Yours is
not
fast.
father.
Yeah. Hers is every day.
What?
Mother's Day is every day.
No, Father's Day is too. Really, we don't get breakfast in bed every day. You know? We don't get cards and flowers every day. That does not happen.
Oh my gosh.
But we get our kids every day, and so do you. It's great.
Yes, on the bob box. You want to say? Yeah. Oh, yeah. I'm all the way out.
Are you gonna do a Happy Father's Day message,
Go ahead.
Andy?
Go for it.
Go, do it.
No, I already did mine. It's your turn.
Woohoo!
You got it? Good.
Happy Father's Day. There it is.
Nice job.
Beautiful. Anna and Constance were hard at work putting this on for you guys last night.
Constance changed the oil while she was there too.
Oh, she did?
Change the
No.
oil.
That was the joke.
Oh.
Oh, I see.
Bella called down from the bridge and asked Chair.
I was up there on the bridge when that happened, actually.
interesting that this fauna's gotten sparser. That on the slope up, it was much more. It wasn't highly abundant, but you rarely saw a frame that didn't have a coral in it.
Yeah,
so
yeah.
One in the front of you, one that off in
Well,
the distance.
that's true here too, but
Yeah, that's true.
now
Yeah,
it's
we
all,
are.
but
Yeah.
now it's all just primnoids. So it's just it's become less
Yeah,
diverse.
yeah.
It's not run out of Crinoids though.
Nope. A lot more sand here, so on the.
And there's another bamboo
on the
whip in the
rock,
back,
so-
right? So
Yeah, we're in it still.
yeah, agreed. Oh, Tim.
Yeah. So.
So.
This is 1324. Right. And what do we say it does? 1260 was
1260.
the top of the bump. So.
I mean, the top of this feature is like 1170 something. So, oh, here we go. Look, there's tons of corals now.
Here we go.
Tons. I mean, do we know
Well,
what those are?
we've
Have we
those-
seen that already?
Yeah, we've seen those.
Is that
We've
an
seen that one.
acanthogorgid, or is that a what is that?
There's-
I don't know.
See, we're on the edge here now, right?
Yeah, yeah,
Get a little more coral
yep.
on the edge.
You got it. Living on the
Yeah,
edge.
that's the acanthogorgid morph that we were looking
Right.
at this morning.
Living on the edge.
Heroes.
Come on.
I got it. Living on the edge. I
I'm
totally
just
get it.
saying.
I totally get it.
Why?
Totally
Beef.
get it. Totally get it.
You, now that I've had a little sleep, we need to get you a little sleep so
Nah,
we can
so I
do
can laugh
this.
at your jokes.
No, so that you can make better ones. You laugh pretty hard now, so that you can make better ones than I can. I'd like to laugh at your jokes. Can we do
I
that?
don't
Can
have
we
any.
split it?
I don't know if I have any. I'm just,
We need a role reversal here, Tim. If we're counting on my jokes, we're all in big trouble. They're terrible, just terrible.
Patchy.
Super patchy.
Can be patchy down here. That's the deep sea is riddled with patches, so it's on all kinds of scales.
So one thing we've been talking about is how Oh, is that a Victorgorgia? What is that?
Right.
Right there, to the right.
Right here?
What is that? I can't even see it.
Yes, so there's
No,
two
maybe not.
Victorgorgia morphs. Thanks, Adam.
Maybe not.
Yeah, I'll take it.
Maybe not. It just looked funny for a minute. I don't think it is anymore, but let's go look at it anyway. It just was, it caught me by surprise, but it looks kind of odd, so let's look at
Can
it.
you bring the light down a little bit? Thanks, Chris.
So trying to understand what's here versus what's not. So
That's another.
that's a very odd looking. What is going on there? Is it just tipped over?
No.
It's growing sideways.
Oh yeah, it's coming at us. That's all. Right?
What?
The whole fast is up there and it's growing
So, into the
parallel
page.
to the seafloor,
Yeah.
if you will. Right? And so, funny that the offshore doesn't take the posture of, unless the current's going that way.
Yeah.
It takes the posture with the coral.
Yeah.
Right?
That's a good observation.
It is a good observation.
Yeah, sure. That's a really good observation.
Zoom on that.
Look at that.
Chris, take it down a little bit more.
Is that an astroschema? It is, right?
Yeah. Oh, there's the hold fast at one
And this is like lateral.
Working on it. Working on it. Come on down. There we go. Zoom in a little bit more. So you can see the offshore had its arm snipped and it's growing it back.
Yep.
And
It's such a funny way to grow, but obviously it's been very successful this way. But you can see that the talk about phenotypic plasticity and changes in morphology, growth form, right? This coral has
the whole
curled
fact.
its arms up. What is that on the base? It's not an aplacophorine. What is it?
it's on the rock that way, but it grew out parallel to the seafloor, it looks like to me.
Tim,
That rock is loose, so you can the rock.
Could have.
Tim, what is that?
I don't know.
Is it a?
I'm all the way in.
I don't know what it is.
It's a vermiform two-eyed polychaete.
Purple people eater.
Oh, I'm doing the base.
Oh, I'm
So
sorry.
I'm in.
Yeah,
I'm
you
in.
see, do you see
Sorry.
that little
That must
little?
have been confusing for you.
Do you see the little wriggly
Yeah.
thing on the base?
Oh, I
That's
see
what we're
it
looking
now.
at.
Yeah.
Adam, is that slurpable?
Oh my gosh.
Uh huh.
What's that? The little worm face. The bottom? Yeah. You want it?
Corallivory?
I don't know. That's why I think it might be. Maybe it's. Look. All right. See, look at that. Look at that branch over there that's got nothing on it.
Yeah.
Can we? Hang on.
You're right. You almost got it. Keep coming.
Oh, I'm going the wrong way.
No,
Nope,
no,
I was going the right
no.
way.
No, closer to you, go left.
Okay.
If it's in manual, if we have control.
I don't think I have control. I'm getting nowhere.
Try that. Try this. No. Is it come? Yeah, there you go. All right, so there are polyps on that.
But not very many.
No.
And there's some exposed space down here, but that's where the
It looks
oof is. But
it all has the mesenchyme, the skin over it.
yeah,
right?
but it has the
Doesn't have
same
tissue growing over the top.
coloration as the coral, which says to me that it's either been eating it or it just it
Interesting,
has this. I don't know. Maybe it's just a coincidence.
Do you want to try to sort that?
I think so, and take a little piece. No, you don't.
That little vermiform thing is probably like.
All right,
try that.
fine,
No,
fine.
no,
We don't
no,
have to do
no.
it. I see. I don't have
You
any
don't know till you
I don't
try.
have any evidence of corallivory, so we should let it go. But I don't know what
You
that is.
sure?
Yes,
I mean,
we can let it go. There's damage, but that
wanna
thing's
look
in
at
the base, and all the damage is on the top.
I can just bump up and turn 90 to it, get like a long ways view real quick.
That would be great.
And then if you want to sample, that's probably better.
That's probably a good idea. Tim, can you zoom out for a second?
Roger that. I was trying to get the bare skeleton
I
there.
see it.
All right, ready?
Yep.
Come out.
What's that? Blue thing that's moving.
I don't know. Yes. I'll go left. You're still here?
I'm still here. I'm leaving though. After we look at this blue thing and this crazy worm thing.
Sorry.
All right.
Randy, you know, it's a shell.
All right.
It's a
I'm
gastropod
stopped.
shell.
It was blue.
Things
All
are
right.
rarely blue in the deep sea. There's no doubt about it.
Is that another picnic wounded right next to it?
No, that's an isopod.
Okay, fine.
Good catch. You got astounding.
What are you talking about?
Ability. No, I didn't even see that. I was looking at other stuff. I saw the isopod. That's great.
All right, now let's
so
go
what
look
do
at
I
that
do?
weird little thing, and then let's get out of here.
All right, you want Adam to move? Reposition.
Yeah.
Okay. Yep.
You only got one shot. No, it's not fun. I can get one as soon as I get home.
Oh, that I think is the Victorgorgia up there. I keep seeing it, but and maybe I'm wrong again. So we can pan up when we're done looking at this thing, and I think it's right there in line.
Yeah. So it just whether the rock fell over or
Yeah.
not, but I didn't think the rock fell over.
You do? You don't think?
I'll pass the mic.
For that to be
Yeah.
upright, you have to do a lot of moving
Make
of
an
that rock,
exception on the far end, but not early.
and for it to fall down like that.
Yeah. Before we zoom into that, can you just pan up for 1 second when they're doing their ready?
Can we pan up once they set up?
Yeah.
All right.
Try that.
Okay.
Mhm.
Ready?
Yep. Pan up though. Wrong way.
Oh, you want to pan up first?
Yeah.
Roger that.
And then I just want to see if it's there, just so I have a. Yep. See. Just so you know for after.
Yeah. So this is
Okay.
the
Yeah, just,
the
we're just gonna take a sample here and just transfer it, and then put you in a good position.
plexaurid form, I guess. Not the squatty form. I was calling it the squatty form earlier.
Have you seen Victor
We've
Gorgia on
we
this
have
dive
seen
already?
this, just
Okay.
on this several times.
All right. I didn't know that. Sorry.
Yeah, I know. I know.
Okay. Yeah.
It's good
There
to
it is.
document it though. It's got the same oaf on it,
Yep. Good.
right? Remember the squatty
Yeah.
morph that we saw
Mhm.
last time?
For sure.
That's what I was looking for on this dive. I didn't see it.
Okay. Well, then let's go take a look
There
at
we
this
go.
weird little whatever.
Well, stop camera. Alright, you want some lasers on?
I always want lasers
Show,
on.
I know you did. So this thing is.
Very tiny. I agree, we probably shouldn't take the time to surf it, but it's bizarro and missing.
And no longer there. No.
No,
is it
it's there. All right. Well. Okay, I'm done.
I think it's a worm.
I think it is too.
And that may be the head of the. At the
Yep, I
down
think
there.
it is too. A couple amphipods down there.
You want a suction tank?
I don't. I think I'm
Okay.
good.
All right.
Thank you.
And found the isopod, right?
Yeah, I see it.
I know.
What?
You see things before I do, so I don't have to. Okay, great. Adam.
So what's your theory on why it got knocked over? Where did it go that way?
Aliens. No, I'm just kidding. I'm just kidding.
I feel like it grew that way.
Yeah, I agree
I don't know
too.
where that rock would have come down from to do that.
But you see see
How
how
the
the
rock
branches
fall
are
over.
curled up? I think it's been that way for a long time.
Okay.
You know,
I like that. I think it's just stretched out. Like, oh yeah, like right here. You're
Mhm.
saying there's. Just
Yep.
started here. So it probably
So could it-
it
well,
started.
it could have fallen over
vertical,
now, right?
Fall over.
Mhm.
Now it's-
Exactly.
Where would the rock have tipped over from? In other words, how would it reposition itself? Did it fall off those rocks? Or did it just, maybe it tipped a little bit, is what you're thinking? Well, you know what the evidence is, see this oxidized
Yeah,
layer here?
exactly.
Yeah.
This was down more. I think you're
Yep.
right, I think it did tip over.
So something bumped into it.
Something
or-
bumped into it, maybe. Shark bumped into it.
Maybe it was that little worm.
Octopus. You think it was the little
worm
worm?
pushed the rock over.
Oh, M.
And
Do
then-
you want the worm?
No.
We're in a good position to grab it.
No, we're good.
No.
Thank you.
All right.
I'm gonna rock.
Nope.
A lot of rocks.
Yeah, we've got 3 rocks now.
I want to roll.
Oh, good answer.
Oh, stop.
That's one of your jokes. That was good, actually.
That was pretty bad.
I appreciate that one. That
I've
was
made
good.
it before, and that's why it's not even good.
Yeah.
But you forgot, so it's great.
What'd you say?
I didn't mean it like that. I just meant that we're sleep deprived. Tim.
All right, 13, 20
Oh,
meters.
Tim.
Here we go.
Yeah, just maybe just get her paws around behind her.
Let's see, 2111,
Yeah.
13, 20.
Bazingid.
Yeah.
Another Plexaurid. We just saw the Victorgorgia.
Question.
Okay, question. These rocks are all smooth and rounded, usually the result of running water, some other abrasion or glaciation. What do you think has shaped these rocks? That is a great question. We are not geologists. But these rocks were most likely most likely rounded because they're very old and they're covered by what Steve Askovich likes to call a crusty crust that has covered the original rock. And as these minerals are precipitated out of seawater, they've formed this crust that has surrounded the original rock, giving it this rounded shape, or this botryoidal shape, which kind of, yeah, so there's basalt underneath with the crust on top, and that's what gives them this rounded shape, we think. But that's what I got for you. Look at that boulder. You can really see what the waves and the currents have done around it by looking at the sand.
Yeah. The ripples.
Mm-hmm.
We've seen that. We've seen those.
What
Yep.
does the zip code stand
Another question?
What? They're playing trivia.
Oh, they are?
Yeah, he said,
They
what
love
does zip code stand for?
What is a zip?
What does it stand for?
Zippity.
for? Zone improvement plan.
Oh,
Never knew that.
zone improvement plan. Thank you, Chris.
That's a fun fact. Well, the trivia question right there.
All right.
My trivia.
What are there are different numbers and area codes. When you have low numbers or high numbers, what's that from?
When they were assigned?
Nope. I'll give you a hint. I only know this because I'm originally from New York.
Okay.
So when you had old rotary phones, it would take forever to go around, and so the most populous places that where people were dialing the most often had the shortest numbers. So you'd go like 212 or 112 or something like that if you had.
That's what mine's 904.
Right. So, but with an old rotary phone, it would be much faster.
Yes, we're just moving to your starboard and then we'll continue south.
west there.
Copy, okay, I'll be on the move and I'll make my way to starboard.
Thank
So we're just seeing a few sponges. It's all euplectellid sponges then.
Yeah.
Not seeing any coral rubble. per se,
Nothing.
right? So nothing.
But still some bases, right? So
Still some holdfast. Yeah, right. Thank you. But,
there's another one.
got a steeper slope here. We're at 13, 16 meters.
Climbing the mountain.
Heading for 1260.
What were the other answers? Sound out of project.
Normally when we have corals living up top, we get a preview of, because the dead coral and the rubble come down slope and fill in pockets and places, ledges and whatnot, and we're just not seeing that. So it's no indication we have a lot of corals up top.
But
But we haven't seen a lot of
look
rubble
at
here
this
in general.
landscape though. It's perfect. It's rocky. It's steep. It's pitted. It's got
You'd think that'd be currents coming
everything
through,
you
but
need.
We don't have a lot of current though. Today.
That's true.
Here comes a stalk. Here comes a...
But everything that's here clearly has enough food to be
Yeah,
fine
right,
and grow large.
That's good, yeah.
So maybe, but again, maybe it's larval supply. Maybe the lack of current isn't delivering enough larvae.
Get up here.
But there's enough individuals that I don't know. I don't understand.
Here's the Chrysogorgia again, doing its spiral thing.
Oh.
Adam's gonna peek at this. Oh, it's losing it.
Look for jellies.
It's lost a little whirl there.
Maybe it's just growing.
Yeah. And we've seen those. Haven't seen Chrysogorgia at all time. Or Metallogorgia. We're
Nope.
both deeper. Genera, I'm trying to.
Oh, there's another anemone on the top.
Yeah. Do you
But-
spot a crab?
Nope.
Nope.
Do you want me to look at it, or?
No, we're good.
We got some more. Are these are plexaurids now, right?
Chrysogorgias, plexaurids. No, there's also
There's that
Acanthogorgia.
acanthogorgia. Oh, there's a sea cucumber on the bottom.
Adam, I'm gonna do a 50% zoom here just quickly, if that's okay. You don't need
Snap
to do,
zoom.
You don't need to do anything.
Oh yeah, I think you're right. I think that's the acanthogorgia. I can't really tell, but
Look at the funny thing. Yeah,
okay.
I agree. Yeah.
Okay.
Just want to make sure.
Yep. Check.
Super.
Well, we're seeing more shrimps than we did before. Maybe that's suggestive of the change in habitat. Or maybe not. Maybe it's just a couple
I don't
shrimps in
know.
the frame.
I feel like we're climbing though. We're at 1309 now. We're just
We
going
are
up 6
climbing.
meters. No, we just came up 10 meters in a minute. 2117. It's 3.5 degrees C out there. An old stock with an orchard on it.
And a crinoid.
And a crinoid. Eagle eye. Eagle eye Andy.
It's amazing that we don't have like deep sea t-shirts with like team name, your team name in the back, or whatever, your sports name. I don't.
Yeah,
sports.
right. Sports name, right.
What would you
We
be?
should all be sports teams, you mean?
Exactly. What would
Oh
Team
my
ROV,
gosh,
Team
yeah,
Science?
right.
What would you be? What would
I
your
guess
name
I'd be
name in
Team
the back of the
Science.
t-shirt be?
I don't
No,
know.
I know, but what's
I don't know.
So, Tim and Randy, so.
Yeah. How we doing?
Obviously, we're flying.
suggestion that corals have bioluminescence when you, some corals do, when you disturb them.
We did a experiment on one of our guys where we shut down looking for.
That was on our
Was
cruise.
it? Okay.
We didn't see it though. You need really special cameras to see it. I don't know. Tim may have a better answer.
I have two quick answers. One is that as a smaller form, a larval form, they can have eyes that function. They migrate up in the water column and actually really 200 meters or whatever, catch light, eat up there. They come back down as adults. They still have the eyes, but they don't use them. Can't use them. The other answer is that a lot of these animals have eyes or eye stalks, but they don't have any capable vision because over evolutionary time, they've lost the ability to see, but they haven't lost the morphology. It's like we have an appendix, right? We don't use it, non-functional, but it's a holdover for something we used before. So it's just, it takes evolutionary selection to get rid of the appendix, right? They just haven't lost that yet. There's no big selection. Why would they have to lose their eye? It costs energy to lose an eye, right?
Is that because that maybe they were staying close to the surface at that point, or the water wasn't as deep?
Either at some form, right? So there's time scales you're talking about over the life of an individual fish, or shrimp, for example. They may migrate up to the water column, up high into the photic zone, and feed in some part of their life cycle as a larvae or juvenile. Then they come back down again. We've seen shrimp from 3,600 meters that live at vents at 200 meters depth as a larvae, right? So they come up with eyes and they use the eyes to feed, and then they come back down and colonize the deep sea habitat, and they have no functional eyes. So that's one model belief theory. The other is that they're born down here, they live down here, and they have eyes, but they don't ever use them. There are crayfish that live in the Texas caves, never seen light for generations, but yet they produce opsin pigment as if they had eyes. They still produce the proteins, right, for vision. So it's just a holdover from evolution. If that makes sense.
So it's possible that when we dive with our vehicle, we are giving them the gift of sight for the very first time with our lights.
The atom, you could look at it that way if you want to.
Burning out the last little bit of optical nerve that they
Well,
have.
I would think that if we were really affecting their vision, there would be a behavioral response, right?
I agree.
So it could be we don't see fish today because they're all don't like the lights, but as you know, as we get shallower, they do like the lights.
Yeah.
But I don't.
I'm saying more from Noah's.
What'd you say?
We're
Anna?
not from Noah's. She's thinking, yeah, we're looking for 'em. We're looking
Don't
for
be. We
'em.
have a bunch already if we need them.
They aren't. No, they should probably figure
We're looking for 'em.
Yeah, they'll do that GoPro down
All right,
the
here's another little plexaurid.
You get a good understanding of what like it seems silent, like the flying silent.
Same.
So Al, I think the plan is to motor to the top, see what we see.
So basically, you continue on past the end of the track, is that you mean? Or is
No.
the end of the track is the top?
Can we see the
track is our
next
little
waypoint
top,
on the screen?
And then,
Yeah.
so that's okay. Yeah, but it might be a little early. I don't know.
Those
It's
are
that
meters.
fast.
So we've got.
Yeah.
That's good.
So we're a good 500 meters away from the top.
220 miles from here to Titan. So you're looking at 21, 22 hours.
But I think we're gonna do a second dive. Here.
Yeah.
Okay. Yeah, got
I'm
it.
just saying.
But once we go.
Perfect. Thank you. Yep, that's good.
Just a quick
Thank you very much.
snap zoom.
We have an incredible captain and crew here on Falkor who have been really amazing at accommodating these
Good.
dives and helping to make everything work. It is a complicated dance, but we're
We're
very
good.
lucky to have a very adaptive, very flexible group of
Thank you.
people who recognize the value of being able to adapt a plan in real time based on what you see, which I think is really, we changed some plans in Baker and Howland.
Mollusk crab down there with
we
anemone?
go. And I think,
Sorry. Sorry, and Randy.
no, go ahead.
Sorry.
I just want to say thank you to them.
Yeah.
I did.
No, it's okay. I just wanted to recognize that sorry.
You're so cute. There's
That's an
another
ammonic
Crinoid.
crab with an anemone on the back.
Right, I'm gonna.
These are also associates on corals, so we like to document that they're here. I didn't know that they were at this depth here, at 1293 meters. What's that?
Can we
Do
look
you want
at,
to sample that?
I want to look at it.
And those
When you're
homo-
done with the crab.
Yeah, I'm done. The homologs will grab sponge, they'll grab coral, they'll grab, like you see there, anemone.
Is that an anemone or is it like a paragorgid wrapped into an
Where?
Crinoid? What is that?
Oh, on the mullet with the.
Oh no, I'm talking about this. So is that a paragorgid? Yes. Look at that. That's crazy. There's so much space everywhere, and they're instead.
And they jammed on each other.
they're- What are they doing?
It's true. And that's natural schema on that.
I know, but there's no
There's no
oaf.
oaf on that, yeah.
Do
There's
you
nothing at all, right?
want a
no.
oafless coral?
What do you...
Because it doesn't happen very often.
Got it. What?
Try that now. Was it not focusing very well?
It is. It is now. I feel like it's in focus. Is it?
It is.
Right.
You know?
It's an auto right now.
Yeah. It's an auto, I think.
Can you zoom
Maybe.
in to the polyps?
I can. Here I go.
There we go.
That's all in.
Well, it's the polyps are are downward facing on the crinoid. Probably.
Yeah.
Yeah. I don't know.
I don't see any abrasion by the. Well, see, maybe I do. See, is this abrasion here caused by the oaf?
Yep, I do. See
Right.
it. Eagle eyes.
I'm seeing more abrasion in the last couple of dives. Maybe impacting the coral.
There's a little shrimp in there too, or a little amphipod. You see its
Yeah.
red eyes.
I do. I see it.
That's crazy.
Shrimp.
You know, so again, when we're trying to figure out why everything is anywhere.
Yeah, I know. I can't.
Yeah.
So I don't know if that's a different paragorgia than we've seen before.
I don't know.
I don't know either. I don't know.
Let me check Slack. Should we sample it?
Just. Would you want the PRIMNM for your experiments?
Yeah, I think so. I think we could use another one, but we don't have to take. This Steve says.
Is he watching? Are the lasers on?
They are, yes.
Sorry, let me just come out a little bit then so you can see them.
Norella, or could be Polyptrophora with oh no, this is the previous one. Okay, sorry.
So these things are, excuse me, good gosh, tiny. Sorry guys, we're waiting on a call here.
See it?
So, I think they're quiverable.
Yeah. Do you want the oaf or not?
Well, if you want the for experiments, if you want the know what they would should take it.
Okay.
You're gonna want the polyps,
No,
right?
I don't want to take the whole thing because I don't want to leave.
Not
this
the whole
filter
thing.
eroded. No.
It's like really small.
No, it is small. He could try to cut here.
Okay.
Is that okay?
Yeah.
We're up.
Oh, the crabs will just pop over.
Roger.
All right. I am gonna
Take off. See ya. Thank you.
You're welcome.
That's great.
Bridge ROV. Well, we're gonna apology for just a minute.
Yep, I've just stayed on station while you guys stopped moving.
Look at you. Alright, thank you. Fuck that. Yeah. Which one?
The white one.
Yeah. So yeah, we have a question whether or not the ophiuroids are feeding on the coral, and we don't think so. We are answering that question and have been for. Yeah, our evidence so far and the data we have so far suggests they are not eating the coral. The one thing I just saw there was that maybe they are, with their arms and their spines, they're abrading the coral tissue so that they're creating a little bit of an injury to the coral. We don't normally see that. I've seen it on this cruise here. And so that could induce other things to settle, barnacles, crinoids, and hydroids that can do additional damage to a coral. That's why it's certainly of interest. The theory is that these ophiuroids are using these corals so that they can get up off the seafloor and take in organic particles into their mouths that are in their disc, and they use the coral to. Coral captures all these organic materials, and then the ophiuroid can just clean them off the coral, if that makes sense. So the coral could be, in part, taking food away from the coral, or cleaning the coral, or taking. These corals also have mucus that they produce, and the ophiuroid could be taking some of that mucus that's laden with protein, and using that. So. One of the questions we really do hope to answer, because some of these ophiuroids are very specific to the corals they're on, and there must be some partnership, coevolution that's happened as to why they're on these things. And so, just like many of the crab species here, we only find on certain coral species, and I've been looking at the morphology of the dactyls, the tips of the crab legs, and they actually are shaped in a way that matches the diameter and shape of the branches. So they're sort of designed to hold on to these corals, as the crabs are. So, how we doing? You ready? Oh yeah. You waiting on me?
Yeah.
Oh man. I was just.
Were you waiting on us?
Oh yeah.
Yeah?
Well, we're just letting you know.
No. Everybody's got a cup of coffee listening to me chatter on. Oh, that's nice. Okay. Yeah. So I don't know. I think it's big enough. I think it's small enough that we could cut it here. Does that make sense? To halfway or something like that, so that we get this.
You want like the right half, including
Yeah,
the,
including the oaf. Yeah. So that means maybe this branch here you'd have to get.
Yeah. And then
Quiver.
yeah. Or porch oak. Yeah.
What number are we on?
So I don't even know what number we're on. 323 should be our next sample number.
Maybe 5.
We also want to collect this Crinoid because we are actually doing respirometry experiments on board. And Anna, who's sitting next to me, is doing those experiments. And there's a Hamala crab in the background moving,
Oh, yeah.
right, taking off.
There's a little anemone.
And the Crinoid polyps are really good for these experiments.
Yes, they can last up to 48 hours, we've seen, when you keep them cool in the refrigerator, and they're still alive. So we can measure their use of oxygen, and the use of oxygen, or the rate of the use of oxygen, is a surrogate for measuring metabolism and also changes in metabolism. So if the oxygen is being used up quickly, indicates that there is a metabolic change. And finally, that metabolic change indicates that there is an immune response happening, and that's what I'm most interested at getting at. Last time in my studies, I looked at how mammals respond to deep sea bacteria, how their immune systems do, and now I would like to know how deep sea corals respond to the deep sea bacteria that we previously cultured from this area in 2017, and using a respirometer at sea with live deep sea corals allows me to answer that question. Additionally, the primnoids are also really great for this experiment because, as you can see, they have up to 4 to 8 polyps per whorl. That's a lot of surface area the bacteria can touch, whereas Chrysogorgids are a lot more wispy, and it's sometimes hard to get a measurement with the lack of surface area on some corals.
But I'm not taking the entire half, I'm just taking like the right half. Right, correct?
Thinking right
Yeah.
below the oaf, right? You
Yeah.
get the branch of the oaf's on.
It's just,
If you get a little
I
more,
want to come up from underneath it and grab it, but can't get the angle, so I'm just trying to figure out how to come up like this.
come up with a diagonal cut. You mean?
Yeah.
You're right.
Instead of like,
Hmm. There you go.
look at the from the side.
reason that we're trying not to take the whole thing?
We're not gonna take the whole thing, we're trying not to take the whole thing.
Why?
Randy said something about it. Did you come up from the bottom of that?
I can't do it.
That's a tough spot.
Well, it's just if
What-
I come in from a front angle, I could get half. If I do this angle, I wouldn't get to eat all of it, or
Right. So if,
so.
What about if you just come like around it back and snip up these three branches to regrow on this side, we'll take the whole top baseline.
more from the back?
And it came, might have a different view of what that looks like.
It's
Oh, I see.
No, I'm just saying like with the
Yeah.
shape of it.
if I come from here and down and up, but I don't have enough room to bring it down and crack it up. You know what I'm saying? I can't do it like that. Well, I might do it too, but
It's alright, if you gotta take more, then take more, I mean, whatever, you
Okay.
know, just do that, don't. There you go. They'll let go.
get off of there.
Nice.
That's,
Great,
yeah.
that's fine.
Okay.
That's totally fine. That's a tough angle. Nicely done. Really. It was
Not an easy
tricky
one.
one.
Yeah.
It's a tricky mouse.
Whole thing was
Well done. All right.
off we go.
Yep. Got about 500 meters to the top. And I'll check it again for the 1,000th time. It's 1260, so it's only 27 more meters. Sorry. 33 more meters in elevation. But about 600 meters.
2 more waypoints, is that right? We're not at one yet? Or we're about at the last second one?
Excuse me, yeah, that's we're just the next waypoint.
Where's your ROV?
About 500 meters to the next waypoint.
You can continue on the waypoint now.
Excellent. Are we gonna be on the move?
Yes.
You can continue on.
Excellent. I'll continue.
Is that same homolic crab?
Yeah, I think it is. So interesting that they do that they have those modified 2 legs that bring the anemone, the coral, the sponge, whatever, they clip off and they hold it over their heads and as a way of camouflage. But you know, how do you? It must have something to do with smell or scent or something. You know, some volatile, some chemical that the anemone will secrete that keeps other things away. I don't know. Just hypothesizing how they do it.
If you had to
Why they
say
do
what
it.
you see a majority of them holding, like is there any, there's anemone, there's sponge, and there's coral, do you feel like there's a preference towards anything?
You know, 60-40, maybe 70-30 corals over sponges. And then I would say that
We've seen more with
Yeah.
anemones than I expected than last time.
it's about 30% total, I think, with anemones. But you're right. On this cruise, we see mostly. I don't know. We've seen some coral. No.
Yeah, no, we've seen corals, we've
It's a
seen
healthy,
sponges.
a healthy portion of anemones. Yeah.
Saying that Adam's got holes.
Large crinoid looking over. Pneumatic carcinid shrimp we've been seeing on almost every dive. Long-legged shrimp. Yeah, not seeing too much here. Maybe come down a little bit. Let's see.
It's the land of the stocked crinoids.
They've been almost throughout the dive. Those proisocrinus. Haven't seen many fish. Is that a halosaur?
There's a cucumber. Let's see.
Yeah. So as we roll along here, we log a lot of the observations that we're making with the software we have on board, and can review them later and bring us back to. Ooh, what is that thing there? Oh, it's just a sponge on a rock. Or it's a coral. Sorry.
Is that a Chrysogorgia or Plexaurid, do you think?
Yeah, this is so tough without seeing the polyps close
That's
up.
okay. It kind of looks.
Can we look at this, Adam, for a second? And then when you're, can we take the lights down a little bit?
I'm just not gonna rock here a little bit.
Yeah, I think that's a good idea because This is something different.
That's a sea spider?
Yeah. I'm waiting for Randy for the door to open. Good to go? Yeah, thanks.
Bridge RV, we'll pause you there for a minute.
A little down on the lights, please. Okay. So we saw a sea spider, a picnic on it earlier in the dive. We imaged it and followed it along, and it was not on a coral, just walked across the seafloor. Now we come across one in a coral amongst a lot of ophiuroids. And there's two different ophiuroids on this coral. The question was whether it was Chrysogorgia or something else. And for me, I have to see the polyps to know, or to make a guess rather. Sorry, I'm guessing.
What is that?
Okay, so we have a ringet on the far left of a ring anemone that's facing
Okay.
the other way,
Oh.
right? And that has taken the polyps off of the skeleton. They land and we think they absorb the polyps. But you see it's growing between branches too, right?
Yeah.
So it's just sort of, it does that. It anchors itself really well. I'm waiting for Randy to,
We
I'm
both
waiting for
are.
the door to slam open to say, look at that spider feeding. So here are the ophiuroids. There's a astroschemid like right there below the anemone, and there's another one here low with spiky arms. It's an ophiacanthid. You see that's up in the far right too. So there's two astroschemids on here. I'm trying to do it. Yeah. So there's a picnic sea spider, another anemone above the spider. The proboscis of the spider, I don't know what it's in. It might, it's in the anemone. Okay, so the sea spider is feeding on the anemone. Let me go up here and get a picture of that. This will be, I don't know if,
Oh,
see that? You see that long, that really wide? I'm all the way in. Oh, no, I'm not. Here we go. Here's a close-up view of a Pentagon sea spider eating an anemone. And you can see the anemone has sort of been juiced. It's collapsed. I don't know how many days you see a Pentagon spider eat an anemone on a coral.
I know.
But this is your,
Are
if
those
you
its
like that,
eyes
it's
on
your
the
favorite
top
day
of
today.
its head?
What?
Are those eyes on the top of its head?
No, it doesn't. It
Doesn't have eyes,
eyes.
so.
No, it's just a rudiment. Yeah, we're just gonna hold on here for a second. Yeah.
Wow.
So I don't know. Let me just check we have a Slack channel on board where we communicate with all the scientists, and I'm gonna check to make sure it's anybody's. 10:49. Yeah, and Randy says. So we're gonna get a good image of this. I think we don't know how long these spiders feed. We don't know how clearly they have the proboscis stuck into that anemone. We've seen this before in the Phoenix Islands Archipelago. I think we had one observation of this before. I'm going to come back out a little bit and just frame it better.
I want to have a nightmare about that.
So if it did have eggs, it had eggs just behind this, I guess in the center part of the spider. You will see it. Pretty amazing. Agree with you.
Its legs are like the length of the whole body of the coral. It's amazing how
So it just found this coral, crawled up, found an anemone. You can see the anemone on the left versus one on the right.
Mhm.
So you see, but the anemone is pulled in tight, tried to prevent the proboscis from going in. And sorry, the anemone is not long for the world. Again, we have no idea the rate of feeding of these things. It's like part of the leg of the spider on the right hand side is taken off. And that's a different ophiuroid again. There's 3 ophiuroid types on this one, on the far bottom right. So I'm just going to frame this for a few seconds here. All right. Not hearing anything else, I'm going to assume we're good. We can move on. Oh, little fish come in.
Nice shot.
Just holding it.
Right.
All right. Come on. 5 more seconds. No, we're good.
You want a sample in here?
Nope. Nope. Confirmation. We're not going to need a sample, but it's a great observation. Yeah, I think that's a plexard. Sorry, I don't know
That's
if
what I
if
put.
I ever answered you. Okay, Adam, we're great.
I said,
That's cool. Very cool. I'll swing forward, swing front.
Go ahead, Bridge. Yes.
I think the other
we are.
anemone is still
You can go ahead
healthy.
and, carry on to the waypoint.
What? The
my impression was the other
On
anemone
the
was
left?
still alive, yeah.
Yeah, yeah,
There's
that's
its
fine.
tentacles out.
Not for long though. Anyway, yeah. All right. I didn't write it down. 1272.
That was pretty cool.
Yeah.
A lot of biology going on in one place. I think that's a Victor gorge? I can't tell. Or an Anthomastus in the right corner.
Anthomastus. Yeah. Anemone in front of us. This, you can really see the patina on this old manganese encrusted rock here. Looks super thick. Again, this happens over a great deal of time, so we've had, I don't know, 100 million years since this volcano was active.
Are those all hydroids?
Those would be all hydroids or bryozoans. Another isocrinus, crinoid.
You said number?
No, I'm good for going.
Okay.
Sorry. Sorry, we can keep going down the track, I think.
There's a Crinoid to the bottom right. Just go by.
There we go. See these old skeletons. Not too much of coral rubble today. Just a couple of down skeletons like this. I don't think we've seen any colonial scleractinians today, no hard coral colonials.
Those Persingent sea stars are just so crazy. Some of them their size. Or is that a crinoid? I can't.
Is that there?
Yeah. Can't tell.
I think it was a Persingent actually.
What causes like the cobweb-like
No.
appearance? Is that just like algae?
No, not at all. I think it's 4Ms do this. They build stacks like that, like little threads, and then they. I mean, most people would say it's marine snot. It's just organic material. I think it's also 4Ms building little. So, yeah, saw that earlier. I don't know if they collected one. They collected two sea pins. I think that might have been one of them. We're good on that.
Some up there on the left.
Yeah, I see it. Oh, they saw these two. This was the no, that's Anthomastus,
Yeah, it
isn't
looks
it?
like
I was
a
gonna say
giant
bushy
one.
Chrysogorgia, but it's not. Yeah, they saw that too. We keep a record of things that we see during the course of a dive, and we often, once we've seen them and make sure, maybe sample them, then we have representative voucher of what we see, and so we can move more quickly across the seafloor to find more things that we haven't seen here before. So in some ways it's cataloging, it's cataloging what we're seeing here. So we pass these big crinoids a lot. Depth is 1,258 meters. It's a pretty good rise right there.
We made some good tracks.
What's that?
So we're making some good tracks.
Well, I hope so. Yeah, we were trying to get to here by the
It's a nice
midday
little
or so.
sponge.
So another anthomastus. We're seeing more anthomastus here.
Yeah, they're popping up. Wonder what changed.
It's a vase sponge. That's a glass sponge. Two of them. Usually it's getting more sort of, well, these are collapsed, really old collapsed pits here. And the lava cools, it
Here for example.
becomes more heavy and under its own weight, it'll collapse and break. So that may have been what happened there. It's like undulating collapsed pits that we're going over. What's that, crinoid? Yeah. And these crinoids are everywhere. You know, they really are gorgeous to look at. They have lots of animals living on them too. Worms and even some parasites, some crustacean parasites. Sorry, I gotta type something in here. If you see something. When was our Niskin sample?
Oh, up, looks red. Just likely.
Sort of going back down again a little bit here. But there is sort of a pit before we come up to the tip top.
You see where I'm facing to the
Yeah.
south there? There's looks like there's another ridge.
Yeah. So that
So
next.
if you look on those views, so like Memorial Cooking Store. Dark map. So yours is different than mine too.
Mine just looks dirty.
So on top of this ridge, it actually has some higher points. We'll go upslope a little bit, then down into a depression, and we'll go up back up again. And I think we're ending on the sort of the highest part of this ridge at the end. Again, at 1,260 meters. Yeah, 1,260. Which is we're at 1250. We're 1257 now. So we're on the. We came up. What is that?
Oh.
It's a rat tail. We came up to.
Size.
Yeah. So there are multiple little
Pretty awesome. Fish.
highs here, local highs. Okay. So
You're giving the gift of light.
that's a chimerid. Oh, with the light. That fish likely fills the pressure wave for the vehicle. It's turning away. That is these chimerids are really beautiful. Gracious. they have such large eyes. Right? They would be maximize surface area for visualizing light, and maybe they do at some point in their life cycle. They go up higher, but this is a typical depth we see them at. I've seen them down at 2,500 meters, 2,600 meters. They're likely going deeper than that. There's another yellow coral that looks familiar. What we've seen already? Yeah, we've seen that one. It's got the ultralight on it. Yep. So another primnoid off to the left here. Looks like. Very large crinoid here. Let me make sure I check my schedule. So what else? You got the primnoid, right? Did you want? So we're
That was it.
pretty good now with that. Okay.
Yeah, I think between the other ones you got earlier and that one. Yeah, what is that?
It's a Chrysogorgia. That's Chrysogorgia with a bunch of stuff. Well, we've seen this too. Numerous oaks on each of the branches and crinoids at the top.
Is
Big
that a type of
percentage
PRIMNM?
What's that?
Or a black?
Chrysogorgia?
Yeah.
It should be right. Yeah, it should be a PRIMNM. You know that they had that designated somewhere. No. Okay. We normally don't make track lines that go from shallower down to deeper like this downslope because you see less. It's easier if we come upslope.
Oh, there's plenty of PRIMNMs, yeah.
Yeah. But for ROV ops, probably also helps to go upslope. So
Yeah, for lighting.
that's the preferred way to go.
Looks like it's not bad.
No. So we may have track lines once in a while that we design that do go down slope a little bit and then
Is that
come back up.
Chrysogorgia?
What's that?
I can't tell.
It's eerie, bro. Lights.
See more sponges here. This is a sort of an
It's a-
Chrysogorgia that's has some
Maybe a young
damage
one?
per se, but it looks like it has some problems. What's that?
Oh, I was gonna say, is it young?
Could be. Could be.
It's nice fat glass
there you go.
sponges.
Yep. Crinoid, glass sponges. More stalk crinoid.
And it looks sort
Okay. Actually, can we take a Niskin?
Right here?
Yep.
That?
Yep. So we're gonna pop a Niskin bottle here. We're gonna sample water. We just passed over a Chrysogorgia, which is something would be interesting to see if we can detect with environmental DNA sampling. So let's take a Niskin. Now it's 2206.
One is the 2, 3, 4.
I think, yeah, one has been shot. Number one.
Alright. You want a 5?
Roger that.
Liter? Alright.
Yes, please. In the notes, please put in Plexar.
My phone's about to keep in the pocket.
So Plexards, Chrysogorgia.
Maybe it's good.
Two's good?
Yep.
324. That's correct.
324. 324. Depth was 1275. It was 1274 meters. You seen anything else? Oh, yeah, okay.
Chrysogorgia.
So like,
Can't believe it.
okay. Got that, 324. Oh. More of a pavement here. There's one coral down there, but seen that Chrysogorgia over quite a bit of depth too. I'd go back and look, but that was with the same associates on it. Wow.
How we doing?
Good.
Yeah, how you doing?
Good. Doing
Happy
great.
to keep going up here?
What's that? Yeah.
What's your target depth?
I had 1260. You know,
We've
but it's-
gone past
we've
it,
gone
under
up-
it.
Yeah, we've gone below it, over it, right?
Yeah, past and under, I guess they're the same thing.
I don't know what that means. We're 1270 now, so it just means we're gonna dip, gonna come up.
Yeah, like, you look on my map, this looks like
Where?
a ridge, so we kinda went down this little dip here, from the ridge curve around, we just kinda went down, and then this,
Down.
I think this is,
Should be coming up then.
should be coming up.
Yeah, right. Okay. There's a little, I think there's a rock pen there.
I missed that.
The two species of Crinoid we've been seeing today. I think it's just two. I don't know, it could be more, but to my eye, I think there's about two more. A little more vertical relief here with the rocks. Good lord, corals down in little,
Look at that.
these little
wall.
scallops. There's these little corals in here. We've been seeing those though. Kind of a smaller Chrysogorgia on top of this. We're running along a ridge now. 1259 meters. To 12. Okay. We're looking for things we haven't seen yet today. Running a little transect up here, 1254 meters. So we are indeed coming up.
Yeah, we're about getting close to well, no. ROV is not where the ship is. I keep looking at the ship.
Yeah.
Yeah, we're gonna follow this ridge, it's not really on a track per se.
terrain has certainly changed, though.
Being a follower. I'm not trying to follow this, I'm trying to follow Rich. But it looks very similar, very similar.
It's almost no current.
It's a little bit on the nose, but like very, just a little bit. When I noticed when we were doing that last sample, you could just see the snow and
Color right at us.
Right.
This reminds me of Star Wars. All of a sudden, I don't know why it always should with the, well, with the snow, like or whatever coming
Oh,
at you.
no, seriously.
Like,
Oh my
me.
gosh. Oh my gosh. So if we do get to our our end waypoint, which was about 1, 2, 3, I don't know, it's 400 meters away, we will. We don't see any other additional new taxa or things of interest to stop for. We will recover the ROV. We will then go and do some more mapping with neighboring Seamount for maybe 5 hours, and go back in the water and dive on that Seamount. Little shallow collapses here. Looks like to me. Too much more sort of volcanic. I can almost see flows. A couple of sea stars on the seafloor. Maybe I'll come down a little bit. Some crazy morphologies. Sort of like lobate lavas here.
That waypoint right there. Yes.
No, that's what I'm saying is I don't.
Maybe a slight current coming from the north.
Maybe got moved. This looks like it's kind of oh, you still got more of a ridge up there. Yeah, which direction are you facing?
This is the way where you're going.
Oh yeah, so there's
There's more range. You can go all the way. You go another 800 meters, but
Well, let's not get silly.
let's not get silly. Past my last one, but now it's
yeah, this might be where it was the end was
What do you mean?
Start.
yeah, this way I got moved this time.
I'm just saying, yeah, the last one that we had in here that Steve drew on was here.
So that's
Starting
where
to
we're at
go
right
back
now.
down, yeah.
Oh, this is kind of the top then?
Yeah.
Well,
there's not.
there's
one more top.
The waypoints don't go to the top.
There's more top, but it's
Yes.
way far down.
Yes.
Right.
Right. We were stopping at sort of a reachable high.
Yeah.
This is.
So is that indeed a high?
Here, hold on, I'll show you.
Thanks, Debbie.
It's too far away.
Tell you what, shoot me your last marker.
There, see, this is.
So that's right where we're at.
That's where we're at.
Yeah.
So.
What do you get?
That last one for me, Adam, was 1.00. So, and 179.1.
This one is not decimal place.
It's just a little off. I'm just a little past it, so.
We're a little past it, yeah.
We're past the final
which is
waypoint?
fine, but like, yeah. So this is where we are, Tim. We've gone up over this bump.
Yeah. Yeah.
Now we're starting to come back down.
Yeah.
And like the very top is way up there, but that's like 800 meters away.
That's C right here.
Yeah, that's
It's all up here in red.
yeah. We weren't
That's
thinking
like
about that. We're trying to go to the local high on that reefs that we had been on.
Yeah, we've been
So
off
we've
more
done
often.
we've done that. We've done
Yeah.
it.
Wanna
Alright,
go?
I'm gonna go chat with Randy.
Yeah, sounds good.
Roger. You good? You just keep on rolling?
Sure. And it gets the hot seat.
What?
You get the hot seat?
Oh, I guess. It is more comfy. I'll take it. And it is hot. Basically.
Where do we go? What do you wanna do?
We're waiting for Tim to discuss with Randy, but
No,
I.
but
You're in
Where do
charge.
you-
You're
Yeah,
not
so
a
So
seat.
where do you wanna go? What do you wanna do?
Oh. Just keep going. Just keep swimming. Go on this ridge, I guess. Personally, I think it's time to end the dive, but we'll see if I'm right. Hello.
Help me?
Tim find you?
No.
Oh, I like your shirt.
Why didn't you WhatsApp me or Slack me?
I don't know, he said he was going to the library to get you.
I wasn't in the library.
Oh, okay. I'll go grab him.
That's okay.
Probably looking
I'm
at you
here.
then.
Okay.
Well, hang on. I'll get Tim.
It's, I think we passed the last waypoint.
Oh, then
So
we
that's it. So we wanted to know if it's okay to end
I
the dive.
think it's okay. Let me go
His
find.
phone is sitting in front of you.
Yeah, his
What?
phone's right
His
here.
phone is sitting
Okay.
in front of
Okay.
you.
So I can't text him.
Yeah.
Okay.
He was trying to sit.
Okay. Go get
Thanks,
Tim.
Dabs, I totally forgot that. That's why
Sorry.
staring
I was
at
like, I'm
that.
trying to text him. What?
I don't know. So
I
we
don't
just
know.
sit that on this.
So we got to the last waypoint and more of the same? More of the same. All right,
ready to
well,
go up.
it's good. We've characterized this section of seafloor.
Here you go,
I
Tim.
think we could call that a wrap.
So it's 11:20. Is that okay?
Fine with me. Is it okay with you?
Yeah, it was still 1200, so it will be not to the surface till an hour
Are we gonna enter after lunch, like lunch hour for you guys, or? are we okay?
They'll come in. Yeah, they'll come in.
Okay, I just wanna make sure everyone gets a chance to eat,
Yeah.
especially on Father's Day, everyone gets taken care of. Okay, you guys are really good? Okay. All right, in that case, thank you all for joining us. We're gonna be back on this general feature, but on a sort of eastern.
Yeah,
What?
just not here.
Yeah, Eastern.
I don't know, not really eastern. Another feature of
We are going
very
to
close.
call the
I don't
dive
even know the
here and
distance.
return to surface.
In just a few hours. So we're going to do about 4 or 5 hours of mapping and then descend back down. So we will see you shortly. Happy Father's Day. You get 2 dives today, all the dads out there. This is for you. Woohoo!